Recording in progress.
You don't
Oh,
have
okay, let's
permission to
see.
play. What is that?
Ah,
Mm.
oh, good. I'm glad we're doing this. It looks like there's a couple of videos in here that require access.
Yeah.
Actually, maybe if you all don't mind messaging whoever is doing slide 321, for them to make that video accessible to all, because I may not be driving also for those slides when we get
Yeah.
there. So,
Okay, but it's maybe towards the end, so we can also have the lunch break to resolve this. So, but
okay, perfect.
we make
Great.
a note.
Okay. Thank you for catching that, though, Georg. I'm just going to message Slack quickly as well and let everybody know that we're about to start.
Yeah.
Yeah, Alireza. Sound check?
Yes, so I was checking the slides, and one of the slides had transition between each step. So when you click next, it would the next text would appear instead of going to the next slide. So I wanted to just share this.
Sorry, what's going on?
Yes, so one of the slides use transition inside it, and because of that, when you click next, instead of going to the next slide, it would show
Got it.
their
You can see
transition.
Yeah, that's fine. Just tell me next, and then I'll just
Yes.
click forward. Great.
All right, thank you. Yeah, I was also going to do the finishing touch of my 3 slides. So basically, thank you for, having allowed access again. Yeah.
Okay, no problems. Everybody finished? Shall we close it again?
Oh no, I just so that you have, I'm just going to do the finishing touch now, and then basically I'm going to be ready. Sorry, I didn't realize you had like,
Okay.
turn on the access again. Sorry.
Okay, cool.
So I'm gonna refresh the slide deck in 1 minute, and then
Yeah, Georg, I think you should close it.
well, Inger, if he's editing it right now, it's okay.
Oh, okay.
I'm gonna start the recording. Okay, friends.
Recording in progress.
It is that time. It is 9 o'clock Eastern time. We are about to begin an epic marathon of How to Grow Madness. We have never had this many presentations and this long of this many hours of continuous How to Grow magic, but I could not be more excited. And so how this is going to go for those that are joining just now, okay? I am driving, okay? So I'm going to share my screen right now. So this is the magical slide deck that all of us are in. And I'm actually about to hit reload on the slide deck right now. And so unless there's like an emergency of some kind, I'm not going to reload this deck again until we take our break. So once we get going in about 3 minutes, we are going to go continuously until 12:30 Eastern time. So for almost 3.5 hours, we are going to go straight. I got my coffee. I'm ready. And I also went to the bathroom, so I'm ready not to move for the next 3.5 hours. And then we're going to take a break for an hour and a half for lunch. Okay, so we will take a break. And so for those that are just joining, in a moment, I'm going to hit play on the slideshow. Okay, and what's going to happen is we're, of course, going to go in order. Everybody that's presenting, you're going to tell me when to move to the next slide, or if there's an animation, you can just say next, and I will hit the forward arrow to advance either an animation or go to the next slide. Okay. The other thing, Georg is going to be our timekeeper. And of course, again, just so much deep appreciation to all the TAs. We're going to do a little gratitude at each session. And we're going to advance to the next slide. And then at 3 minutes, Georg, a timer is going to go off from Georg's screen. It's going to make a little noise. And at that point, you basically, here's the timer, here it goes. And then you can hear the noise that's going to happen. And there it is. That's your noise. Okay. That happens at 3 minutes. That is your sign to start wrapping up. 3 minutes is going to go fast, everyone. So this is going to be a big, we're all going to have a big exercise in speaking quickly. But when that alarm goes off, you have another 30 seconds or so to wrap up. If you get to 3 minutes and 30 seconds, I'm going to give you a little nudge that says, hey, really wrap up. And at 4 minutes, no matter what's going on, I'm going to interrupt you and stop you. Okay. So 4 minutes, you're going to get interrupted. And then we're going to have a minute of reaction or a question from a TA. And then we're going to move on. All right. And we are going to do this for 3.5 hours. And it's just going to be how it's going to go. All right. So we're going to keep everybody on time. So I hope you all practice your talks to try to keep them short. The other thing I want to mention, okay. So at 12:30 Eastern time, we're going to take our first break. We're going to break for an hour and a half, and we'll be back at 2 o'clock Eastern time. And we basically have 3 sessions starting at 2. So from 2 to 4, from 4 to 6, and then from 6 to 7:30. Okay, so there's 3 seconds. There's probably not going to be a break. There may be a break depending on if we go quickly or if a person or two doesn't show up. But that's basically what we're going to do. At the end of each session, okay, just as a little warning or a little heads up, we'll take a little group photo, okay? Because I don't think we're going to be able to catch everybody throughout the whole day. But we'll take a little group photo by Zoom, and this is the kind of a little How to Grow tradition. But we're going to take a little picture also with an object that is near us that we find interesting or meaningful. So we're going to do this at each break. Okay, so at the end of this, I think we'll have 4 kind of group photos, and we'll stitch them all together and make a big collage out of it when we're done. Okay, so that is the plan. Does anybody have any questions about any of what I've just shared? Do these logistics all make sense and sound good? Okay, if that all sounds good, everyone, I'm going to hit start on this slideshow, okay? And literally the second that the clock hits 9:05 on my phone, I'm going to, well, this is me giving the welcome right now. Actually, how do I pronounce your name? I want to make sure I get it correct.
It's called Alireza.
Alireza. Okay, great. And if I mispronounce your name, deep apologies in advance. But I will do my best. But okay, Alireza. Actually, why don't you get started a minute early? Okay, we'll let you get going. But anyways, here we go. The floor is yours, and have a great talk.
Hello everyone, my name is Alireza Hekmati. I am a master student in medical biotechnology from Iran. My project focuses on designing peptides to protect patients against ischemia reperfusion injury or IR injury. IR injury, next slide please. IR injury occurs when blood supply to a tissue is cut off and then suddenly restored. For instance, during a heart attack or stroke. By restoring blood flow is crucial, the reperfusion step can productively cause severe cell damage. Inside the mitochondria in the diagram, reperfusion leads to a massive influx of calcium. This calcium overload activates a protein called cyclophilin D or CYPD. When CYPD is active, it triggers the opening of the mitochondrial permeability transition port, or MPTP. When the MPTP opens, the mitochondria can no longer produce ATP. They swell and release signals that lead to cell death and tissue damage. Next slide, please. Our solution involves a custom-designed peptide targeting sarcopterin D. On the left side, you are looking at a 3D model of CYPD with our specific target sequence highlighted in red and yellow. Just below that, the sequence in red was developed to bind to CYPD with good confidence. Our hypothesis is that this will prevent the opening of the MPTP during the stressful condition of reperfusion. We will rigorously test its effectiveness and conduct experiments like MTTT assay to evaluate cell cytotoxicity and viability. Next slide, please. Ischemia reperfusion injury is a critical factor in many medical emergencies, like heart attack and during organ transplantation. Despite its significance, there is no FDA-approved treatment for this condition. My project aims to first computationally design peptides targeting this mechanism. Second, experimentally validate these peptides through in vitro and in vivo studies as part of my master thesis. And third, if results prove promising, pursue clinical trials for regulatory approval. Ultimately, successful development could lead to a new class of approved clinical intervention, offering better protection for patients. And finally, I would like to thank David, George, Paul, Tobin, Austin, and other TAs for their organizing the HTGA course and giving me the opportunity to be a part of it. Thank you for your attention.
Okay, great job. You have 2 minutes left. Maybe just to follow up as a question, and for all the TAs, again, please be ready with your questions. We can raise your hand and please ask the question. But can you say a little bit more about your aims? You've got them written out here as in silico, in vitro, in vivo. Can you say in a little bit more detail what you would like to do in your aims?
Yes, so for my aim one, I decided to choose something easy that I could do in the HTGA. So because of that, I only focus on computational part of my project. So I use machine learning models and different bioinformatic tools. For my aim 2, I am planning to use these designs and results and do an experiment in lab as part of my thesis in my master thesis. And if the result was promising, I would like to make it further and go from in silico to in vitro and in vivo and possibly a clinical trial as a part of my entry.
Can you say a little bit more about the in silico and the in vitro experiments? Like what actually would you do? Like what are you modeling in silico?
Yes, so the protein we choose was sarcopterin D, and we choose the residues between 137 to 170 and use AlphaFold multimer and other binders like Bindcraft to create a peptide binder that can bind to it. So it is one of the things that we did. The other one was designing the benchling part, optimizing the sequence in Twist and other things.
Okay, great. Ben, super quick question.
Great. Thanks so much, Lorenzo, for your presentation. Really amazing. Just a quick question about your aim number 3. Did you think in anything about maybe the delivery of the peptide or the way that you will be commercializing or giving the world your technology?
Yes,
Thanks.
that's a good question. So because it is going to be used in problems like myocardial infarction, so heart attack, the time is really important. Because of that, we have to use it in intravenous administration, so inside a vein.
Okay, we'll stop there. Thank you so much, Alireza. Great job. Up next, we have Chloe Lee. Chloe, the floor is yours. And actually, TAs, if you
Thank
don't mind
you.
helping me spotlight the speaker, that'd actually be really great. I'm not gonna be able to do that. So if TAs can help me spotlight the speaker. Chloe, the floor is yours.
Yeah, thank you. Just checking if you can hear me.
We can. There you go.
All right, great. Thank you. Hi, everyone. I'm Chloe. I'm a policy researcher in London. If you could go to the next slide, please. I'm designing a bacterial solution to reduce indoor air pollution caused by benzene, one of the volatile organic compounds and indoor air pollutants found in homes today. We spend a large amount of our time indoors. We are exposed to benzene when we use detergents, paints, and pesticides, and when we smoke. Benzene is not only carcinogenic, it can cause mild symptoms like headaches and severe damage to our central nervous system. In the past, researchers applied directed evolution to bacterial strains to select those that have mutated to withstand and metabolize benzene, or isolate bacterial strains from harsh environments and use them for bioremediation of benzene in soil and wastewater. So what I want to do is to genetically engineer an entire benzene metabolic pathway in Pseudomonas foetida, a common and safe soil bacterium for genetic engineering. What this means is Pseudomonas foetida is programmed to use benzene as a sole carbon source. While many indoor air quality technologies like filters and ventilators are effective, they may not always be affordable or accessible to residential homes. So my vision really is to reimagine air purification with an affordable bioengineered bacterial solution. Next slide, please. My aim one is to design a synthetic benzene degradation pathway in Pseudomonas foetida KT2440. Aim two is to develop and test this engineered strain for benzene degradation and extend the pathway assembly to include genes that will degrade formaldehyde and toluene. Aim three is to investigate the production, regulations, and commercialization of this product. Here on the right, I started by analyzing the genes of proteins involved in the complete mineralization of benzene into acetyl-CoA. I then identified the relevant nucleotide sequences and modeled the construction of a plasmid on bench-link. Next slide, please. Next slide, please. Yeah, thank you. The illustration on the left shows the construction of a plasmid that contains 8 genes in benzene degradation, shown in light blue rectangles, D and P gene classes, and MHP gene classes. This is a one-step assembly via the homologous recombination technique in yeast. The illustration on the right is my final design of the plasmid. Each yellow chevron represents my genes. My future work will be to actually construct this plasmid and transform the final chassis. I will validate this by testing gene expression and degradation activity of the transformed bacteria. Finally, more genes will be included before the final product commercialization. Some ideas include growing the bacteria in a container or inoculating the roots of houseplants, which will hopefully degrade VOCs in indoor air to improve our health. Thank you.
Hey, great job, Chloe. Questions from the TAs. I have one question. So can you say a little bit more about your measurement? So you mentioned that you would measure the degradation. Can you say a little bit more about how you would do that?
Yeah. So this has been done in some studies where in a gas chamber, scientists have input the benzene in gas form and then measure the degradation or the reduction of benzene in the air concentration as we have the plants with the bacteria, transformed bacteria in the soil. And so to measure really the reduction of the benzene as time goes on. So that's one way of testing it. There are other ways, which is like liquid VOC. So testing the bacteria in the liquid with the benzene solution, benzene concentration, and then that would go down as well. So different ways of testing.
Okay, great. Any final add? Go ahead, Ben. Thank you.
Great. Thanks, Chloe. Amazing project. Just have a question for your aim number 3 again. Do you think anything about the final application? I can see something like an aromatizer or something like that there. But can you explain a little further on that? How will we will work and take the air from the environment? Yeah, something. Thanks.
Yeah. I think the what you are seeing here is a device. I haven't really reached that far in terms of actually designing it. But the commercial solution right now is that the commercial solution is that the bacteria solution actually comes in the bag, and then you can add the solution into the soil, and then soil of your house plants. So you can actually use it at home. And then I think it's like every few months or so you need to top up the bacteria solution. But because of the GMO regulations in the EU, this may or may not be very possible. So then what I'm thinking of this is a separate device that actually can grow the bacteria, but I haven't thought very far about like whether it can be done. Yeah.
So Chloe, we're gonna have to end there. Patrick, if you don't mind asking your question over the chat, then Chloe can answer that way. But unfortunately, we got to move on. But thank you so
Thank
much,
you.
Patrick, for your question. And Chloe, thank you so much for the great talk. Wonderful job.
Thank you.
Okay. Up next is Charnusta here.
Yes.
And can you pronounce your name? I want to make sure.
You can call me Amanda.
Amanda. Okay.
Yeah.
Okay, Amanda. The floor is yours. Go ahead.
All right. Hi everyone. My name is Amanda, and I'm from Indonesia, and I'm working as a research assistant in bioinformatics at IPB University in Bogor. And can you next slide, please? All right. My research focuses on natural product-based drug discovery and beyond plants. I'm especially interested in marine sources, and that's why in this final project, I propose a scalable production of marine natural products using genetically engineered bacteria. We all know the ocean is full of organisms that produce compounds with antimicrobial, anti-cancer, and antiviral activities. And one group I'm focusing on is marine sponges or porifera. And interestingly, over 50% of their bioactive compounds actually come from their symbiotic microbes, not the sponge itself. And in this project, I look at T. mesophyllum, a microbe associated with the sponge Thydania species, which produces Bisocaverin B. This compound has been used for treatment for iron poisoning and has an anti-cancer potential. But the problem is harvesting. Unlike plants, you can't just easily farm or send sponges. So my question is, what if we could engineer a microbial chasis like E. coli to replicate the complex specimens found in marine environments? And there's already a success with provision of artemisinin in yeast, and Bisocaverin also has been partially studied in E. coli. Next slide. And so the goal of this project is to build a genetically engineered E. coli that can produce Bisocaverin B, starting with the biosynthetic genes that were computationally constructed. So my first aim is to understand the biosynthetic pathway, and I start by annotating the biosynthetic genes cluster and breaking it down into molecular gene blocks. And fortunately, because this compound has already been studied. And the second aim is to redesign the gene sequences for E. coli system and design the synthetic construct, encoding optimization and regulatory elements. And I found a paper in 2018 that proposes a gene layout using synthetic parts called BSBD1 and BSBD2, which are then fused into a functional gene cluster for expression in E. coli. Next slide. Okay, lastly, the third aim is a more visionary step to develop scalable biofactories using E. coli. Because of its fast growth and ease of genetic manipulation, E. coli is ideal for early stage prototyping of biostatic pathways. And the steps include transforming the engineered genes into E. coli, validating both the product, detecting Bisocaverin B, and validating both the product and the gene sequence. Phase 2 will focus on improving the system by maybe exploring another chassis like yeast or another bacteria. And phase 3 is about building a modular platform, kind of a plug-and-play system for different marine gene clusters. And in the long run, this could support a more sustainable way to produce marine-derived drugs, similar to what this picture here is a photochemical process developed by Sanofi that produce semi-synthetic artemisinin. And yeah, that's all from me. Thank you all for listening.
Good job. Okay. Questions from the TAs. You can raise your hands. So one question for me, again, really lovely slides. Can you say a little bit more about like what you'd expect from ocean microbes compared to other microorganisms? Like, why are you so interested in drug production there? Like, are there particular types of drugs you think that are going to be interesting? Can you say a little bit more about your vision there?
Yeah. Basically, like in my daily life, in my daily activities, we deal a lot with plants, and some researchers also research the compounds, like the bioactive compounds, their potential in the marine life, and there are so many of them. But what I think is, how do we get that compound from, let's say, marine sponges or bacteria? Because unlike plants that you can grow, it's hard to harvest them, and it's not that you can extract many times. So there is some we need to think of some ways of producing them without extracting them from the sea. Yeah.
Yeah, great. Okay. Thank you so much again, Amanda. Great job.
Yeah. Thank you,
We're gonna
David.
move on. Thank you. We're gonna move on to our next presenter. Is Irendra here?
Yes. Hello. So let's
Hi, did
start.
I
I'm
pronounce
Irendra
your name?
from Life
Okay,
Labs.
good.
Yeah.
Go for it.
Let's go. Yes, let's go. Yes. Good morning, everyone. So every year we discard 840 billion use of food packaging items. Only 9% of that plastic is ever recycled. In Sri Lanka, we have a cultural alternative. Meals served on banana leaves, which are naturally hydrophobic, food safe, and biodegrade in weeks. Unfortunately, raw leaves lack the strength and flexibility to deliver the modern catering needs. Based on this, my hypothesis was if we boost the cellulose while trimming lignin, we can create a tougher yet foldable leaf that still composts quickly. My solution has 3 genetic steps. In the AIM1, I am working to strengthen the structure by overexpressing the CESA for thicker cellulose fibers and flexibilizing by knocking down the PAL to lower the lignin levels. In the second aim, I plan to add wax, heat stability, and antimicrobial peptide to the banana leaf structure. The impact aligns with 4 UN Sustainable Goals. Next slide. As I mentioned before, AM1 covers the first 2 genes. I started with literature and KEGG pathway review to confirm CESA sites in cellulose biosynthesis and PAL in lignin biosynthesis. Then I did a blast motif check to verify the catalytic motif. I used FIRE2 to produce high-confidence 3D models, and I quickly inspected the active site's accessibility in MOL to double-check. And using benchling, I then delivered 2 gRNAs with on-target scores of 71 and 57. And then I attempted Golden Gate cloning into the first ligation. And the protocol is validated. In the future weeks, I redo the assembly, transform Agrobacterium, regenerate banana plantlets. And by the week 20, run the tensile and compost test. Next slide, please. So my aim to extend performance. Activating wax will cut the water uptake and also hold the stiffness, serving the temperature and microbial spoilage in the material. So the success of the project is defined by using 4 benchmarks that I mentioned here. So for biosafety, we plan chloroplast targeted edits and mesh house field trials. Economically, the target cost is under 10 cents per plate, and it's cheaper than a PLA in many markets. So my aim 3 fulfill my broader vision. I vision this project to scaling through farmer co-ops in banana regions, meeting global demand for eco-tableware and porting the platform to other crops like hemp. And we are currently in the conversation with LifeFabs in the UK for bioprocess design and with the Wageningen University for agronomic scale-up. In short, we are upgrading a cultural tradition with precision genetics to replace plastic plates worldwide. Thank you very much, and I'm happy to take any questions.
Great job. Awesome slides too. Wonderful job. So can you just say a little bit more? I know everybody's speaking so quickly, but where are you experimentally in the project? Like, what were you actually able to get done experimentally up till now, and what are your next steps?
Okay, so in terms of experiments, I didn't currently do any experiment wet lab, but when it comes to the dry lab, I have done until the plasmid generation from literature review to KGBG pathway review, and I use MOL to and also FIRE2 and MOL using this pathway. I have done experiments on that.
Perfect.
Also, in the future work, I want to go beyond the golden assembly cloning attempt. Yes.
Great. Quick question from Greg, and then Ben, maybe you can do
Yes.
yours from the chat. Go ahead, Greg.
Hi, so really creative idea. I'm just wondering, have you given any thought to how this might affect the growth of the banana plant? Because once modified, you'll want to be able to grow this in mass.
Yes, that's a very insightful question. Actually, I thought of that. So instead of growing this as a plant, I was thinking of growing this in dishes or growing in another way where we can only use the plant leaf rather than treating the whole plant and the trunk of the tree. Because otherwise, if the trunk get more lignin and cellulose, it will affect. Yes, that's true.
Okay, well, we'll have to wrap up there. Andrew, great job
Thank you.
again. Thank you so much. Up next, we have Abilea. I hope I'm pronouncing your name okay. Are you there?
Yes. Yes. Am I audible and visible?
Yes. The floor is yours.
Thank you. So, hello everyone. I'm Abilea. I'm an undergrad student from Hyderabad, India. Today I'll be presenting my project and its core aim. Next slide, please. So my project and its core aim is basically building a fusion protein to remove excess phenylalanine from the brain due to phenylketonuria. So I'm a genetic student, and as a genetic student, I've studied many genetic disorders, and the one that caught my attention is phenylketonuria, which is an inborn error of metabolism where the amino acid phenylalanine isn't converted into tyrosine due to the lack of phenylalanine hydroxylase. And this leads to a buildup of phenylalanine, especially in the brain, causing developmental problems. Current treatments focus on dietary restrictions and avoiding foods with phenylalanine. And while this controls blood levels, phenylalanine levels in the blood, once it enters the brain, there is really no effective way of removing it. So my aim is to actually design a fusion protein that can detect this excess phenylalanine in the brain and bind to it when it exceeds normal levels, and mark it up so that microglia, that is the brain's immune cells, can clear it through phagocytosis. Next slide, please. So the components that I used are, actually there are 3 components for this. That is a phenylalanine binding protein. I used phenylalanine dehydrogenase. This is actually just an enzyme that can identify, but this actually breaks, it has an enzymatic activity. So it is not efficient per se, but somewhere to start, and a flexible linker that would reduce steric hindrance and improve flexibility, and the FC region of the human IgG antibody, which helps microglia recognize and respond for this fusion protein. So I retrieved the amino acid sequences from UniProt and reverse translated them into DNA. And then I used BenchLink to assemble and analyze the construct. After that, I translated the protein sequence from DNA sequence to protein sequence using XPCE. And AlphaFold, I kind of fixed a small issues of which I ended up using FIRE. And through homology modeling, I was able to get a design, but the design is still in progress. Next slide, please. Next slide. Yeah. So going forward, my short-term aim is that I want to optimize the phenylalanine binding part and maybe just isolating the binding region from the protein instead of the entire enzyme, and test the binding through docking simulations, and also to properly visualize the fusion protein that I edit later on. And if this works, it could actually offer a targeted way to remove toxic metabolites from the brain, starting with phenylalanine and PK, but actually applicable to other neurological conditions as well. Very grateful for the opportunity. I'm very happy to take a few questions. Thank you.
Okay, great job. Thank you so much. Let's go to questions. Martina, to you first.
Yeah, wow, it's a really amazing project. I really love your idea. I just have a question. How will you regulate the excess of phenylalanine that you're gonna like phagocyte? Because this is also like a precursor in a metabolic way. So you need it for tyrosine. So how will you regulate this?
So that is one challenge that I've been trying to figure out how to do it, because I think maybe using a domain that is having kind of moderate affinity to low affinity instead of high affinity, so that once it reaches a certain level, only then it will bind to phenylalanine instead of binding it as soon as it sees the molecule.
Okay, thank you. Ben, the next question.
Thanks so much for your presentation. Just have a quick question regarding your final aim. Did you think in the final delivery of your protein, maybe you will using, I don't know, in blood, you will using in liver? Will it be the delivery of your protein itself or your therapeutic agent? Yeah, thanks.
Okay, so again, I haven't thought that far yet, because I've still yet to figure out how to do the fusion protein. But then, yeah, so I will probably have to think more about that.
Okay, why don't we, Adrian, if you don't mind asking your question in the chat, we're gonna move on to the next talk, but great job. Thank you so much again. Great work. And moving on to our next speaker. Thank you. Anastasia, are you there?
Yes, can you hear me well?
Yes, we can hear you.
Okay, great.
Okay, the floor is yours.
Okay, great. Hi, everyone. My name is Anastasia Arkipenkova. I'm an undergraduate intern at the Design of Cells Lab. And for this project, I was exploring DNA as a form of a database. So please, next slide. So when we think about data storage, there's many different mediums that we can use. So like diaries, CD disks, but DNA is also one such medium that we can potentially use. And what's great about it is that it's very dense, it's efficient in terms of its energy, and it's very stable. So especially in our age, of AI and all of these data accumulating, I think this could be a potentially great way to efficiently store all of the data that we are accumulating. And one of the reasons why I got interested in this is this little, I think, lecture that was given by Feynman, where he mentioned that to biologists, it's very obvious that so much information can be stored in such a small space, alluding to DNA. And so I wanted to look a little further into this DNA as a database. And so this is kind of a simple way to think about it. I mean, digital data at the very basic level is, bits, so zeros and ones. And encoding data into DNA takes it one step further. And so you can see here DNA in binary, and then subsequently in nucleotide sequence. And there's many different ways to encode it actually, but the one here is the one that I decided to choose because it's quite compact in terms of the information. And so the aims of this project were more to just explore and see how we can optimize like DNA databases, if there's anything from computer science and data structures that I could perhaps translate into the DNA world. And also I've explored some ways to automate this process. So please, next slide. Yeah, so first aim to explore, some concepts from computer science data structures and translating them to DNA. I, thanks to George, was looking at kind of initial data structures, like very, very simple ones that were used at the very beginning. And I was thinking about how that can be translated into, storing data in DNA. And I took tree-based data structures as my kind of basis for exploring it. And I decided to represent the levels in the tree-based data structure as parts of a barcode. So you can see here, me and some of my friends have made some 10 by 10 bit black and white images and encoded some pictures in there. And so what I wanted to do is to retrieve specific images that I want from my pool. And I was going to do that based on the barcoding system. And so you can see here, it has several regions such as constant, presenting the root of the data structure, as well as variant A. So that would be, for example, a category here like animal, and then another variant region, which would then kind of point to a specific item, like a cat. And so that was one way of kind of trying to represent that tree-based data structures and DNA databases. And please, next slide. And another part was automating the encoding and decoding. And so I hope you can see video here. I made a very simple web app so it's easier for my friends and me to encode some 10 by 10 bit images into the binary and DNA. And so you can see here kind of a speed up process of us encoding a picture of an alien here. And so that was one of the ways I kind of tried to automate it. And I think I'll take that a few steps further after this project. And for decoding, actually, I was going to use an Opitron to then, after sequencing my nucleotide sequence, to print it out on the plate. And so a very basic workflow is, of course, processing the data and encoding it, binary and then nucleotide sequence, design my barcodes and primers, order them, and then sequence. It's very, very simple. So currently, I have ordered the DNA strands, so I hope that once they come, I'll be able to actually decode my images and then print them out into the plates. So that was a really fun project. And also I want to say thank you to all the TAs and everyone who helped me and gave me ideas. It was really, really fun to explore this. So thank you.
Okay, thank you so much. We're unfortunately out of time for questions for Anastasia, but please ask any questions in the chat. But that was a great job. I love this project. Really, really neat. Yeah, but please let's ask questions in the chat and we'll move on to the next speaker now. Thank you so much again. Great job. Rushta, are you there?
Yes, I'm here.
Okay, floor is yours.
Okay. Hi, everyone. I'm Rushta Parveen, and I'm currently in my 4th year of BSMS degree. And my final project is about photoswitchable therapy, focusing on proteins that work in the nucleus. Next slide, please. Okay, so the biggest drawback of traditional drugs is their lack of spatiotemporal targeting, which photoswitchable therapies overcome. Current photoswitches are azobenzenes that use UV light to isomerize, but as the figure on my slide shows, UV light barely penetrates animal tissue, so it's useless for photoswitchable therapies. Now, by contrast, the human body is nearly transparent to 740 nanometer radiation called near-infrared radiation. While azobenzene absorption can be redshifted, what's convenient are bacterial phytochromes like BPHP1 that convert to active form on illumination and bind to PSP2R to shut down gene expression. Now, the application for this can be such that an effector fused to BPHP1 is activated only after binding to PSP2R. To keep the system compact, QPAS1 can be used, which is a truncated variation of PSP2R. My aim 1 was to develop a general light control platform for nuclear proteins. By fusing the effector to BPHP1 and a nuclear localization sequence to QPAS1, the protein remains inactive until NIR triggers binding, which leads to the whole thing migrating to the nucleus. Now, BPHP1, could you maybe, next slide, please. Okay, so BPHP1 has 3 domains. The complex shown here was predicted using AlphaFold, and according to this, the phi tongue is for binding, while the other 2 domains sense changes in the chromophore biliverdin. I would prefer to use a truncated variant, but there is no prior literature on this, so I would validate the truncated variants based on the spectral analysis and binding efficiency and nuclear localization. Biliverdin, the chromophore, is actually produced in mammalian cells during heme degradation. So it should incorporate into BPHP1 directly. However, because biliverdin degrades quickly, we'll need to verify if this happens properly, or we need to co-deliver heme oxygenase enzyme with the therapy. The system enables precise spatial and temporal control of therapies, as I previously mentioned, and it is applicable to cancer, neurodegenerative diseases, and so many more. Next slide, please. So my aim too would be to apply this system to epigenetic editing, which has been tested for APP gene in Alzheimer mice models before. This would involve having a DCAS9 domain fused to methyltransferase from DNMT3A, and that would be fused to BPHP1. This would help with gauging how effective the therapy really is working. And my aim 3 would be to expand to other localization motifs like the cell membrane. Ultimately, these could become a one-time treatment that can be maintained with periodic NIR elimination by the patient themselves. Thank you for listening to me. Questions?
Okay, great job. Great job, Rishta. Questions from the TAs? One question I have is, as you're thinking about this as a therapeutic, have you thought a little bit at all about the kind of drug delivery methods? How would that work in a clinical setting?
Yes, so I think that fusing a chemical to this would still be fine, but I think it would work a lot better with peptides.
Okay.
Yes.
Very good. Other questions for Rishta on our project? Not then, Rishta, we'll ask any more questions to you in the chat, but great job. Thanks so much again for your great work. Wonderful. Okay, let's keep moving. Again, everybody's just doing such an awesome job. Thanks so much again for the great work. Yasmin, are you here, Yasmin?
Yes, hello. Can you hear me well?
Yes, Yasmin, the floor is yours.
Thank you. Okay, my name is Yasmin Yacoubi. I'm a second-year bioengineering student from Tunisia, and today I'm going to be presenting my final project about an intranasal delivery system of BDNF nanoparticles. Next slide, please. Okay, so an overview about this final project. The traditional treatment for bipolar disorder using antipsychotics and mood stabilizers such as olanzapine or lithium has its limitations, whether it's drug-related side effects, treatment-resistant cases, and long-term stability to prevent recurrent episodes. This project suggests a novel therapeutic approach, and it's basically an intranasal delivery of nanocarriers of BDNF. So what is BDNF? It is a brain-derived neurotrophic factor. It is a 247 amino acids long protein. It is a signaling molecule implicated in neurogenesis and synaptic plasticity. And the idea behind this project was basically when I read this paper about brain-derived neurotrophic factor and bipolar disorder. And it's about, it showcases the link between low levels of BDNF and patients with bipolar disorder. Developing this idea, there are two main bioethical considerations that arise. The first thing is to prioritize the research in this field of a new method of neuropsychiatric drugs in order to develop a safe form of BDNF and safe doses for this recombinant BDNF. And the second consideration is the fact that recombinant BDNF using genetic engineering and cell-free systems to produce it, as I'm going to explain later on, is quite expensive. So we need to make sure that people have access to such therapy. Next slide, please. My first aim was to design a DNA cassette and to produce BDNF in a cell-free system. So the first step was to design this cassette that has the CNV promoter, a COSAC sequence, my coding sequence of human BDNF. And basically, the native form of BDNF has signal peptide and the chain of BDNF. So I had to remove the signal peptide. Then I proceeded to designing a cell-free reaction based on HeLa lysates because I wanted to make sure there are the correct post-translational modifications of this protein. Third step was to purify it using nickel NTA affinity chromatography because in my cassette I have already added a histidine purification tag. The fourth step of my first aim would be to run an SDS page validation. Next slide, please. The second aim is the nanoformulation. So I have chosen this paper that showcases the design of nanoparticles, PEM nanoparticles with BDNF and polyethylene glycol in order to deliver BDNF for controlled release and protection of BDNF. For my third and final aim is to design a non-invasive targeted delivery drug to combine nanomedicine and genetic engineering. Yeah, that's it for my final project. Thank you very much.
Okay, Yasmin, great job. Great work. Questions for Yasmin? One question, can you comment a little bit about kind of the novelty of using BDNF for this application? Is this something that people have used before? Curious the innovation part of your project.
Well, there are some articles I read about using BDNF as a therapeutic protein, but there are no, they were like experiments on animals, but not actual clinical trials or entire papers about developing such a drug. Also, I think about the innovative part of my project is that there is a potential of a curative approach, meaning that all the drugs designed before to treat bipolar disorder or general mood disorders are symptomatic, but BDNF therapy has potential to be curative, and there is like a possibility of people being cured of their mood disorder.
Okay, we'll have to leave it there for time, but Ben, if you can ask your question in the chat, that would be wonderful. But Yasmin, great job. Thanks so much again. Moving on to the next speaker, Yash. Are you there, Yash?
Hello. Can you hear me?
Okay, Yash, the floor is yours.
Hello, I'm Yash from Mumbai, India. I'm currently pursuing my master's in biotech, first year. So my HTGA project mainly focuses on developing SynBio-based wearable patch for non-invasive volatile organic compound detection for mainly diabetes. So can I get the next slide? So this is a graphical abstract I created. So the current diabetes diagnosis or detection mainly relies on the invasive or non-invasive methods. There are mainly invasive methods like glucometer or sample collection that involves sample collection, and non-invasive methods. There are non-invasive methods like glucose patches and NMR, but these can be expensive, complex, and a bit inconsistent. So my solution was to create a wearable cell-free SynBio ribo switch, which will give a non-invasive real-time monitoring. So can I get the next slide? So my first aim was to build a sensing circuit. So this is a circuit I built in BenchLink by getting the aptamer sequence. So I ran docking simulation with acetone and potential aptamer. So the structural model I used was yeast phenylalanine tRNA, being a stable and reliable model. So the outcome was the acetone bound to the plausible pocket and showed that there is a chance of conformation, and the docking energy was minus 2.428, indicating that this is a modest affinity. But experimental validation is still pending for this, which is my second aim. So here's I visualized the acetone aptamer and analyzed the binding sites and tried to predict conformation changes. And this is the aptamer sequence in the rightmost column. Can I get the next slide? So my second aim mainly focuses on overcoming all the limitations I came across the in silico methods, and I'll come across the experimental methods. So there's a challenge with acetone that it is a very small molecule. So it shows a less binding affinity, or the conformational change has to be verified with some spectroscopic methods, some experimental methods. So I ran an RNA fold analysis, and I saw that that without acetone and with acetone, we can see the entropy graphs. and there is a decrease in the minimum free energy that says that the acetone, when acetone binds, there is local entropy peaks, and there is structural flexibility showing that there is a conformational change, but this has to be verified experimentally. So that's my second aim. So there is an increase in acetone. Another challenge with acetone is in sweat, the amount of acetone is very less, so the sensitivity has to be very established. So my objectives and future prospects for this would be not just create something for diabetes, but something that gives a VOC profile. And I'm not suggesting a alternative, but a complementary detection with glucose. So it can, because in diabetic ketoacidosis, there is a spike in VOC profile, so it can give a better monitoring. And my third point, eventual integration into smartphone, which is my third aim, to create a real-time portable biosensing patch by integrating it into a textile, and then monitoring with the application or mobile user face, which is very the visionary or broad viewed aim. So thank you so much.
Okay, great job, Yash. Dominica, your question.
Yes, I wanted great project. I wanted to ask whether acetone is emitted like homogeneously across the body, or have you thought about where to place your device? Do you know?
Yes, if we are detecting from sweat, so we has to place this the particular sensor where the amount of perspiration is high, or it can be like underarms or wherever that the perspiration is relatively higher than the other body, because the acetone is not released homogeneously.
Great. Adrian, quick question.
Yeah, thank you, Yash. Very great presentation there. Just a short question there. Is there any other disease or any other changes in the physical being of the person that can also release acetone and give you false positives?
Yes, a lot of factors affect the acetone or VOC profile, like alcohol, like the consuming alcohol, or some types of dietary products can cause VOC release, like excess VOC release. So that's why I said this is not a alternative, but a complementary to detection.
Thank you very much.
All right,
Thank
we'll
you.
wrap up there. Thank you so much. Arisa, are you there?
Hi.
Okay. Hi, Arisa, the floor is yours.
Okay. Hi, I'm Arisa Tani, and I'm a high school student in Tokyo. Can you go to the next slide, please? Okay, so the problem that I focused on is heavy metal pollution, and heavy metals are released from industrial plants or natural sources, leading to large-scale pollution. And they have detrimental effects on ecosystems, human and animal health, and they can be very challenging to remove. I was inspired by two fields of previous research. First, bioaccumulation and bioabsorption are remediation methods using biomass. They are cost and energy efficient with minimal environmental damage compared to other methods. And also, some plants have evolved tactics to minimize the damage caused by heavy metals, such as proteins that can bind to or transport heavy metals. Can you go to the next slide, please? My plan is based on the idea that if we apply these adaptations of plants to bioremediation tactics, then we could make their remediation methods more efficient. And my aim is to produce phytokeratins, which are molecules that bind to heavy metals in S. cerevisiae. And I chose this chassis for the reasons that are on these slides. Sorry, I don't have time. Phytokeratins are produced from glutathione, which are produced from glucose, urea, and biotin. And S. cerevisiae naturally produces glutathione in high concentrations, but it does not have the phytokeratin synthase gene. Phytokeratin synthase is the gene that produces phytokeratins from glutathione. So my plan is to express the phytokeratin synthase gene from these two organisms with the PYES plasmid. That's the plasmid map. And can you go to the next slide, please? So to integrate the yeast cells into a filter system, I will first kill them with heat, then put them into a fixed bed column, which is the most standard method of bioabsorption. And the absorbent will be lined with glass beads and wool, which will prevent the leakage. For the expected results, I expect that increasing the number of binding molecules that bind to heavy metals should theoretically improve the metal removal efficiency. And to further enhance this, I couldn't get to doing this, but developing methods to use like industrial byproducts or regenerated materials could bring down costs and make this method more feasible. As for the biosafety concerns, I don't think containment shouldn't be too big of an issue because dead organisms can't replicate on their own. But on the other hand, where to dispose of the waste, so the absorbents that are used will, I think, be an important issue. That's it. Thank you so much.
Amazing job. Arisa, you're in high school?
Mm-hmm.
That's amazing. Thank you so much for the awesome project. Questions from the TAs? I have a question. So, for environmental projects like this, can you say a little bit more about the efficiency of the metal binding? Do you think there's do you expect the system to, maybe, I don't know if you have thoughts on how efficient the process would be in nature, in the environment?
So, I don't know. It's kind of like a theoretical, but I thought like according to previous research, apparently in bioabsorption, the metal ions bind to like lots of binding sites inside the biomass. Like for example, in the case of yeast cells, they typically bind to like the cell wall proteins. But so I thought like increasing the number of places that the ions can bind to should increase the efficiency, but I
That's
don't know. I
great.
haven't
That's a
tested
great
it,
answer.
so.
Oh no, that's a great answer. That's a great answer. Fantastic job. Any other questions? If not, Arisa, great job. I wish I was as smart as you when I was in high school. I definitely could not have done a project like this. So great work. Thank you so much.
Thank you.
Okay. Up next, Yehuda, are you there? I apologize if I
Yes,
mispronounced your
you did not mispronounce it. Can you hear me?
name.
Hello?
Is your video? Yes, we can hear you. Is your video on?
Hi.
Oh, there you go. Okay, great. Okay, go for it. Floor is yours.
Okay. I can't see the presentation, so just try to change the slides when it makes sense. Hi, everybody. I'm Yehuda Binnick. I am a member at GenSpace. My project involves oleaginous yeast. Okay. Oleaginous yeast are yeast that are able to convert more than 20% of their body mass to lipids under certain environmental conditions. Their high lipid content gives them a potentially wide-ranging applications in chemical synthesis, including the production of biodiesel, vegetable oils, and the makeup industry. Among the bottlenecks is the cost of destroying the cell wall prior to lipid extraction. Among these methods are ultrasonication, bead milling, microwaving, and chemical treatment. However, all these methods are cost prohibitive for various reasons. One promising and underexplored method of cell disruption is enzyme-assisted extraction. In the 2020 paper, Heshef et al. Outlined a process where the enzyme produced by the microparasite T. herzianum was used against the oleaginous yeast to digest the cell wall. This is the basis of my experiment. The experiment involves 3 aims, each of which are self-contained. The first aim is to construct and program a bioreactor on a budget of $800. I will be uploading all the specification docs and software on a GitHub repo for anyone to use once it is done. The second aim is to reproduce the modified version of the 2020 paper that is able to be done in a community lab setting under a limited budget. The third and final aim is to genetically engineer Y. lipolytica, the yeast I'm using, to express the 2 main proteins found to induce cell lysis in the 2020 paper. Due to budgetary constraints, this will be done at a later time. Once this is done, I would compare the lipid yield of this method to the method outlined in the 2020 paper. So Results. I am well on my way to completing the bioreactor, as you can see from the photos. I have made my bioreactor is a modified version of an existing design known as the flavor reactor by Kuro 99, and it's built upon a Raspberry Raspberry Pi. And I'd like to thank him. He's been a great help. The final design will be able to monitor and regulate temperature, pH, and optical density for a cost of approximately $800. In addition, I have gotten my yeast 10 days ago, and I am in the process of making a batch culture to measure the growth rate, and that'll be done soon. And then in the future, I can't see at the moment the list of things that I'll be doing in the future that still need to be done, but you can see to the right of the screen the 4, 5, or 6 things that still need to be done before I can call the experiment complete. And this includes finishing building the bioreactor software and running all the experiments. Thank you. And if anyone has any questions, I'll be happy to answer.
Okay, great job. Dominika, question.
Yes. Hi, Yehuda. I was wondering how you would want, or like how in particular you want to ensure that your bioreactor stays under $800, was it? What considerations?
So I've already bought all the materials, and my budget was $1,000, and it came out around to $800. I use a bioreactor as the basis for that, and that's not too expensive. And then for the most expensive parts, I use Atlas scientific circuitry and probes, which comes around to $400. And that's $600 total, and then $200 for all the other electrical and mechanical components, such as pumps and flasks. The rest I would be 3D printing.
Great. Adrian.
Oh, hi, Yehuda. That was great, great project there. Can you tell me about the bioreactor? So you said you want to monitor the optical density. I assume you're going to use a peristaltic pump and a flow cell, correct?
Yes, I would use a bioreactor with peristaltic pump to monitor the optical density, because the bioreactor is quite small and it already has everything set up. It will just work fine right out of the gate as a flow cell.
Thank you.
Okay, great job, Yehuda. Let's move on to the next speaker. Thank you so much. Ada, are you there?
Hello, yes, I am there. Yeah.
Okay, the floor is yours.
Thank you. My idea was about to utilize the chirality, nematic chirality that the cellulose nanocrystals have. Maybe, could we maybe move on to the next slide, please? No, okay, maybe I can't see it. Yeah, thank you. The aim is to explore how to design ways to utilize the chirality of CNCs, which are cellulosic nanocrystals, that change color upon detecting specific biomolecules that can be a plant pathogen, I thought, because it came to me from a joke that saying you can take your plant to a doctor or a vet. So I first wanted to focus on how can the chirality of CNCs be rearranged, and why the optical color change is based on which parameters. And I found out that it's about, could we move on to the next slide, please? Thank you. It's about the helical pitch that they have, and the helical pitch is actually the nematic. It's the distance a rod turns around 360 degrees on its around while forming the helicoidal structure of the CNC. So the reflected wavelength depends on the helical pitch, and the helical pitch can depend on various things. One was also having a charged particle reacting and interacting with it, and the other one could be water intake. And I thought water intake could be more feasible to do since it just naturally occurs and then changes the CNC's chiral structure. And it visibly can just show results of changing the helical pitch from smaller wavelengths like blue or green to bigger ones like red. And I wanted to design a mechanism that would that the pathogen RNA could be, could be understood, and then it can be bound to a primal reaction, and the primal reaction can be triggered other reactions. So I first initially imagined it as a chain of reactions, and the last reaction output would be the H2O water, and then it would swell the CNCs, and then it would occur in an optical color shift. While trying to understand how I can do that, I thought that I learned that biosensors use a lot of pathogen detection utilizing the toe-hold switch, which I actually tried to write in BenchLink, but I failed. I'm sorry. It uses a toe-hold switch, that a programmable RNA-based gate. It blocks the protein production until it sees a specific RNA sequence. For this case, it's the pathogen, and then it would start expressing a peptide. I thought maybe a glutamate it would be, which is a swelling peptide that interacts with water a lot, which is quite hydrophilic. And then the output would result in CNC interaction and water uptake, and then water uptake would again changes the chiral pitch, and then it would expand it, and then the color shift would turn from either green or blue to red.
Ada, I'm so sorry, you have to start wrapping up.
Yeah, okay. And as for the users, yeah, it's designed for customs or gardeners.
Okay, fantastic job. And a quick question from Ben. Ben, you can ask the question if you don't mind answering it in the chat. But Ben, go ahead and ask your question.
Sure. Just to know about if you think about how you will be binding your riboswitch into the cellulose material, you will be using some domains to bind it to cellulose or something like that. Just that. Thanks.
And Ada, you can answer in the chat. Thanks so much again, Ada. Appreciate you. Okay, Anushka, we're skipping Anushka. Is that right, Garrick?
Yes, I don't think there are any slides.
Yes.
So
okay. Okay, perfect. Ooh, we get 5 minutes back. That's exciting. Taylene, are you there? I hope I'm pronouncing your name okay. Okay, the floor is yours.
Thank you. So hi everyone, I'm Taline. I'm a designer, and I started very recently working with mycelium composites, and for this project I wanted to tackle the build industry. So as we know, like the construction industry is responsible for nearly 40% of global CO emissions, and while there is a growing interest in bio-based alternatives, especially mycelium, among these materials. These materials are currently limited to non-structural applications like acoustic panels, insulation, and decorative elements. If it's on the right slide, sorry, I forgot to find that out. So, and the main reason for that is that because
Oh, we lost your audio, Taylene. I think you muted yourself.
Okay, sorry. So yeah, as I was saying, the reason why these materials are currently used in very indoors settings instead of structural ones is that because mycelium composites like the mechanical strength and durability. And this leads to an important question, which is if we already have the acceptance of these materials increasing in society, why not push forward? And for that reason, I try to experiment with the Metis versicolor, which is on the next slide. And I chose Dermatis versicolor because it is a fast-growing species known for its dense mycelium mats, and it is genetically tractable and well documented. So what I chose to focus on was on strengthening the material from within by enhancing two critical pathways. So chitin overexpression to increase cell wall stiffness, and beta-glucan overexpression to improve elasticity and load-bearing capacity. And the way I did that was using benchling, design a genetic circuit, the GPDA promoter for constitutive expression of chitin to increase chitin production, CHS1, sorry, the inducible ALCA promoter to control FKS1, allowing the glucan expression, and then the TRPC terminators to ensure stability and proper termination of the transcription. And for gene delivery, I selected agrobacterium tumescens mediated transformation method. The reason for that is that fungal organisms do not accept plasmids very well, so the transformation method has to be like, if I understood well, not by genome modification, but presenting the organism with something that will say, hey, produce more of this. And next steps and visions, which are on the third slide, is that my aim is to see if that works without stressing the organism as much, and try to enhance mechanical performance through tensile and compressive strength tests, and environmental resilience, including water absorption, biodegradability, and weather resistance. And yeah, I think that's for me. I probably ate some of my words.
Great job. No, you did great. That was awesome. Dominika, question.
Yes, hi Talene, great job. I was wondering if you have already like an idea by how much you want to tune the overexpression of those two respective genes. Do you have any guideline there, or do you want to perhaps experiment with different strengths, like promoter strengths or different expression strengths?
I have not yet, but please, if you have any guidance on that, happy to hear more.
All right, let's talk about it.
Ben, quick question.
Yeah, thanks so much, Talene, for your presentation. Just a quick question. Did you think about maybe the growing of the fungi? Since it's using glucose for production of the chitin, maybe that will impact on their growth. Maybe it will grow slower or something like that.
Yes. That was something that I was studying and deciding what to overexpress was something that I had as a trade-off. Like, okay, it can have chitin and glucan to kind of forbid it to become brittle, but then it will take slower. So that was kind of a trade-off, but we'll see how the test goes if I get it done.
Great, thanks.
Okay, thank you, Talene. Great job. Antonio, are you there, Antonio?
Hello, yes, I'm here.
Is your video on?
I don't have a camera. I don't. Is there a problem?
No, I was just trying to spotlight you, but if you have your camera, that's fine. Yeah, go for it. The floor is yours.
Okay. I can see my slides. I don't know. I'm not sure if they are supposed to show up.
Just say next slide. Say next slide.
When you're ready. My bad. Next slide, please.
Okay. Go ahead.
So good afternoon, everyone. So I am presenting a project that focuses on the development of microbial biosensors using synthetic biology. Current methods are outdated. They are slow, costly, and rely specifically on specialized lab equipment. This, of course, makes on-site and real-time monitoring extremely difficult and delays necessary responses. Our objective is to tackle these limitations by harnessing microbial biosensors and overcome them, making biosensors not only highly effective, but also usable in the field, hence the need to innovate. Can you switch to the next slide, please? Thank you. So we came up with a solution to create a new generation of microbial biosensors. The vision is to make pollution detection as simple and accessible as possible, turning this problem into a truly global solution to a global problem. Next slide, please. Thank you. So we can break this down into 3 main stages. So stage 1 or aim 1, we begin by designing and testing genetic circuits in E. coli. We can also choose different organisms for specific types of pollution. For example, Cupriavidus metallidurans. We can use it to specifically detect heavy metal pollution, or even Pseudomonas putidus. This is done by choosing a specific pollutant-sensitive promoter and cloning it through Gibson assembly, followed by heat-shock transformation with a plasmid. After we can plate it and incubate it to purify the strain. On to aim 2, it's all about performance optimization. This involves adding tuning elements like RBS with further Golden Gate assembly to easily swap circuit elements like the promoter if we want to detect any other form of pollution. Aim 3, finally, we transition from lab to field. This phase focuses solely on scalability and usability. So the objective on this aim is to build a real-time on-site detection system that's viable for widespread deployment. So expectations are that this will significantly outperform current prototypes in terms of detection speed and sensitivity. Looking ahead, future work includes expanding the range of detectable pollutants and exploring other microbial hosts for broader applications. We also aim to engage with policymakers and environmental agencies to ensure real-world impact, and this is probably the hard part. So that's it.
Okay, Antonio, thank you so much. Great job. Ben.
Thank you.
Question from Ben.
Yeah, thanks so much, Antonio. Just one question. You mentioned it that your idea is to like upgrade it and have more biosensors, right? Do you have any idea from where you will be taking those? Because, for example, you're using the ARS1 system that is a transcriptional factor-based biosensor. So do you have any idea where you will be taking your other components? Only that, thanks.
Thank you for the question. So I tried to design a plasmid on BenchLink. It's actually on slide 3. So I did some research and I found that all the components that I had to that plasmid theoretically should add some efficiency to it. Although I haven't tested it in the lab, so I'm not really sure how accurate that is. So yeah, I'm not sure if I answered your question, but
Antonio, we'll leave it there for now, but thank you so much again. Great job. We'll move on
Thank
to
you.
Matt next. Matthew, the floor is yours.
Yeah. Hi, everyone. My name is Matt Berkshire. I've got a background in product design. And so if you go to the next slide, this project was really inspired by the idea that a lot of people kind of want to work on making products biodegradable, but I was kind of interested in how they could have some sort of like symbiosis with the earth during their life cycle. And so I wanted to focus on trying to make sort of fertilizing biomaterial. And so I'm working with the nitrogenase enzyme and the nitrogen cycle essentially. And so that process of turning atmospheric nitrogen into ammonia fertilizer for plants. And so the major industrial way that we do this is this beautiful sunset photo is the Haber-Bosch method, which is responsible for, like a huge chunk of the population's survival, and just the way we scale agriculture. But it's incredibly energy intensive. And so essentially looking into the enzyme and certain bacteria that do this automatically. And so most scientific efforts are kind of, the ultimate end goal that many people are looking into is how to incorporate this gene into plants so we can have self-fertilizing plants. But it's incredibly difficult and it's never been done. So I want to try and kind of make a stepping stone for this sort of material between like biofertilization that we have today and then the, future of plants. So if we can go to the next slide. My first aim is, again, there's a ton of studies trying to optimize and assemble this enzyme. It's incredibly complex and difficult. There's like a huge amount of genes. But so essentially I'm taking a lot of the optimization strategies and literature that I've found for people trying to incorporate this into plants, and I'm taking it back into the native bacteria. And so that kind of sidesteps a lot of the major assembly issues and stuff. And so the main things I wanted to focus on are oxygen sensitivity, because nitrogenase is just irreversibly destroyed by oxygen. And then also just the overexpression and the optimization of nitrogen fixation. So I designed 3 plasmids to kind of go over. The first 2 are sort of a basic circuit that are either activated and repressed by the presence of oxygen. And so we have sort of this overexpression of the nitrogenase structural enzymes on one side, and which is repressed by the presence of oxygen, and then on the other side we have these SID A and B genes, which are these oxidase enzymes that essentially eat the oxygen before they get to the nitrogenase, and so it's sort of a protection barrier. And so that, and when we get oxygen spike, we get these enzymes to protect the nitrogenase. And then we could see through GFP as well, we can sort of visualize oxygen spikes in the system. And so if we go to the next slide, I guess I've sort of done a few different things, and sort of the aim 2 and 3 is to sort of obviously scale the native bacteria in sort of a bioreactor setting and sort of measure the optimization of the nitrogen fixation versus like how much ammonia it secretes. And then obviously looking into, a whole other research intensive is just how to implement this into a material, whether it be like a hydrogel or a biofilm, and then, looking into actual applications of what this could be used as. If it's like garden stakes or larger agricultural sales, that could be, a material that, sidesteps actually modifying the soil for fertilization, but uses this sort of material as a catalyst for the ammonia.
Okay, Matthew, awesome job. Really, really great.
Thank you.
Questions for Matthew. I guess I have one question. As a designer, and bringing a lot of the design sensibility here, what do you think about kind of the durability of these materials, and kind of incorporating genetic engineering into materials that could be a part of a shoe? How do you think that this would kind of, how do you engineer or design this to last more over time and have it be a real useful
Yeah, I
product?
think lasting over time is definitely the sort of difficult part, because keeping bacteria alive long term. And I think also it kind of goes into the bioethical stance where the shoe might not be the most practical solution. Ultimately this thing like secretes ammonia gas. So how much in trace amounts, I don't know, but I think the shoe is kind of what really inspired this project. And so I think it's just sort of relatable to a lot of people to, help get this idea across. But whether, whatever sort of, material this ends up being, I think the long-term viability is definitely interesting. I think I'd be interested in looking into like cell-free systems. If you can have almost these like plug and play components that, you can sort of one-time use for a spike in fertilization and then, replace it later, I think would be interesting. But yeah, it's it's a complex topic, but.
Great, thanks a lot. And speculative design is also really useful. These ideas are provocative and can be really valuable in and of themselves as a visionary kind of process. So thanks so much. Great job, Matthew.
Yeah, thank you.
Up next, Cindy. Cindy, are you there?
Hello. Yes, I'm here. Can you hear me?
Okay, Cindy, yes, the floor is yours.
Perfect. So hello, my name is Cindy. I'm part of the Life at Node in the UK. So yeah, I focus my research in the diagnosis of endometriosis. Next slide, please. So endometriosis is a long-term condition that affects 1 in 10 women of reproductive age. It usually takes 7 to 10 years to get a diagnosis. And why? Is the reason is there's not easy or not invasive test. And right now, the doctors are using laparoscopy, as you can see in the image. That is a surgery that it takes a biopsy from the endometrium. This is invasive and often very delayed until you reach that point. Then our vision is to use RNAi sequence, machine learning, and quantum models to find early signs or potential biomarkers for the disease. These predictions will be tested in an organ-on-chip system that simulates the endometrium, and the goal is to help make the diagnosis faster, easier, and more accurate. Next slide, please. All right, so those are the aims, and the research were divided in 3 phases. The first one, I started by choosing a balanced group of 60 samples from the RNA-seq data, 20 controls, 20 ovarian cancer, and 20 endometriosis samples. Then I look at the data using heat maps to see the top 5 most expressive genes, as you can see at the image 2. Then I use PCA to check how the samples were grouped. And finally, I tested 2 models, a logistic regression model with an AOC of 0.36 and a quantum SVM model with a 40% of accuracy. These early results are not strong, but we were expecting that. It's normal because we are working with a small data. But still, we get some signal, which is great. Finally, the second phase is the full data set that I will test 120 or more samples, hopefully, to create better features based on gene expression. And the last part of the phase will be quantum optimization to choose the most important genes that are more effectively. Last slide, please. However, the model alone is not enough, and we need to test if those results are true in biology. That's why we are planning to validate the predictive genes using an organ-on-chip system that simulates the endometrial environment. This system will include endometrial cells and exposes to hormones like estrogens and progesterone, and the inflammation signals like interleukin-6 and TFM-alpha that are similar in real conditions, in real endometriosis conditions. Then I will check those genes that we were predicted in the previous steps and understand if it actually changed activity in this setup. This closes the loop from data to predictions to biological testing, and if it works, it could lead to a future non-invasive way to detect the endometriosis. Thank you so much.
Hey, great job. Thank you so much. Questions for Cindy? I have a question. So maybe can you say a little bit more about the organ on a chip process? You know, organs on a chip, you know, you typically can only kind of take a certain feature of the organ and replicate it on the chip. So can you say a little bit more about the design of the organ on the chip?
So to be honest, I didn't have a lot of time to look at this because it was the last step, and I've been like spending more time with the data, which was a bit difficult because I didn't have enough balanced data. So that was like just the based on the information and the others researches, what they were doing, and just like the basic concept of what are the elements that I need to add if I want to reach this step.
I guess what I'm meaning is what is the organ on a chip like? What are you using it for drug screening? So you're trying to see how well, you know, can you just say a little bit more about what the goal of the chip is? What are you using it for?
Yeah, so we want to understand basically the behavior. So basically, if the signal of those genes that we say that are part of the disease, it will be a potential biomarker. So we basically want to add the conditions of the disease and add those genes and understand if there's signal or not.
Okay, got it. Great. Okay, thank you so much, Cindy. Great job. Thank you. Moving on to the next speaker, Farhani, are you there?
Yeah, hello.
Okay, the floor is yours.
Okay. Okay, thank you so much for this opportunity. My name is Farhani Shefwani, and my final project is about biodegradable, bioengineered fiber for sustainable fashion. Next. We know that fashion industry is one of the largest polluters due to its use of synthetic fibers like polyester and nylon, and it cannot break down in the environment. It's not degradable. Every time they are washed, the material like polyester and nylon, they release microplastic that contaminate ocean and also enter the food chain. Another problem is natural materials like cotton and hemp are biodegradable, but they don't always have the same durability or performance needed in modern textiles. Bacterial nanocellulose or BNC that made by bacteria like Comacteobacter has a unique fiber structure that's strong, flexible, and biodegradable. If we can increase the BNC production, it has a huge potential as a sustainable textile. The main idea of this project is to engineer the bacteria to enhance the production of BNC and combine BNC with the natural fibers like cotton or hemp to create like hybrid fabric. Next. This project has three major aims. Is to select the best bacteria for BNC production and to design a DNA construct that improves BNC synthesis. Also explore how BNC can be combined with natural fibers to make a biodegradable textiles. The scope of the methods also included retrieving the sequence from NCBI and then designing the primer, assembling the construct, and the validation needed is to like do PCR and electrophoresis and then transforming the bacteria to test the yield improvement. Next. For the bacterial strain, the recommended bacterial strain is Comacteobacter reticus because it has multiple advantages because there are research published that state that it produces more bacterial nanocellulose than the other strain, and also it's genetically accessible, meaning it can be transformed using methods like electroporation and also accept broad range of plasmid. If this successful, the engineered strain will enhance the BNC production, which can be blended with natural fibers, and this fabric can be produced to like produce a flexible and biodegradable fabric, and also it have a potential to tackling the issue of textile and waste plastic production. I think that's all from me. Thank you.
Hey, great job, Farhani. Awesome. Questions from the TAs. I have a question. So, again, I think these biomaterial projects, bringing in engineered organisms, really, really interesting and wonderful. How do you keep the organisms alive, or how are you interfacing or integrating the organisms into your nanofibers?
I mean, you combine with the natural fibers, right?
Yeah. How would you like to do that, or how do you plan to do that?
Yeah, based on like previous like similar studies related to this fabric, there are several methods. The first one is like soaking the original the cotton or hemp into the liquid of this bacteria and let the bacteria grown yeah, to the natural fabric. So yeah, something like that.
Okay, no, that's great. Thank you. And Ben, if you don't mind asking your question in the chat, we'll move on to the next speaker. But
Yeah.
Farhani, great job. Thanks so much
Thank
again.
you so much.
Let's move on to Alonso. Alonso, I see you there. The floor is yours.
Thank you. Hello, my name is Alonso Reyes. I'm from Peru, and I'm designing a protein-based biosensor that detects cyanotoxin in fresh water. Next, please. So the problem is that freshwater reservoirs in Peru are threatened by seasonal cyanobacteria blooming that are not that predictable, and that's why we need some monitoring. This MCLR inhibits regulatory phosphatase that causes cytoskeletal destabilization and ends up to that cell death. However, constant monitoring of this toxin is limited due to lack of specialized equipment on site and also qualified personnel that can handle with monitoring of this toxin. So my goal is to design, express, and test protein binders, taking advantage of current deep learning protein models that can diffuse binders for almost any molecule, in this case MCLR, which is a ligand. And this is for develop a biosensor for on-site use in reservoir facilities. Next, please. So my first aim is basically take this PP2A enzyme in 3D structure and diffuse binders and filter designs using alpha-folded protein MPNN. And these binders, I will make construct for expression and cell-free TXTL systems and purify these binders and test in GFP split reassembly assays. Briefly, without MCLR, these binders are not able to interact, so GFP fluorescence will not be detected. On the opposite side, when MCLR is present, these binders interact and GFP fluorescence is emitted. So I will test also this with ELISA to compare how much the design is accurate and efficient to develop fluorescence. Next, please. And to go further and I want to optimize this GFP assembly assays for MCLR detection on freshwater samples, because we know that component inhibition is an issue. And finally, I will integrate all of these technologies into a single, easy to use and microfluidic sensor that detects MCLR, and I want to manufacture and distribute these microfluidic devices all over freshwater reservoirs in Peru to accomplish regular monitoring in these setups. So that would be my presentation. Thanks.
Awesome, Alonso. Really quickly, your figures are so nice. Did you make these figures?
Yes, I made them.
They are awesome. Great job. We'll hire you as a professional designer here. They're so good.
Oh no, I just by render in a couple of hours.
Yeah, no, they look awesome. Really, really awesome job. Patrick, go for it.
Really cool work. I like the protein sensor. I was wondering what the current methods are for detecting cyanobacteria in freshwater is. What are some existing methods?
Yes, for like lab setting, ELISA with antibodies that one can purchase from a kit, and also HPLC with some we will need like a column, HPLC column, and for biosensors, there are a lot of biosensors which recognize MCLR using antibodies or aptamers, which are more common, but protein-based biosensors are not as common and are scarce. So that's why my focus is on this kind of binders that are based on the MCLR target. So yeah.
Yeah. I was also thinking that you can make, instead of a microfluidic chip, you can make it more accessible by making it some sort of paper-based test, sort of like a COVID test kind of thing.
Yes, I also think on that, but I don't know how that test will be like filtering out freshwater compounds that, for example, soil particles, and there is a lot of freshwater, and not every freshwater compounds are equal. So yeah, but also thanks for your feedback, Patrick.
So great job. We'll leave it there for now. Thank you so much. Robina, are you there?
Yeah, I'm here. Am I audible?
Is your video on?
No. Am I supposed to on it?
If you can, that would be great, but if not, that's okay.
Oh, okay. Give me a second. So I just
It's
prayed.
okay.
So I hope
It's
that's
okay. Yeah.
okay. Yeah,
Yeah,
let me
go ahead. Go ahead. You don't have to
Okay.
turn your video on.
Am I visible
Oh, you're
and
good.
audible?
Yep, go ahead.
Okay. Thank you, everyone. Assalamu alaikum. This is Rubina Khan, and I work as a research assistant at Precision Medicine Lab. And also, I'm a self-proclaimed environmentalist. So next slide, please, for my project. So every year, over 8 million tons of plastic enter our oceans, and by 2050, it is projected that there will be more plastic than fish by weight. Microplastics in particular are in invisible threat because they are smaller than 5 micrometers and they travel through the entire food web. So despite the existing cleanup efforts, microplastic pollution remains largely unaddressed. And this is where my project begins. I chose Alkanivorix borkemensis, which is a marine bacterium well known for cleaning up oil spills. It is already adapted to ocean conditions and it thrives in hydrocarbon-rich environments. So that made it the perfect chassis organism for my solution. And then I started looking for enzymes and found PTase, which was originally discovered in Dionella, and it can break down PET, but it was not the ideal enzyme as it is not thermally stable. So in literature, I found this another enzyme by the name durapitase, which is a more thermostable, more efficient variant engineered for environmental resilience. Next slide, please. Okay, so for my construct design, I selected this broad host range plasmid as it has already been used in marine species as well as gram-negative species, which was the perfect plasmid for my microbe, my chassis. And to control expression, I used a native inducible promoter, naturally responsive to hydrocarbons. And for the extracellular cellular secretion of this PTase enzyme, I added an OMPA signal peptide to my construct. And so I assembled construct in silico using Gibson assembly. And I also codon optimized it for Alkanivorix. Again, I used GenScript, and GenScript didn't had Alkanivorix listed as the species. So I looked for the closest species related to Alkanivorix, which was Pseudomonas pitida. And so I codon optimized it for that. And next, I also wanted to validate my design. So I went one step ahead and I predicted the protein structure of this construct using AlphaFold. So if you look at the diagram below, it shows the output shows a high confidence fold in the central enzymatic domain. And the score for this was over 40, which suggested that the catalytic site adopted a stable functional conformation. And then the N and C terminal regions, especially the signal peptide that was attached to it, showed lower confidence, which was expected due to its flexible nature. And I've also attached the PEE plot, which displayed a strong diagonal TM score. And it supported my intra-domain folding with moderate uncertainty in the flanking regions. And I also wanted to ensure that this durapatase would actually bind to its target substrate, which was patase terephthalic acid. So I ran a molecular docking simulation using SwissDoc. Next slide, please. And among the results, you can see that cluster 0 showed the most favorable EC score, which suggested a stable binding pause with the terephthalic acid. And cluster 2 gave me the best Swiss param score, which indicated a strong interaction from a force field perspective. And these results gave me the validation that these are moderate but stable binding. And this was overall my project, and I just want to say in the end that my hope is simple, that a future where microbes help heal the planet from the damage we as humans have done. And yeah, a quick shout out to my mentors, Austin and Mario, and also my inspiration, Annie, who made me take this course. So yeah, thank you.
Great job. Outstanding presentation, Robina. That was awesome. Thank you so much. Questions from the TAs. I have a question. So, when you're doing obviously environmental kind of related degradation of toxins and plastics and so on, how do you envision this actually being deployed? Like, would we actually, would you grow up the microorganisms and just literally put them out there? Or, can you say a little bit more about how you imagine this
I don't think so
used?
that it is going to be like the efficient method if we are just going to deploy it directly into the ocean. I feel like the initial step that we should take is to have these bioreactors which are deployed near the oceanic areas, and maybe we can run that as a test first. And the reason that I chose an oceanic microbe was also because that I wanted to not disrupt the whole food chain. So to minimize the damage. So that was also one of the reasons.
Okay, great. Awesome. Robina, great job. Thank you so much.
Thank you.
Holly, are you there?
Yeah, hi.
Holly, are you able to turn your video on? I'm not sure I see you. Oh, there you are. Okay, Holly, great. The floor is yours. Go for it.
Thank you. So my project was about creating a UV absorbing biotextile for climate protection, genetically engineered using extremophiles and cellulose. Next slide, please. So as we're well aware, climate change is an ever-looming issue, and 2024 was the warmest year on record, with temperatures exceeding 1.5 degrees above pre-industrial levels. We produce an estimated 92 million tons of textile waste, and 60% of the manufactured clothing is made of synthetic fibers. So I wanted to create a biotextile by isolating the UV absorbing biopigment MyC, which is found in cyanobacteria, to produce a bioink and incorporate this with a non-living biotextile grown from cellulose to create a garment which aims to replace synthetic UV protective clothing. And obviously, growing living materials produces 100% natural and fully compostable biotextile, and it challenges our current relationship with clothes, as the living textile is now dependent on the user. The bottom left diagram was just an original concept of the design with the cellulose and MyC bioink. Next slide, please. So my first aim was to keep it simple, define the problem scope, explore the potential materials, generate the plasmid in Benchling, and develop a methodology for material fabrication of the separate systems. So first of all, the living part of the material is the MyC pigment, which is transformed into a bioink, and then the non-living element is the cellulose fibers. I gained inspiration from research papers that looked into cyanobacterial pigments in environmentally friendly sunscreens to find the plasmid that I needed for this project, which was PET28A+. And then the methodology for the separate systems before I combined them would be to use a wet spinning technique to suspend the bioink in hydrogel and then weave this yarn into the cellulose fibers, and then the weaving would be done with the jacquard loom. Next slide, please. So the restriction enzyme digestion with Benchling, as I said before, use the PET28 plasmid, and I inserted the MyC gene into this. And you can see on the top right the resulting transformed plasmid. And then on the bottom, I've drawn out how the textile fabrication process would work, where it would start with the spinning solution, which would involve incorporating the plasmid that was already created and integrate it into the textiles with the cellulose. And then the life cycle would, during the life cycle of the product, it would absorb the UVA and UVB rays from the sun, and then it could be put into the soil and degraded back into the earth afterwards. Thank you.
Hey, Holly, great job. Wonderful presentation. Thank you. Questions from the TAs? Yeah, Ben, go for it.
Great. Thanks so much, Holly. Amazing project. I just want to ask you if perhaps you have searched any protocols or how you will be validating that your wearable or the protein that you're using are UV suppressor or UV protectant. I don't know, maybe you have searched for some tissue culturing or bacteria tests to see their survival, something like that. I don't know.
I haven't actually, but that is a good point that I should. That will be for future.
Yeah, for sure. Take that in account because you need to validate it. It's
Yeah, of
good with
course.
bacteria or with cell. Great job.
Thank you so much.
Okay, Holly, great job. Thank you so much. Just a little note for everyone. We're more than halfway through this first session. We're going until 12:30 Eastern time. We're gonna take a group photo, by the way, at 12:30. We're gonna do one at the end of each session, so hang around then. Up next is Eduardo. Eduardo, are you there?
Yes, I'm here. Everyone can hear me properly?
We can. The floor is yours.
Good. So yeah, whenever you tell me, there you go. Can you go to the next slide, please? So yes, hello everyone. My name is Eduardo, and as you saw, my project is about bacterial pigments and their use for tattoo and cosmetic applications. But when I say bacterial tattoos, I don't literally mean this. I mean using pigments produced by bacteria, such as violacin, which is produced by the bacteria Chromobacterium violaceum. This compound has several interesting properties for industrial and biocide uses, which make them really interesting for their use in tattoos. For example, like their anti-inflammatory or antibacterial, their anti-inflammatory or antibacterial properties. However, pigments like these have really complex metabolic capacities. Violacin requires 5 different genes to work together if not, the whole process falls apart. These genes, Vio A, B, C, D, and E, need specific expression levels to keep the process balanced towards the production of our desired compound. Next slide. Sorry, David, next slide.
Is this the slide?
There you go. So yes, the first step in the project was to assemble a plasmid containing all violacin genes with their individual promoters and RPSs and a resistance marker. 6 different fragments from that. And whoa. No, that's the third slide. What happened? David, we changed to the second slide already.
Yeah, you have a bunch of animations, so it's a little, I'm not sure exactly what you want to show.
No,
right now,
it was just like let it. It was supposed to just it was just gonna play by itself, and I would just let you know when to go to the.
I see. It was a video. Yeah, I wasn't sure what was going on. The
Yeah,
slide
yeah.
just changed.
Sorry.
Sorry. Where
So
should
yeah,
we
we
now?
could just go to the previous slide.
Here on initial experiments.
Yeah. It was.
Okay,
That's fine. Yeah, the fragments and the corresponding primers were designed and ordered by from Twist Biosciences, so we could set up PCR reactions out of our hanks and reassemble the plasmids with Gibson. This plasmid would then be transformed with E. coli into E. coli BL21s, and then the whole assembly process could be automated with Opentrons. But in this case, since it's just an example, there's only one construct. But the procedure could be used to design combinatorial libraries, which and then we would use 96 well plates. This could then be measured on a plate reader in order to identify the candidate with the highest producers of pigment. And then for the purification, we could just use high-speed centrifugation and a method for cell lysing, such as an incubator. This will give us a final yield of the pigment of interest, in this case violacin, which is soluble in ethanol, which makes the process fairly easy, but other pigments would require other compounds. Next slide, please. Now, once we purify the pigment, where do we go next? First, we would have to find the ideal tattoo formulation for each pigment. Then one of the biggest hurdles, which is the biosafety test, where the project could like sink or swim. And then if it goes well, the scale-up process. Eventually, we could work with some of the multiple color bacterial pigments that exist in nature, such as we have in this diagram. And since we know the biosynthetic pathway of many of them, we could know which genes to work with. So we have some examples for like it's not working again. Beta-carotene, pyroverdin, or indigoidin. For human consumption, they would be ideal candidates to head this new stage of development. In the best of outcomes, we could eventually move towards pigments from other sources or use intelligent design of proteins to create novel pigments. There's even some research about the possibility of incorporating biosensors as tattoos. Thank you.
Okay. Eduardo, great job. Sorry about advancing the slide. Actually,
That's okay.
when the slides started changing, I was like, I didn't understand what's happening. I was like, is somebody
No,
else
no, no.
controlling?
Sorry, it was
Yeah,
all automated so that I would like when I told you next slide, it was just gonna be like everything would go
Yeah.
along with what I was saying.
No, I understand. No, but really, really great and innovative structure for this. Ben, quick question.
Great. Thanks so much, Eduardo. Amazing project.
Thank you.
I have a question for you. Can you elaborate a little further on how you will making your tests for assure the security of your dice, and also how you will know that you don't have any immune response from the person that you only that. Thanks so much.
This is a lot of this is essentially like very speculative, right? There's no. I haven't tested this. I haven't done any of like I'm not like full on biohacker just testing the things on myself just yet. But we have experience like in this image on the right, we have experience on like some of these pigments are being used for example, food dyes. So we could take those as an initial list of pigments to work with, since they don't cause already an immunoreaction. Obviously, the skin is different, but this could be a good list of initial starters to go with.
Great, Eduardo. Thank you so much. Great job.
No worries. Thank you.
Awesome. Moving next to Valeria. Go ahead. The floor is
Hi.
yours.
Thank you so much. Yeah, saying hi from Germany, even though I'm originally from Mexico. And I'll just get started. Next slide, please. So the problem that I'm trying to tackle is that essentially the textile industry is one of the most polluting industries in Europe. And the thing is that at the end of a garment life cycle, the user has to have the desire to recycle it, like bringing it to a facility. And then like sometimes it's an obstacle, but also on the other hand, the supply chain is quite complex to achieve like this proper recycling. And while there are many materials used, it's difficult to have them not contaminated and so on. So many different problems to actually recycle textiles. And what I found is that like from all the variety that we have, about 70% is synthetic fibers, and from which 90% is polyester. So what I was trying to like challenge is like, what if instead of recycling at the end of the garment life cycle, we could change the status quo and have the garments with some enzymes that could recycle them already integrated into the garment. So kind of like a self-degrading textile. Next slide, please. The thing with polyester or PET is that it can have like an amorphous or a semi-crystalline structure. And there have been already like some advancements to recycle amorphous structures. These are like typical that we see on bottles, on PET bottles. And we have like some examples of bottle to fiber and so on. But when we are speaking about polyester textile, the main challenge is that it has a semi-crystalline structure, and this makes it like way more resistant to enzymatic degradation. I found out that there's this company Carvalhos, who is working with another chair. Could they develop like a laboratory scale process to recycle this crystalline polyester? And I basically like got inspired on their process to try to like find a scalable or like industrial scale processing. And the main challenge with this is the energy cost. So what I wanted to find, next slide. So what I wanted to focus is like to design like an entire workflow to have like some minimal human assistance for the fermentation using OpenStrong. And the idea is that like we can do like this fermentation right after the cloning steps until scaling in large volumes. I already found like a couple of issues with that. I don't think that it can be fully automated, to be honest. And together with this, like it's also like the challenge of having this protein production. So I selected using cutinase, embedding into a plasmid PET28A, which I found to be like the most like fast for my aim. And then at the end create this textile proof of concept by embedding this cutinase into the fabric. So, and I would do this by covalent immobilization. That is a very stable and water resistant way. So yeah, that's basically what I was working on. And yeah, I would love to hear your questions.
Okay, great job, Valeria. Wonderful. Questions from the TAs? I guess one question I have for you, like for these kind of material projects where you're incorporating kind of living organisms, what do you think about that interface? How do you keep the, you're embedding the protein in the fabric? Can you talk a little bit more about the interface between your molecules and then the textile?
Yes, definitely. Well, I think like, there are like already like some ways to have, to like make it possible to be embedded, but definitely it's like a biosecurity concern, like how much safe is it for humans to have these enzymes next to our skin. And actually, like what I found out, like, so I did this with cutinase, but actually then I decided that probably cutinase is not the best one to have next to our skin. Instead, like some esterases could be a better alternative, because I found out like when cutinase like breaks down PET, one of the compounds has some controversial opinions about like health security. So definitely I thought like maybe it's just like better to select like another enzyme to make this feasible and not just scientifically possible.
Great. Okay, awesome, Valeria. Thank you so much. Great job. Kavya, are you there?
Yes, David. Could you refresh this presentation once in case the slides haven't loaded?
Oh, you need to refresh. Hold on.
Maybe you need to refresh the slides, the recent ones.
Okay. So just in general for folks, we really want to make sure that you're not updating the slides anymore. We lose a little time when we do this. So just give one more second here. Oh, so sorry, what is going on? Okay. Okay, go ahead.
Yeah. Good evening, everyone. So I'm currently working in a lab where we are working on certain strains of microalgae of throstochondrids for algal oil secretion, EPA, and DHA. And in addition to that, we are also working on a few cyanobacteria and genetically modifying them for certain bioproduction of high-value, low-volume compounds, and few other compounds like biosuccinic acid, 2,3-butanediol, and mannitol. So since we are also advancing to few other compounds which we are also integrating in our platform technology, I thought of creating a model, a computational model, which can predict the yields of compounds, because when I am observing the researchers who are working on the bench, it's taking a very long time for preparing each of the cultures, isolating the wild type strains from the nearby lakes. So I thought of preparing this model so that we can create the flux-based analysis for every strain that we are developing computationally to predict the yields. So some of the compounds that we are looking at are therapeutic proteins. Some of the compounds like nitrilases, alcoholic dehydrogenase, which will be used for some of the API synthesis, which we are having in our pipeline. Next slide, please. So I also want to give a background on the work that I had done last year with Fab Academy. So I got graduated from Fab Academy last year, during which I created this project Swasa, which is a breathing library. So the idea was to basically look out for the challenge which we faced, create community installation sort of an infrastructure, because there is always a lack of real estate and difficulty in finding land and space for cultivating algae. So to for the POC level, for some of the proof of concept level, I thought of creating little structures like this, which can be placed at community installation, which can serve as an algal photobioreactor. At the same time, it can also be used as you can see, this is a bookshelf, sort of a library. So they place a satellite library every week in the lab. So in order to place the books, this could serve as a library. At the same time, it will also be a photobioreactor. So every strain that we are developing for scaled production, we put it inside this Swasa, and we basically harvest the algae every week. So this part of the project in HTGA, I am exploring how this computation platform can be created. So some of the compounds that I am exploring right now are quercetin. So astaxanthin, zeaxanthin, and the final derivative quercetin. So for creating this in Thraustochitris is something that I am planning to develop during the final project. Next slide, please. So this is the methodology that I'm planning to develop, create a metabolic network of all the processes that's happening within the target model organism. So I have some limitations of showing the results of the work that's going on, because that part of the work is slightly confidential. So I'm planning to translate the same work with some of the open source models that's available, and basically create the yields for certain compounds. So some of the compounds that I'm looking at, intracellular acetyl CoA, succinic acid, and some of the other derivatives from the TCA cycle. And I'll be presenting that as well in my final presentation. So that's the scope of my work.
Okay, Kavya, great job. Thank you so much.
Thank you, David.
Questions from the TAs or anyone? I have a question. So I wonder if you could just say a little bit more about why you choose algae compared to yeast or E. coli or some other more common strains, common kind of models.
Got that. Because the platform that we are aiming to develop, we are also planning to make it a carbon negative biomanufacturing platform. So in Swasa, there is also a lot of sensors that I had put in when I had made that product, so that it can also track the amount of carbon dioxide that's absorbed by the algae. So we see when we are able to scale this as a part of the production, we can place these photobioreactors in some of the energy intensive areas like thermal power plants, or maybe other cement plants, so that their flue gas can become the feedstock for them. Like basically the carbon dioxide from the flue gas can be used by the algae. So some of the algal strains will also require very minimal sources of expensive carbon and nitrogen based feedstock. So the idea was to avoid expensive feedstock for the target organism that we are using. And I also got inspired by one of the lectures, self-reproduction. So I'm exploring that as well. So how we can get to that.
Great, Kavya. Thank you so much. Great job. Asmin, go for it.
Can you hear me?
Yes, go for it.
Hello everyone. I'm Armentos Wasi from Bangladesh. With Bangladesh to do that, I will talk about my simple project on designing better lignin degrading enzymes for biomass valorization. Next, next slide, please. Okay. Okay. Is the slide changed? Yeah. Okay. Lignin is a complex and rigid polymer found in plant biomass, and it's a major bottleneck in uh, converting biomass to useful biofuels and chemicals. It's a stubborn structure of which is hard for enzyme to break down, and which also limits the efficiency of sustainable and renewable biodiversity. To address this, I focused on computationally designing novel lignin degrading enzymes. The core idea is to create better synthetic enzymes that are more effective and stable under industrial conditions. Next slide, please. Okay. I started by I started by analyzing two natural enzymes that degrade lignin, like one G baby and one KOI. These two enzymes are uses different mechanisms like heme and copper catalysis, making them uh, ideal templates. So using RRF diffusion on those templates, I generated some new protein backbones, guiding the model to preserve key catalytic residues like heme and copper binding moieties. Then I tuned the model to ensure that the like bulky lignin fragments like guaiacol and cincol could fit inside the AAK site. Next, I used protein ARPNN to design amino acid sequences for this new backbones, optimizing for both function and thermal activities. And then I used AlphaFold 2 to validate the sequences and verify the new generation. Next slide, please. So, that was the AM1. The AM2 is in silico validation. So I haven't completed the validation fully. So for validation, I had two plans. So what is like use Vina and like alcohol multiple to perform like docking standards perform docking studies and see like how they like how the binding forces and affinity functions work. And then I am also planning to test industrial viability by testing using like high temperature and high pH environments using like molecular dynamics tools like Genomics and GlowMax or like open ARM. Then I have also plans to plan to use like Foldex to check the like stability effects of the mutations or so. Then
I'm so sorry to interrupt you, but excuse me, I'm so sorry to interrupt, but we're actually at the 5 minutes. So I apologize, but we're gonna actually have to wrap up and move on. But great job. Thanks so much for the great presentation. And if TAs could ask questions in the chat, that would be great. Thank you. I'm sorry to interrupt, but we're actually over the 5 minutes now. So thank you so much again. Okay, so questions in the chat for Azine, and up next, Hansen, are you there?
Yes, I'm here. Can you hear me clearly?
We can. The floor is yours.
Okay, hello everyone. My name is Hansen Jonathan, and I'm an undergraduate microbiology student from Bandung Institute of Technology in Indonesia. So I'm going to present my final project with the title Genetically Modified E. coli BL21 for mercury and microplastic bioremediation with biocontainment contingency. Next. So it is not a surprise that our ocean has been an important reservoir for a lot of anthropogenic compounds, specifically mercury and microplastic. So my idea was to build a genetically modified organism with building genetic circuits that would enable the organism to degrade those compounds, but with some boundary measure that is for biocontainment effect. So as for the context why I chose E. coli BL21, so actually I wanted to use Halomonas as a natural casis, but the circuits I have built, it is not compatible with the Halomonas because some of the parts require some certain polymerases, and it can only work in certain strains in E. coli, specifically E. coli BL21. Next. So as you can see in the left side, there is the logic gate for my genetic circuit. So the idea was to build an OR gate. So the cells can only survive if there are mercuries on the environment or they are on the biofilm form. So they must be attached to the sediments in the deep sea, and they form this biofilm. And if they escape this biofilm, they will produce this toxin gene that will make them do suicide like that. And so these requirements, both of the requirements, the mercury and autoinducer as the compounds of biofilm must be met so the cells can produce the mercury binding protein and the PTase for microplastic. So as for the right side, that are the genetic circuits I have built and how I optimize it and analyze it, the codon adaptation index using GenScript. So that is the result. Next slide. So in the left side, that is the digest and ligate process. So the main journal I use is suggesting that I should make a low copy plasmid using PSEFA 221 for enhancing the biocontainment effect. As for the right side, it is the downstream application of how my in silico methods may be confirmed and do further analyze. So that is from me. Thank you so much.
Hey, great job, Hansen. Questions for Hansen from the TAs or from anyone? Yeah, I guess I have one question. I know you're at, I believe you're at BioClub Tokyo. Or what are some of the first wet lab experiments that you would like to prioritize in your process?
I think it is to culture the bacteria and before that transform it first, because I have done some transformation, but it takes a lot of time, and sometimes the efficiency and the effectivity, it is not really high, especially if using heat shock. So I will prioritize using transform first, then culture the bacteria, because it takes a lot of time to incubate the bacteria.
Okay, all right, Hansen, great. Thank you so much, and appreciate your bioremediation project. Thank you so much. Great job. Up next, we have Beatrice. Beatrice, are you there?
Yeah, I'm here. Yeah.
Okay, Beatrice, the floor is yours.
Okay, so can you press to the slide, please? Yeah, so I decided to approach the version of 1.7 that it's a human protein in chronic pain diagnosis. It's known nowadays that the treatments are not exactly the best for this type of patients, and it's a disease that has a lot to know yet. But I I tried to mix and fuse the NAV 1.7 with a green fluorescent protein in order for us to be able to quantify the activity and the expression of this protein and monitorize better these patients and maybe give a possible early diagnosis of it, which I will explain a bit further. For this, I would use a fluorescent reader for us to be able to quantify it better. Can you pass, please? The slide. Okay. So on my aims, my first aim which is the one I've been working for so far, is basically try to analyze and engineer this fusion, and see if it was possible without it, the NAV 1.7 losing its function. And the aim 2 is try to test this in a lab, try to fuse it. Firstly, I would start with mammalian cells, so we could see and adapt better this project and quantify it and try to find ways of finding a pattern for it. My aim 3 would be probably when we start clinical trials, if possible, is not only staying in the NAV 1.7, but a mix and combine all the proteins that we know so far that have a huge impact in pain activity and inflammation on this type of patients, and try to define this pattern of combined proteins. I would take samples from either blood or spinal fluid through a spinal tap or lumbar puncture. Yeah, this would be my aim 3. Can you pass, please? Oh, I cannot see. Why doesn't it appear? Can you maybe refresh it? I don't know, because it appears in my I don't know why it's not appearing.
Yeah, let me try to refresh.
Yeah, because it's sorry.
Okay. Is this your third slide?
Yeah. So what I've done so far, it's basically analyze and see the best way to introduce the green fluorescent protein without making it lose its function, which is probably the C terminal. And I would have to remove or take out the stop codon of the NAV 1.7, introduce the green fluorescent protein, and continue the sequence. I would have a final product that is the one I'm going to work on benchling. So far, I've used Chimera X and AlphaFold, but I would start with benchling to try to get this final product of a promoter. I would have the NAV 1.7, a flexible linker, which I'm planning on using the one right up there, and the green fluorescent protein, and then I would get the rest of the polymer. This would be the final product I would obtain. I want to thank to my mentor and the TAs of Life Labs for all the support. And yeah, that's it. Thank you.
Hey, great job, Beatrice. Dominika.
Yes, great presentation. I was wondering whether you put thought into selecting the GFP molecule, because as you might know, like different fluorescent proteins or even like derivatives of GFP have different strengths of fluorescence. Yeah.
Yeah, I am trying to consider the best one. So far, I was more concerned of finding ways of introducing it at all on the NAV 1.7. I was having a hard time trying to understand it. I even tried to see if I would use intercellular loops or extracellular loops, which I ended up with a C terminal, because I found an article where they used fluorescence for with other purpose, which I have on my Notion if you're curious about it, but I'm planning on following a bit of that just so I am able to make this fusion work better. I don't know.
Okay. Definitely look also into like fluorescent potentials of different GFPs to optimize your detection device a bit more.
Yeah.
Great. Thank you so much, Beatrice. Great job. Moving on, we've got Mikhail. Mikhail, are you there?
Hi, yeah, I'm here. So hi everyone. My name is Misha, and today I would like to present my BioEd platform, MonoEqualizer. Can you get to the next? Oh, perfect. So my project focuses on how we can bring synthetic biology closer to the public through accessible bioart through accessible bioart. While synthetic biology is advancing fast, most people have little idea of what it is. In fact, 83% of Europeans surveyed had never even heard of the term. In Eastern European countries and even in some part of the Netherlands, where I study, educational institutions, they lack the proper exposure to synthetic biology. At the same time, we already have the bacteria that produce a variety of pigments like purple violocin, blue indigoidin, different types of chromoproteins, but a very small amount of affordable kits exist actually for educational purposes. So our goal is simple: to create accessible kits that allow students and artists to grow pigments and bacteria and control the expression using light inducers. And my vision is grow art, learn SynBio, and change minds. You can skip to the next one. So here are my 3 aims. So the first one would be to engineer optogenetic circuits for pigment production with BL21 E. coli strain as chassis. Then the second one would be to create a toolkit exploring the potential of different bacteria to express the same pigments in the microbial consortia. For the third aim, I would like to create a bioart platform with AI-driven painting design. So my experimental workflow, first we start with designing the plasmids using the SnapGene that encode the different pigments and assembling them with a Golden Gate assembly. Once the sequences are finalized, I order the DNA fragments from Twist Biosciences. Then when the plasmids arrive, I verify them using the PCR and then start with lab validation. Yeah, the constructs are transformed into BL21 in the lab, and afterwards the OpenTrons liquid handler is used to disperse the bacteria across the petri dishes. And then comes the fun part, using LEDs to induce gene expression, right? Depending on the system, whether it's PDON or CASCAR, we use different wavelengths of light to trigger the production of pigments right on the plate. And once validated, the reagents are built into the toolkits. Basically, the plasmids, the reagents, and a booklet will be packed in this kit. And afterwards, when I develop this kit, I'm planning to collaborate with different institutions like LifeFabs, Maastricht University, and Hasselt University, where we kind of I study now. And eventually, I would like to build an online database of different already made plasmids with different chromoproteins that people can order from. And afterwards, I would possibly contact different PIs and galleries for even more exposure and gaining more public. So going to the last slide. For the results, I have designed 2 plasmid fragments. So the one with red chromoprotein with MEFRED, and the one with pink chromoprotein. Yeah, like for future considerations, I would like to create and validate the kit actually in the lab, and then expand the palette of the pigments, scale up with help of bioreactors, and actually in the end bring the kit to the market. So, and for bioethics and biosafety, the strains would be used that are that can only grow in the contained environment, and also all of the products can be reused, and it would also trigger the the popularization of a concept of responsible innovation. So that's all for me.
Okay,
and also
great.
and also I would like to further design the light inducible kill switches for that could be also integrated into this system.
All right, so we'll have to wrap up there. Great job. Thank you so much. We have a lot of questions, which is great. But can all the TAs ask questions in the chat as we are over time to your presentation? But great job. Thank you
Thank
so
you.
much. We'll do the questions
you.
in the chat, and then we'll move on to the next speaker. Daphne, are you there? I apologize if I'm mispronouncing your name.
It's okay. Hi. So my project is to make a self-sustaining luciferase system for brighter biolighting. Can you go to the next one? So I wanted to use Gaussian luciferase system because I looked at some of the systems that were used before. Can you wait, next? So this is a plant lighting system where they used firefly luciferase, and the main issues with this is that because it's not producing its own luciferase, it's only lasts 21 hours, and also it's not very bright. So next, I suggested to use Gaussian luciferase, but this is a ATP independent. It's one of the smallest known luciferases, and it's also the brightest. Next, but it doesn't have a known biosynthesis of its luciferin, which is cholinterazine. So my first aim was to come up with a pathway to make cholinterazine. Next. My second aim was to test any viable pathways, and then my third aim is to express these in photosynthetic organisms so that it's a self-sufficient system, and I would put it in PDUSK so that it's expressed in the dark. Next. So this is what the luciferin looks like. It interacts with the luciferase and produces this really bright reaction in jellyfish. Next, it's thought to be a cyclic form of the tripeptide phenylalanine tyrosine tyrosine, but what's not known is how you get from the tripeptide to the luciferin. Next. Next. I suggest using a cyanobactam cyclization system, which next has a these recognition sequences that can be cleaved by proteases, creating a C-terminal nucleophile so that the core sequence can be cyclized, and the core sequence is variable, so we can just input the FYY motif to cyclize that. Next. And you can also use next PAT-D enzyme to add any additional modifications to the backbone before the cyclization process. Next. And one issue with this is that it does this by interacting with serine, cysteine, and threonine residues, which our motif doesn't have. Next. But because the amide nucleophile is going to be in really close proximity, as in with the GFP system I'm showing here, I was also thinking of calculating the possibility of having autocatalysis after the initial head-to-tail cyclization occurs. Next. There's also isopenicillin and synthase homologues in organisms that produce cholinterazine, which creates this double ring in fungi. Its actual function is not known, but I was thinking of docking it to these peptides to see whether or not that could be useful. Next. Next. So I'm going to come up with these different pathways and calculate the Gibbs free energy associated with each of the reactions fold my constructs and use molecular docking simulations to dock each of the enzymes to my cyclization constructs, and also my likely products or analogs to the luciferase, and see if it's positioned where it should be, and if it could potentially lead to a luminescence reaction.
I'm so sorry.
And
I'm sorry. I'm so sorry to interrupt. I'm so sorry to interrupt.
Sorry.
You're actually over your 5 minutes, so we're gonna have to
Okay,
wrap up
yes,
there.
correct.
But thank you so much. Great job. And we'll have some questions for you in the chat from the TAs. So thank you so much again. And sorry for interrupting. Up next, Itai. Itai, are you there?
Yes.
Okay, the floor is yours.
Thank you. So hello, everyone. Today I'll be talking to you about this final project idea I was developing of a modular biosponge. Next slide, please. And so the essential idea of this approach is using already tools that have been out there, also somewhat clinically translated, of using ferritin as a method to deliver drugs and also for vaccine development. My suggestion or addition to this approach is to use a modular ferritin nanobody for protein capture. This could be used for a variety of conditions to remove toxins, cytokines in the case of cytokine storm, capture misfolded protein, or use for detection. The aims, as you can see on the bottom right, include first a proof of concept of capturing GFP. The second aim in later stage would look at trying to remove more clinical relevant proteins such as interleukin-1 and TNF-alpha. And the third aim would focus more on clinical translation, including different type of samples such as a blood bag and beyond. And what you see at the top left is the essential idea when you start relying on the 24 subunits of ferritin. So we are using here a SPI system where SPI tag and SPI catcher bind together, creating a covalent bond. And using that, we can combine 2 units, one of ferritin and SPI catcher, another of nanobody and SPI tag. And when combining them, we can create our construct. Next slide, please. Here you see a description of the workflow as I see it. It includes first in the top left, the DNA design, which included some linkers, the ferritin itself, and the SPI component, along with histag for purification and the enzyme sites for digestion, for cloning and ligation, which you can see in the bottom left. From there, we can then transform culture and select the bacteria, lyse and purify. We can verify with SDS page. And then because of the modular nature, we can try different stoichiometries and assemblies regimens. And then in the bottom right, we can move from adding it to a GFP solution, centrifuge, and then look at the supernatant and measure the fluorescence remaining, which can assess how effective it was. Next slide. And so where I hope this ferritin biosponge can go next, I'm looking at things that ways of improving it, which can include things like picking the best nanobody, optimizing the ratio for best response, improving bioproduction maybe beyond the generic bacteria, things to avoid aggregation, immune response, non-specific interaction, cell penetration, if I hope to focus on the blood, looking at clinical translation aspects, how we can deliver it, whether it's IV bacteria, adding factors to try and remove it more effectively, the models. And finally, there are a myriad of options at the bottom. Like I mentioned, some protein removal, protein detection, DNA detection, and other non-protein targets, maybe through aptamers. That's all I have, and thank you all for listening. I'll be happy to hear your thoughts, and thank you for this class.
Great job, Itai. Thank you so much. Adrian, question to you.
Thank you, Itai. That's very great. So first of all, I wasn't very sure if you talk about this in vitro or in vivo. And if you talk about the in vivo biosponging, how do you make sure that you don't biosponge other things that are not actually pathogens or things like that, basically off-target effects? Thank you.
So these are definitely you make a good point. And these are things I'm considering where you've seen this slide, what to avoid. So at the moment, I think, again, right, the initial proof of concept is in vitro in a GFP solution, but definitely if it goes to translation, I have to verify and make sure that it doesn't bind to non-specific targets and all these other risks. Yeah.
Thank you.
Dominica.
Yeah, thank you.
I was curious, how would you improve cell penetration? Like, do you have any kind of plans on that already?
So actually, it's the opposite. At the moment, the way I see it is not for cell penetration. And like I mentioned, ferritin has been more used for drug delivery, and then you want it in. I can think of like things like PEG coating to actually avoid penetration, to make it larger in size, so it will not go into cells.
Yeah, right. Okay, thank you.
Itai, thank you so much. Great job. William, are you there, William?
Hi there. Can you hear me all right?
Yep, go for it.
Great. I'm Will. I'm a designer focusing on biodesign and the intersection of biology and craft. My project, if we go on to the next slide, focuses on S. pasteur and how you might be able to use biocement as an alternative to gold lacquer in kintsugi. I'm really interested in how we sort of modernize crafts and preserve them through biodesign and kind of future-proofing them, not only for preserving heritage, but there are a lot of other ecological reasons in terms of resilience. And I think that there's an opportunity here to kind of expand the learnings and establish kind of what we might learn from them within larger scale applications like architecture. So my first aim was to basically harness microbial-induced calcite precipitation to bind fractured terracotta. I chose terracotta because it's porous, and then develop a composite substrate that supports bacterial growth and mineral adhesion. My technical aim, my second aim, was to develop a novel process that integrates bioproduction and material science with traditional craft techniques, a term that I call biocrafting. And then the third, more visionary approach is to basically bridge the gap between biotechnology and craft by establishing regenerative design practices. And so you go on to the second slide. I created a kind of experiment design cycle that goes from bioproduction to scaffold development and then to strength testing. And basically you go through iterations of this in a kind of material science approach to basically then take those learnings and then reapply them as you go through the cycle. And so with bioproduction, I was thinking about, you know, if a craftsperson was using S. pasteur, they might be working at room temperature, or maybe they have access to incubators at certain temperatures. So seeing how that might impact and look at the optical density within a spectrophotometer. In terms of scaffold development, experimenting with different kind of sand, twine, organic materials, and then seeing how the crystal sizes developed under a microscope and looking at uniformity of the crystals. And then in terms of strength testing, using kind of traditional engineering methods, such as tensile strength testing or impact testing to see how much force it can withhold. And then onto the third slide, I then started getting hands-on and dirty in the wet lab. This is my first time in the wet lab, so it was really just a kind of a learning by doing process. And so I was basically creating a process that I could then reiterate on and then develop out further. And so I'd started out with small scale samples, inoculating the media with S. pasteur, fracturing terracotta, labeling and tagging them so that I can reassemble them later, and then applying the materials in between the cracks and then inoculating with S. pasteur. And it was a really kind of interesting process just to kind of get hands-on with the material. But I think going forward, being a bit more rigorous with the scientific method is kind of where I want to go next with this process. And so really thinking about, okay, what are the different material composites? How do I understand, you know, combining hemp fibers with sand and sponge? How does that then result in different material properties? And then also thinking about the scale of scaffolds, really looking at kind of things at a scale of microns, as kind of designing at a larger scale. And then finally think about biocontainment ethics, like, you know, if this is a toolkit, let's say if someone who doesn't have wet lab experience is going to use, what is a kind of self-contained chamber where they can kind of inoculate, design, and grow all at once without allowing the S. pastöri to escape because it's a BSL-1 material. So I'm thinking of like some kind of chamber where you stick your hands in, but that's kind of to come, and I'm excited to keep developing this in the future. Thank you.
William, I love this project. This is great. It's a really great combination, I think, of design, art, and science, and actually very practical. This is something I can really see working. So biocements, I think one of the big challenges in working with S. pasteuri is that, the cements themselves can be not that solid, actually, right? It's like actually creating like really solid structures. I'm curious your thoughts on that and how to improve the kind of strength properties of your material.
Yeah, it's interesting. I think there's multiple ways of just inoculation and going through those cycles that I want to experiment with. So I think there's two methods, just kind of inoculating, then drying multiple times. I only did it kind of 2 or 3 times, but I think getting up to like 10x could be an interesting process, and automating it using Opentrons or like a dripper system so that you have this like continuous drying and inoculation cycle. Or you could also submerge, and I've seen submersion techniques, and so you would like leave it in the media for weeks potentially to try and really kind of boost the amount of S. pasteuri and urease and calcium precipitation that occurs within it. But yeah, it's kind of a learning by, and then there's the composites as well, looking at like hemp and sand and what the combination of that could be.
Great, William, thank you so much. Awesome project. Georg, I'm going to ask me your question in the chat. That'd be great. And Adrian, I think you're doing the clock. Make sure to let the clock continue to run negative. I lost the time there. Nikhil, the floor is yours.
All right. Good job, Will. That was awesome. All right, you can go to the next slide. My name is Nikhil Kumar. I'm part of GenSpace, where Will is as well. And I want to ask a question to start this. What would you say to a society centuries in the future? I started thinking about this partly because of Chris Mason's lecture. He was asking us to think, not in decades, but in centuries. And every week that went by in class, we saw, new technologies that seem to put us at an inflection point, not just for humans, but for life itself. So I started to think about this question some more through the lens of art, and none of what I'm showing right now is going to be sort of practical and product oriented, but I built some software for myself to translate English, text, so poetry into DNA sequences and then into amino acid sequences, and then I passed those amino acid sequences into the ESM fold, which I learned about in this class, to predict a protein structure. And I thought it'd be interesting, partly inspired by what we're learning about DNA storage and some of my previous work on memory and archiving and biology, to kind of see what these material forms would look like, when passing poetry into a protein prediction model. So at the bottom, I modeled out 3 proteins, one called Made by Humans, the other Assisted by Machines, and the last At the Dawn of the Next Revolution. And if you go to the next slide, the next thing I did was I made these into sculptures made out of metal. So I learned how to weld and bend metal, and worked with folks at a metal shop where my studio is. You can go ahead and click those two videos. And we looked at these 3D models of the proteins, and then I ended up going through the whole process and making metal sculptures, and even showed Made by Humans in a gallery in Brooklyn at the end of April. So after finishing this first sculpture, I thought of a few things. One, can I embed DNA sequences into the sculptures themselves? And tried to make a twist order, but the gene fragments were too short. Maybe in the future. And then can I grow the etching primer that I used on my metal before putting paint on? Could I even grow the paints? In the future, can I embed messages in the paints that I grow on the sculptures? Can the sculptures and the messages respond to their environment over time? Go to the next slide. I decided for the biology part of this, for the lab part of this, to try to do the etching And the TLDR here is our twist orders didn't come in time, but I tested yogurt and vinegar as etching acids on metal, and the yogurt ended up working the best, even though its pH was 4.5 or so, and the vinegar was around 3. The lab experiment that we're going to do is grow an E. coli that overproduced LDHL, so overproduced lactic acid. But I think now, after running this very first experiment, that we might want to add something that introduces more of a biofilm, because I think that's probably why the yogurt worked a little better than the vinegar. But anyways, I think, I definitely am going to continue making these sculptures and want to see how much more of the sculpture I can grow, in addition to using metal. Great. I think that's it.
Nikhil, so cool. Love the creativity. Really, really fantastic. Great back-to-back presentations with you. Well, that was awesome. Questions for Nikhil? Oh yeah, Dominika.
Yeah. Yeah. Do you plan on creating those sculptures for like future preservation? Do you want to make them like preservable so that they can be read actually in the future?
Yeah, it's a good question. I think, that was not the intent in this first version. I wanted to do something that felt a little bit more abstract and human interpretable, or no, human interpreted, not necessarily interpretable. I think, first of all, like ESM fold is just giving a predicted protein structure, not something that's like definitely going to happen given the amino acid sequence. Right now, I'm not as concerned about legibility, and I don't necessarily need this to be like a science education project, but maybe more of a thought-provoking, like thought starter. But I do think that there are ways to embed the DNA fragments into the sculpture to give it a quality of like being able to be decoded down the line.
Nikhil, great job. Thanks so much again. And just a quick note for folks too, Stephen is still placing DNA synthesis orders, and you still have 2 more weeks actually to do your documentation. So for all the listeners, so please do keep doing experiments the next 2 weeks and document your stuff. So we'd love to see that. Okay, Amparo, you are up next. The floor is yours.
Thank you. You can go ahead, please. So my name is Amparo. I'm from Chile, and I would like to present to you Synthetic Microbial Consortia for Coral Thermal Tolerance and Resilience. As a really quick background, as you may know, coral reefs are amongst the most biodiverse ecosystems on Earth, and they are being threatened by climate change, especially ocean warming. And this elevated sea temperature can disrupt the symbiosis between the coral and the photosynthetic algae, and that can lead to bleaching and potentially to coral mortality. Next, please. And so in this context, I thought about developing a synthetic microbial consortium using bacteria that is already native to corals and select them for their natural antioxidant activity and other beneficial interactions, and then engineer them to overexpress some of genes stress related. I would then have to evaluate the consortium's ability to enhance this thermal stress tolerance in a model in vitro coral and algae, and further down investigate some of the molecular mechanisms and extend these findings to currently endangered coral species in future stages for an eventual open field application. And so I guess for the first results, I came up with a selected consortium of 3 bacterial strains. Regeria, Endozoicomonas, and Alteromonas are already present in coral ecosystems and have some recorded beneficial interactions. So I thought about overexpressing NIF-H, for example, that helps with nitrogen fixation and nutrient provision for the coral. Also overexpress GROW-EL and HSP20 that are chaperone proteins to overall help the stability of the symbiosis in the coral. And Alteromonas also has a known antioxidant activity, and so I thought about overexpressing catalase and superoxide dismutase to boost this ROS degradation. Next, please.
And so in this overall experimental design, this is really general. In the plasmid design and transformation, which are key steps, I tried to came up with plasmids that had a broad range host plasmids, also selection markers that are compatible with marine life, and the promoters are temperature inducible. And the transformation protocols are really scarce. There are not many for the bacterial strains. So I thought about some conjugations and some electroporations protocols, but this would be probably need to be tested many times to actually get to the in vitro testing. And the data collection would consist mainly of photosynthetic levels, broth levels, phenotype, and even transcriptomics to analyze. And finally, to highlight a little bit of the vision, this would be one of the first uses of engineered bacteria at all in coral ecosystems, and it would have the potential to be scalable, and the biosafety would need more development to actually comply with different regulations and to be applicable in real life. Thank you so much.
Amparo, this is such a cool project. I actually do work myself in coral and related to some coral probiotics related to this. You should look, if you haven't already, into Blake Ishijima's work. He's a professor in North Carolina. He's one of the leading pioneers in coral probiotics. One question, probiotics, even in humans, for example, one of the big challenges is they can be very transient, like they pass through your gut, for example. They don't necessarily incorporate fully into the microbiome of the gut, for example. What are the ways that you think about actually kind of ensuring that the probiotics or the microbe consortia there actually integrate with the coral, as opposed to just like getting washed away in the ocean, or actually becoming embedded into the coral microbiome?
Yeah, I do not have a full answer. But I thought about and I read about the different stages and the establishment of the microbiome of the corals. And I think these strains would help to be like applied in the early stages of the coral, and so that it could establish for the adult life. And also in the previous in vitro test, the compatibility of the 3 strains would need to be validated before the in vivo application. I think those would be the main strategies.
Great. No, thanks so much. Really cool project. I love it. Really, really awesome. Great job.
Thank you.
Up next, Ahmad, are you there? Okay, Ahmad. The floor is yours.
Yeah, is the sound okay?
Yes, go for it.
Yeah, I'm Ahmed. I'm graduating as a medical doctor this semester. I'm actually, the last exam I will attend will be the next week. First, I would to thank you all about this great course, which was really very rich in up-to-date knowledge, especially about the protein design, which I'm very passionate about. So my project aimed to design a binder for HLA class 1. Next, please. Yeah. So you can see on the right, HLA class 1 molecule present on the surface of every nucleated cell and show intracellular peptides to the cytotoxic T cell. That helps T cell to recognize the bad peptide. And if there is a bad peptide, the T cell probably will kill the cell carrying it. This HLA peptide complex shed and float to the blood. So mapping every patient's HLA-bound peptide, remember, the thing we are interested in is the peptide on the HLA. Mapping that called immunopeptidomics. That can cover cancer, neoantigen, infection markers, and autoimmune targets. So current state of the art, the IMPASS mass spectrometry workflow from the Mann's lab in Germany already reduced the required plasma to just 200 microliter and can quantify over 5,000 peptides in 30 minutes. Great, right? But the IMPASS mass spectrometry still depends on biotinylated monoclonal antibody, which are expensive relatively, and mostly one test limited and hard to scale for routine clinical use. You can see the workflow in the image below. Actually, some steps are automated in a device like just like Opentron. Next, please. For this course, the project stayed fully computational. First, I extracted the entire version 3.6 IMGT HLA database, which contain a large number of HLA alleles, about 16,000 alleles. Then I aligned all class 1 alleles to locate 100% conserved residue between those 16,000. Then, as you can see on the left image, I generated a structure scaffold by AlphaFold Multimer and aligned it with actual crystal structure to see the quality of the prediction, which was very good. Then, on the targeted structure, binding pockets were predicted with several geometric deep learning tools. You can see examples in the ABC figures below of the binding pockets. I ran Bindcraft, which produced a lot of designs. Almost all of them were rejected. So, like the three red ones on the left and the right, they had a very good metrics. Nevertheless, they were rejected by Bindcraft along hundreds of designs. The reason for that, the Bindcraft is very strict in its filtering process. So, about Bindcraft process, which seems easy, let me tell you, it's actually not, at least for the first-time user. So it's been about a month of modifying the inputs and going through the trial and error. And 2 weeks in, I literally started waking up from 2 hours naps because I dreamt the design had finally succeeded. Once I had woken up, I checked the computer, surprise, no success. So I kept going on testing, and last week, let me just tell you, I might have danced. Yeah, I got an accepted binder. Next, please. So you can see the accepted one on the red above, which is very good. So the author suggested I still need about 50 to 100 accepted binder to really test them in the wet lab. Next, please. Oh, yeah. Stay on this one. No, sorry. Yeah. So other than generated binder in silico, the next aim cover expression and affinity testing, and further the actual using it in the pipeline involving mass spectrometry. So replacing antibody with recombinant binder decrease the reagent cost, hopefully making high throughput immunoproteomics feasible in ordinary hospital, even to those in underfunded regions. This means faster biomarker discovery, better prognostic test, and personalized vaccine for many more patients. And thank you.
Okay, Ahmed, thank you so much. We are out of time for you, unfortunately, for questions. So for TAs and anybody else, please ask questions for Ahmed in the chat. But thank you so much for the great presentation. Up next, we have Patrick. Patrick, the floor is yours.
Thank you. Can you hear me?
Yes, go ahead.
Okay. So the first slide, should I do something here? Thank you. Good afternoon. Thank you for the opportunity to share work on targeting intrinsically disordered regions. And here in the first slide, we are looking at SIRT6 terminal IDR, and it represents a significant channel. It has about 25% of proline, and the IDR is shown as an orange circle, and it's extremely basic. It's got an isoelectric point of 10, and it creates a really positive charge density in a motif called PKRV-CAC. And this motif communicates with DNA chromatin. And I think of SIRT6 almost like a friendly Scorpio with its core body as a Rossmann fold, and it communicates with DNA with this floppy, flexible, rapidly shifting conformation. And our core hypothesis is that with designing strategic peptides, we can induce specific disorder-to-order transitions in IDRs that have previously been considered undruggable. there are breakthroughs like small molecules that have shown that it is possible to target IDRs, venetoclax and idanazanotide, but they have demonstrated it's viable for specific interaction at a very, very pricey costing. It costs like $200,000 to $300,000. And compared to that, our approach appeals to a much more broadly applicable platform. So the design approach is trying to treat IDRs as opportunities to communicate and create by using pattern-based peptide design, create complementary interfaces with the IDRs and its unique physiochemical properties. So we are not looking to just design a particular therapeutic, instead trying to create a platform technology. So slide 2. Okay, we got to produce these peptides somehow, and it's difficult to produce acidic peptides. It's often a nightmare. They aggregate, they have solubility issues, and they provide really low yield and reliability. So the Sumo is a really elegant workaround here. It functions actually like a chaperone, and it helps the peptide both with folding and protection. And the expression system, you can see here in slide 2, it's a workflow that progresses from left to right in 5 steps. We start with a PET Sumo vector. We put that into the standard E. coli workhorse, BL21 for expression, and begin protein induction at a much lower purification tab. That grabs the his tag on the Sumo part, and nickel resin binds this tightly. Okay, each step is optimized for these challenging peptides with the percentages you see below each stage reflecting our efficiency. Okay, I must go on to slide 3. I'm out of town, I suppose. Okay, this slide brings together our initial computational work with design path and experimental validation path. The left panel shows our AlphaFold 3 predictions, and the second part is the peptide design strategy, our 3 strategies: electrostatic, complementary, where we have like a magnet, and then the second is a proline reads alpha helical inducing, and then aromatic with pication predict. And we have the modular cell peptide work, and then the experimental validation. We have actually this 4-phase validation map on the right, which systematically confirm these computational insights, moving from peptide expression described in the Sumo slide, and then through binding validation. And you got to show this in a test tube that the peptides bind structural conformation. Does it induce order? And we, this project marks a small but meaningful step towards making undruggable IDRs more accessible. I'm deeply thankful to you, David, and to How to Grow Almost Anything as a space to learn and explore and grow. And it's led to 3 early patent filings that reflect the questions we're still working to answer as we move forward. I'm open to collaboration and to keep moving and expanding in this work. And together we can explore new ways to understand and influence complex protein behavior. I'm not so experienced in timing.
No, you did great. You did great, Patrick. Thanks so much. Great job. We will move on to the next speaker. Actually, I would love to hear about the patents you filed. That's actually really cool. Maybe you can mention that in the chat. For those that are joining late, the time is a clock that's ticking. Either Georg and Adrian are taking turns. So just keep your eye on there in terms of the time. It's ringing at 3 minutes, and then that means you've got 1 minute to wrap up, and then we're taking at least a minute of questions. So just for those that are joining late. But great job, Patrick. Thank you so much. Ursa, are you there? Ursa.
Yes, hello everyone. Can you hear me?
Yes, go for it.
Okay. Hello everyone. I'm Isra Al Khalil, and it's my honor to be here. My partner is Mohammed Kraiz. The title of our research or the final project is in silico screening of plant-derived phytochemical targeting the N-terminal domain of EpCAM, a potential strategy for cancer therapy. So we decided to target the EpCAM, which is an overexpressed protein on the surface of cells in cancer like liver cancers and breast cancer, etc. So we decided to using such compounds extracted from plants and targeting this receptor or this protein on the surface of the cell. Our aim from this project is just to scan these compounds and choose the best one to do and develop the research. And the other thing, or the second aim, is to develop a low-cost targeted drug for cancers here in Syria. And why we choose this kind or this type of compounds, because it's safety, accessibility, and sustainability. Beside that, it's a solution for us because we are in Syria, we are struggling with the the research in labs. So I think, or we think, it's kind of a solution. So in terms of workflow, next slide, please. In terms of workflow, we began to we begin to just choose the compounds from the database, which is BibChem, and we just prepare the structure of this compound, convert the format from SDF to BDB, and then we configure the files through Autodoc Vina. Beside that, also we prepare the structure of the EPCAM using MGL, removing the H polar and just identify the grade books, and then we run the Autodoc Vina. And in terms of the result from this docking, next slide, please. We get these results that we choose the top 8 compounds from 30, and like you see the scores of the docking, minus 7.2 and etc. So we choose these compounds and we move to the next step to just next phases in our work to validate and improve the research with Muhammad.
Hello, I'm Muhammad.
So
Can
Muhammad,
you hear me? Hello.
Hello? Yes.
Okay. Hello, my name is Mohammed Krez from Syria too. After screening the plant-based compound, we continue the project to evaluate which ones could work actually as a drug candidate. We start the second phase with molecular interaction analysis using Discovery Studio, where we looked closely at how each compound binds to EPCAM. This helped us to understand which compound form more stable and specific interaction. Next, we did ADMET profiling using Swiss ADMET and the ADMET Lab AI to check each compound absorption, bioavailability, toxicity, and Lipinski rule. After that, we used BioMol to make sure that the compound were actually docking inside the correct binding bucket. And finally, we run MD simulation using NAMD, VMD, and Google Colab to see how stable the compounds stay inside the binding site under body condition. Can you move to the next? Can you move to the next slide, please? Next two. Next two. Sorry, I didn't notice that.
Another one. This
I
one.
already
This one?
talked about this step. Yeah,
Oh,
command.
here.
Okay. As a result, we reduce our candidate. No, before one. No, before. Okay. As a result, we reduce our candidate from 8 to 4 based on a strong interaction, such as hydrogen and hydrophobic Mount number. Then we rolled out more to compound based on ADMET analysis, where one was too toxic and the another one was low absorption. Can you move to the next slide, please? For the final two, we run MD simulation. Both compound were stable overall based on RMST analysis, but the compound 23, which is silibinine, show a strong and more sorry, the video still working? There's a notification happening here.
Yeah, everything's fine.
Okay, for the final two, we run MD simulation. Both compound were stable overall based on RMSD study, but compound 23, which is silibinine, shows strong and more consistent hydrogen bound over time compared to compound 9, which is glycerol. So we choose silibinine as the best candidate. These results show that silibinine is a strong potential inhibitor of EPCAM, with good docking score, acceptable ADMET, and great dynamic stability. In the future, we plan to isolate silibinine from Celebium plant tested on EPCAM cell line, cancer cell lines such as MSF7 or HT29, and maybe improve its delivery or structure by using aptamer, antibody, or motif. Overall, this project shows how we can turn traditional nature compound into a target cancer therapy, even using only computational tools. Thanks.
Okay, Ezra and Mohammed, thank you so much for your presentations. Questions from the TAs?
I'm trying to see the question.
Yeah, I guess one question for me, you know, can you say, so you've done a lot of modeling, and I appreciate very much the modeling work you've done. Can you say a little bit more about how you would try to experimentally test your designs?
I think maybe just I will give you my opinion. For the next step, I think we have to do a lot of validation and assays about the toxicity and other things in the compound levels or the chemical levels and the cellular levels. So I think it's complicated and difficult, and I think it will be our first step, like working in research in lab with Mohammed, and I hope that. I don't know if Mohammed have anything to add.
Honestly, I didn't hear that question clear, so I didn't answer. It's
Okay,
kind
well,
of network, not that.
Thanks so much to you both. Yeah, I was just asking about Mohammed, like what the experimental validation, like what would you do experimentally to test the particular designs that you made? But why don't you answer the questions in the chat? Yeah, why don't you answer the questions in the chat? We need to move on to the next presentation. But great job to you both. Thanks so much again.
Thank you, everyone.
Thank you.
Thanks.
Okay, MD Razel, are you there? I apologize if I'm pronouncing your name incorrectly.
Yeah, it's okay. I'm Mohammad Raseluddin, and my project is about first genetic circuit on Earth. Next slide, please. So hypothetically, the first genetic circuit on Earth had two genes, gene A and gene B. Gene A stops reproduction, and gene B does two functions. One is repress gene A, and second is repair DNA. Gene B does not do these two functions at the same time. When it does repress gene A, it does not repair DNA, but when it repairs DNA, it does not repress gene. So in normal condition, gene B represses gene A. So gene A does not stop the reproduction, so reproduction continues. But in stress condition, gene B goes busy repairing DNA, so it cannot repress the gene A anymore. Therefore, gene A expresses and stops the reproduction. And that's the hypothesis, actually, the hypothetical gene circuit. My hypothesis was that in nature we might find similar genetic circuits. So I did some search, and my chosen circuits were a VAB-BC-TS system, toxin antitoxin system, and MAS-EF toxin antitoxin system. So VAB-B, it's a gene, it replaces VAB-C, and reproduction continues. And VAB-C, which another gene, it overexpresses due to stress and stop reproduction. So this VAB-BC system and the MAS-EF system, they work in a similar manner. So next slide, please. So I found the interaction between VAB-B and VAB-C, and also the MAS-E and MAS-F protein using the string protein-protein interaction database. And the reddish and the yellow, that VAB-B, VAB-C in the left picture, and in the right picture of the upper side, the red is MAS-EF and the yellow is MAS-E. So they have quite spectacular interactions there. And the phylogenetic tree I generated based on the currently available sequencing data in NCBI. And I chose organisms based on their evolutionary significance, evolutionary significant domains and lineages. And I found out that the left image is VAB-BC system. VAB-BC system is found in many archaea, bacteria, and cyanobacteria species. And the a significant one was that Methanochaldrococcus janashi, which has a metabolism that dates back to 3.5 billion years ago. and it has this basic toxin antitoxin system. And in the right image, there is this MAS-EF system. There is Microcystis aeruginosa. It is a cyanobacteria, which is also present, which is also dates back to almost about 3.5 billion years ago. So it has this MAS-EF system. In the next slide. So what we do next is basically we can engineer a model organism like E. coli with this basis system and see if it shows better survival in stress condition. To do that, we engineer the plasmid using benchling, and what we do is we transfer this plasmid into E. coli, and we will have then two E. coli strains. One is engineered E. coli, and there is always present a normal control E. coli. So we will put these two category E. coli into similar situation like stress condition, like heat stress or cold stress, and see that if the engineered one show better survival than the normal one, using optical density calculation and statistical analysis like t-test to see if the value are significant at all. And this is my project actually. Thank you.
Okay, great. Thank you so much. Great job. Patrick, question from you.
Really cool story, and I was wondering, in an organism that has both gene A and gene B, gene A being the one that halts proliferation, what if you just delete gene A so that it doesn't stop proliferation even if you apply stress and even if gene B is busy trying to repair the DNA? And maybe that would be a better way to induce survival even in stress conditions.
Yeah, I have missed on very significant, very important detail actually, that in the first slide, it's actually, there is no circuits that I have found that exactly match the description of gene A and gene B. But the VAB-BC system and the MAS-EF system, they come closer. They exactly, the VAB-B does not disturb the reproduction. There is some genes it can effect that eventually stop the reproduction. That is and the VAB-C gene that is related to some sort of DNA repair mechanism, but it's not 100% like unnatural. So evolution is has its own way, and so it's not exactly the gene A and gene B. It does not exactly mimic the gene A and gene B. So I need to validate it actually through engineering, I guess.
Thank you so much. We'll leave it there. Appreciate you. Great job. Asma, are you there?
Hi, yes, I am. Can you hear me?
We can. The floor is yours.
Okay, thank you so much for listening, everyone. So my project titled In Vivo Fueling Stations to Recharge CD8 plus T cells and Boost CAR T cell therapy. So CAR T cells have been a very successful innovation in terms of cancer therapeutics, but one of the main obstacles to successful CAR T cell therapy is what's known as T cell exhaustion in the tumor microenvironment. You might have heard it's kind of a big topic. So what we do when we need to fuel our cars, for example, when they're depleted, is we go to the gas stations usually. And similarly, I thought we can engineer bone marrow mesenchymal stem cells, the BMSC cells, so that when they are implanted in the tumor microenvironment, they can refuel the exhausted T cells, whether CAR T or CD8, with healthy mitochondria to continue fighting the tumor. So in the next slide, so the main idea comes from leveraging these structures called tunneling nanotubes that many cells, including bone marrow, the BMSCs, naturally form to deliver mitochondria, even viruses, among other components of cells. And they deliver it to many different kinds of cells, including immune cells. And so the first aim is to engineer these BMSCs using lentiviral vectors to overexpress specific genes that regulate the tunneling nanotube formation. However, for the next part, an important loophole is that these TNTs, the tunneling nanotubes, are formed between many different cells, including cancer cells sometimes. And so AIM2 deals with specifying the target cell of the TNT structures so that there's a precise binding to CD8 T cells and CAR T cells. And lastly, we would need to validate this and prove that these mitofueling stations indeed reduce exhaustion in the cytotoxic T cells, which really would be wonderful to see. And so in the next slide, please. Yeah, I mean, the main application is to combat CAR T cell exhaustion through this mitochondrial transfer. And I have done preliminary, just very basic preliminary experiments to visualize this mitochondrial transfers from BMSC to CD8 T cells. But it's been just 2 weeks since, so I don't have result data yet. But another potential application in the future would be to then use these, mitofueling stations to, I mean, it has wide applications, potentially to implant them near lymphoid organs so that this could also optimize the cytotoxic potential of the T cells or immune cells in general in fighting solid tumors. I mean, yeah, the applications are wide, and I'm coming to an end here, but maybe I can talk and answer what my main inspiration came from, and it was from fungi, interestingly enough.
You can go ahead actually and finish. You've got 2 more minutes, but for questions. But yeah, finish your inspiration and then we'll ask that question.
Sure. So, I mean, my main inspiration came from really seeing, you might have heard of the Wood Wide Web, which this massive network that fungi form in forest soil, connecting all trees in the forest. And so when a tree needs nutrients, for example, glucose, can be delivered through these hyphae from one tree to another. And this is all regulated by fungi. And so in the first slide, if you quickly go back to the first slide, I had an image, I think this was from a confocal microscopy image. Yes, exactly. You can see that the green and blue, these are actually, I think, neural cells, but they also form these tunneling nanotubes, very, very thin, and you can transport mitochondria, viruses, many cellular components. So it's very interesting. And this is it's very visually similar to the hyphae communication networks in fungi. So
very cool.
Yeah, open to any questions,
Patrick.
suggestions.
Yeah, Patrick, go for it.
Yeah, I love it. It's such a great idea. And I think, mitochondrial transfer is also a really big topic. So very important work. I was wondering what determines the direction of that mitochondrial transfer.
So interestingly, depending on the cell, I found that it can either be like unidirectional transfer or multi-directional. So like either but in BMSCs, they only do unidirectional transfer. So transfer to other cells like T cells. However, like T cells can either receive or deliver through these tunneling nanotubes. So it's interesting. I don't understand why, but depending on the cell type, there's different directions of transfer.
Okay, we'll have to leave it there. Thank you so much again. We're on our last few presentations. Very exciting. And remember, we'll say it. We'll do a quick group photo before we take our break. Elif, the floor is yours.
Hi. Good afternoon, everyone. My name is Eslan, and I'm a physician graduated from Hacettepe University, Turkey. Today, I'm presenting my project focused on designing a local interleukin-10 mediated immunoregulatory system for hepatic allograft. Next slide, please. Organ transplantation is life-saving for end-stage diseases, but chronic systemic immunosuppression to prevent the most common complication of the transplant, which is organ rejection, leads to significant side effects like infections, myelosuppression, osteoporosis, and even malignancies. This challenge reminds me of a biological paradox highlighted by the father of immunology, Sir Peter Medawar, in his 1953 lecture regarding pregnancy. Half of the baby's genome is from their father, which makes them essentially a semi-allograft, yet it's tolerated for months without rejection. How is that possible? The key lies in the unique uterine environment, rich in immunomodulatory proteins like interleukin-10 and TGF-beta, specialized cell populations such as regulatory T cells and M2 macrophages, and non-classical MHC molecules like HEL, GE, and C. This environment actively fosters immune tolerance, inspired by the critical role of the positive feedback loop between interleukin-10 and regulatory T cells. I propose targeting specific axes as a toy model for inducing local immunosuppression in a transplant study. Next slide, please. My primary aim is to achieve target activation of endogenous interleukin-10 production in hepatocytes using selective organ targeting or short nanoparticles. Deliver is an ideal target for two reasons. First, specific short lipid nanoparticles demonstrate remarkable tropism for hepatocytes, with reported specificities as high as 93%. And secondly, hepatocytes possess the intrinsic capacity to produce interleukin-10 under certain conditions like chronic infection of trypanosome microglobulin F, suggesting that activating this existing pathway is more feasible than introducing a medium. That's why I'm utilizing a CRISPR Cas9 system used to detect pharmaceutical domain, which is a demethylase. My target is this CpG island at the minus 145 region of the interleukin-10 promoter. As the literature shows, its demethylation significantly increases interleukin-10 production. And my secondary aim is to testing the functional impact of the local interleukin-10 production using hepatocyte immune cell co-cultures and developing strategies to ensure uniform spatiotemporal IL-10 distribution throughout the allograft, creating a broadly immunosuppressive milieu. And my visionary goal is to translate this approach into a universal platform for local tolerance in solid organ transplant. Next slide, please. Experimentally, I will use a plasmid encoding the Cas9 Het1CD G50 fusion protein, the specific guide RNA targeting this minus 145 region, and an antibiotic resistant gene. The plasmids will be encapsulated within the hepatocyte targeting stored LNPs and transfected into primary hepatocytes. Successful transfection and viability will be confirmed via fluorescent microscopy. Promoter demethylation will be verified using bisulfite sequencing. And finally, IL-10 expression will be quantified using reverse transcripted qPCR and ELISA for mRNA IL-10 proteins. I documented the project in Notion page, including background resources and even a sci-fi novella that I wrote, which is inspired by this very concept. So thank you for listening, and I'm welcome to your questions.
Okay, great job. Wonderful job. Thank you so much for the presentation. Questions? One question I'm curious about, obviously this is you've got a great experimental design. What are your top priority experiments that you would try to do in the lab?
Actually, it needs so much things, but first of all, short nanoparticles are not experimented on plasmids. So first and the foremost problem is if they can carry this in an effective way or not. So probably my primary experiments will be based on it.
Okay, great. Yeah, thank you so much again. Great job.
Thank you.
Malika, are you there?
Yeah, hello everyone. I'm here. Can you hear me?
Is your video on or no video?
No video. I'm through my phone now.
Okay, no
If
worries.
it's okay.
Go ahead. The floor is yours.
Thank you. So hello everyone. My name is Malika. I'm a second-year biology student, and today I'm presenting my project on drug discovery targeting Trypanosoma brucei, the parasite responsible for African sleeping sickness. So I started this project by thinking about a very real and underestimated problem, I think. A disease that still lacks safe and effective treatment and continues to affect millions of people, especially in sub-Saharan Africa. So I wanted to ask myself, what can I do about this problem using just the tools that we've learned in this course? So I decided to explore how computational tools could be, computational bioinformatics tools could help identify potential drug targets. So specifically, I focus on an enzyme called serine threonine protein kinase, which plays a key role in the parasite cellular processes like phosphorylation, which is known as a crucial step for survival. So yeah, so using a series of tools, I followed a typical in silico drug discovery workflow. So next slide, please. So first sorry, 1 second. So first I retrieved the protein sequence from UniProt. Then I modeled its 3D structure using Swiss model, and then performed motif and structural analysis using Sprite and MIM tools. So after that, I conducted molecular docking using PatchDock, CBDock, and DocThor to identify how potential ligands could bind to the protein. So I used Discovery Studio to visualize the binding interactions and highlight key amino acids involved. So from this, I identified that alanine 667 and lysine 650 on chain A as two of the most important residues for hydrogen bonding, making them strong candidates for future drug discovery, drug targeting. So this is, of course, a very basic level of analysis, but I truly believe that even small steps like this, especially when done systematically and realistically, can move us closer to solving problems that today may seem too big. So yeah, in future, the results from this project could be further improved by refining 3D models, using more advanced talking tools, and eventually testing the best candidates in vitro and in vivo. Thank you.
Hey, great job. Yeah, really,
Sorry,
really wonderful.
could you please show the third slide?
Oh,
I forgot about it. Yeah, so these are the result part. Yeah, thank you.
okay, great. I guess maybe just as a question, since you've got a little bit of time, maybe you can share your results here from your last slide.
Yeah, so here are the results. As I said, there is alanine 667 and lysine 650 on the chain A. So these are the two most important residues for hydrogen bonding. So they might be helpful in the future to serve as a potential target points for future drug design and yeah, solving this problem. So in the best case, these residues could help guide us to the development of molecules that bind specifically to the protein and block its function. Yeah, so this is very basic, but at least, yeah, that's what I was thinking about.
No, great job, great job. Okay, thank you so much again. Great work. Let's move on to the next presenter, Xavier. Xavier, are you there?
Hello, yes.
Okay, Xavier, the floor is yours.
Thank you. Hey everyone, I am Javier Reyes Salinas. I am physician from Bolivia. I'm current ambassador for region in Latin America. Next slide, please. Okay, the final project title is Excalibur for extended chemosensory array for lithium binding and ultimate recovery. The place from where I am, Bolivia, has the so-called unisalar, where is the place where most lithium in the world is placed, almost 20% in the world. And at the same time, it has a dual interest, both in the generation of batteries and also in psychiatric uses for the treatment of bipolar disorder or even depression that is used to enhance GABAergic neurotransmitters. So it's very useful, but it sometimes gets to toxicity levels in blood. So it needs to be monitored with current tools that are very expensive nowadays. Next, please. The proposed project is using a RiboSwitch that is it was discovered in 2021 that is capable of detecting lithium ions, and with the activation of the RiboSwitch, it allows the translation to a reporter gene that usually is a protein. I have designed 3 constructs that each one produces different genes apart from the lithium signal that can be a trialized enzyme, GFP variant, or a peptide from the HCG hormone in order to see and explore repurposing of medical devices. The logic is that the activation of the RiboSwitch will allow to, for example, produce trialize that in the presence of triallose, that is a disaccharide, it will produce glucose molecules, and those glucose molecules will be detected by like a glucometer used in diabetic patients. Next one, please. The plan is that when the twist genes will arrive, we will transform them, insert them in by Gibson assembly in GL1 plasmid. And we have to transform colis in order to replicate those plasmids and do a process of self-regeneration in order to have the system and assess them with the proposing or with different levels of lithium chloride concentrations. With future prospects, with seeing the portability of these systems. Thank you.
Okay, great job. I have a question. So when you think about using this, you mentioned environmental use or testing in blood, etc. Can you say a little bit more about the sensitivity of the detection of the lithium?
Yes, actually, the idea, I was inspired by a Nigerian team from Germany, from IGN Brunswick, that they tested already the sensitivity of the RiboSwitch. It needs to be, especially in the clinical aspect, it needs to be a maximum of 1.5 millimoles per liter in blood, and it is capable of sensing in those ranges, especially for the toxicity registration.
Okay, great. Great job again. Thank you so much. And oh my God, I cannot believe it. We just did 3.5 hours of straight presentations, everyone. I'm going to stop sharing the screen for 1 second. And if we could all give a big, huge round of applause to all of the presenters from the past 3.5 hours. That was so epic. Such a fantastic job, everyone. Wow, 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 wow. Really, really blown away. So impressed. Amazing, amazing job, everyone. Really, really proud of all of the great presentations and all the great work. Before we take our break, it's going to be a very well-deserved break. We'd love to take a little group photo with folks. I know there are a few people in Asia that have been up all night. They need to get some sleep. And so what we're going to do is we'll take a group photo. So you can go find an object, like an object that you think is neat and interesting to bring into your photo. So we go find an interesting object and then come back. I'm going to actually grab something myself. I guess I've got my mug here. So we can all find an interesting object that you'd like to photograph with. So find something that you like. Give another moment here for folks. Ooh, Anandita, I love this. You have some coral there. This is great. Oh, look at all the cool stuff. Yeah, go grab your object. This is good stuff. Perfect. Great, great, great. Oh, everybody looks so good. So we're going to go on the count of 3. We're also going to say grow, just like we did in person. And we'll take a couple of pictures here. There. Oh, this is awesome. Here we go. 1, 2, 3. Grow. Grow, yes. 1, 2, 3. Great, great, great. And keep holding that. I'm going to get some more of the folks. Oh my God, so good. Grow, yes. Good. And then last one. Here we go. 1, 2, 3. Grow. Awesome. Everyone, that was so epic. Oh, I love the photos too. Yeah, this was so good. We will take a break for 1 hour and 15 minutes. Okay. We're gonna start sharp though at 2 p.m. Yeah, then we have another. How much longer do we have? We have another like 5 hours, five plus hours of presentations remaining.
That's great.
I know it is an
just a
epic
quick question,
day.
actually.
So,
Would it be possible for to basically get the opportunity to revise a little bit the slides before we present, in the break time? Yeah.
Yes. Why don't we do that? We'll open up the slides now. So if you want to, for those that are presenting in the final part, feel free to do some revisions, and we'll close the deck again about 10 minutes before we start at 2 o'clock Eastern time. Okay. So why don't we open up the slide for those that want to make some edits, and we'll close it again at 1:50 p.m. Eastern time.
Thank you.
Okay. All right, everyone. I'm going to pause the recording, and we'll be back in 1 hour and 15 minutes. Enjoy the break, everyone. Congratulations again on the great talks. We'll see you soon. Bye-bye.
Recording stopped.
Georg, are you still there?
Yes, slides are open now, so please
Okay.
edit.
I'm going to
So-
be the mod. Oh, sorry, the mod for the next session. Should I appear like 10 minutes early, or how do we do this?
Yeah, that sounds good. 10 minutes
Oh,
early is
okay.
good, Dominic. Yep. Great.
So Georg, we did put in the tiny break in between, right?
Yes, from 15:50 to 16, there are unfortunately two listeners could not come, so there's going to be a 10-minute
Okay.
break from 3:50
Okay.
to 4.
Can
you
listen
listen to me?
Then just to be sure, Piers, you put in her slides, right?
Yes.
His.
Yes, I did.
Okay.
But please check. Yeah, the slides are open now. They will be open until the start of the presentation.
Okay.
I copied the slides before.
Will you be there to close it again, or will David have to do it?
David should do it.
Will he do it?
Hello, sorry to interrupt. I just wanted to check if you can hear me.
Yeah, we can hear you.
Thank you. Thanks a lot. Sorry.
No. Okay, if anybody else wants to test anything, now is the time.
Yeah, I'm sorry about changing the slide name. I thought I could change the background.
No worries, no worries.
But thank you for putting everything back.
No, yeah. Okay, cool.
So we inserted one slide for Youssef, and I didn't hear from him.
Right.
Where was it again?
Well, let's leave it for now, and then we just skip it when the time
Leave it?
comes.
Yeah.
But,
Just leave it. Okay, that.
We don't know. Okay, cool.
Okay. So that's it. You're going to sleep a little bit now?
It's 1:46 in the morning. I sleep maybe for 3 hours. I'll be back at 5 a.m. Okay, so please, please give this to me.
Okay.
I'm
So
sorry.
who will host? For the next session, it's.
Didn't Dominica say she was going
Dominica.
to?
Dominica
Yeah,
will be
Dominica.
the master, the driving the slide. But who will be the timer? Adrian, are you going to be the timekeeper? Or maybe there's going to be another.
I think Adrian is not here at the moment.
It's Adrian for the next slot, and then I'm the timer for the last one.
Okay.
Yeah, not the last one, the second to last one.
Yeah, I'll be back for the last one, but Adrian and I, we switched every hour or so, so maybe it's a good idea to just to keep it fresh. Yeah. Cool.
So will David open again after the break, or will he say hello to people, or?
I think Dominica will do that, and we just start at 2 o'clock ED, right? So I think that's the plan. So no welcome.
Okay.
Let's go.
I'm gonna
So what do you think?
I'm gonna leave the slide up that says break on it, and then we'll let Dominica drive when it comes time.
Okay.
No, who's like
Please all mute yourself.
Hello.
Hey, Dominica. Are you you're driving, right? You're muted.
Yes, I'm moderating.
But you're running the slides too, right?
Oh, yeah. I guess I should do that, yeah.
Yeah.
In which case I would need screen sharing.
Correct.
But I would have to have the screen share on my other. Okay.
Good. I'd refresh it too in case somebody added something new.
Mm-hmm. We break off the speakers after 30 minutes.
No,
Or
there's actually no break. We'll actually let me just double check.
yeah, because I thought we wanted, like, they have like a deadline at 3 minutes, and then most of them continue talking.
Oh, you're talking about for during the presentation. Yeah, so
yeah.
I forget who's doing the timekeeping during the session. Do you know?
I am.
Oh, okay.
Oh, Marco. Perfect,
Yeah.
great. Do you have the sound set up, the same sound that that Georg and Adrian were using?
I don't know what sound they were using. I have the names here.
No, for the timer at 3 minutes.
Yeah.
They had a really nice sound. It was like a subtle bell.
Yeah. I mean, I don't know what's the name that of the sound they were using. And let me ask.
Yeah, so Dominica, I think you were following what I was doing. So just,
yeah.
Yeah, ring the bell at 3 and then really interrupt them at 4 and then just stop them at 5. But they can keep going to 5, stop them completely and just move on.
Yeah. Got it.
Yeah, it looks like we've got a couple of missing presentations at like 3:50. Maybe able to take a break, otherwise we're gonna power all the way through.
Oh, George Church just entered. Seems like he will be joining the session.
Hey George, are you there?
Yes, I am.
George, I don't know if you caught any of the first 3.5 hours.
Unfortunately I was booked solid. But you're a hero for doing all this.
Yeah, it was actually a really awesome morning. Actually, George, I'll share with you a link to the slides if you didn't see it already. The slides actually are great because you can see all the content that everybody's been presenting. They've got the complete documentation in the on their pages, but you can see the here, let me just put this into the chat. this is the slide deck that everybody is presenting from. So that's you can get a quick view of all the projects there from the global crew.
Thanks.
But yeah, no problem. It was an epic morning. Yeah, we took a little break. We're about to get started in another couple of minutes. So
Yeah.
your time perfect.
Yeah, I was very disappointed you took a break. I thought you were going to compete with the guy that just did the longest filibuster in history, 25 hours.
Oh yeah.
He
Court,
he couldn't take any breaks.
Yeah. That's right. No bathroom breaks either. Yeah.
Yeah, exactly. No, but apparently what he did was he didn't drink for 24 hours in advance, and then he didn't drink or go to the bathroom during the filibuster as well.
Yeah. I saw that his body fluids strategy for that day.
Yeah.
Yeah, we're a little more merciful here, so we took.
Yeah, I
a
guess
break.
so.
But the next stretch is the next stretch is 5.5 hours straight actually. So there's no breaks for the next
Wow.
stretch. So,
Yeah.
yeah. So it was a really great morning. The global students, yeah, they've been bringing their A game. So it's been really wonderful. And George, I hope you're excited about the Cloud Lab. I realized I hadn't a chance to update you since I visited Ginkgo. I think it was like a week and a half ago. But yeah, they're so excited.
Well, I even
Jason
said that I was
Tom,
excited at the closing
I
of
know.
the class yesterday. Yeah, definitely
I know,
excited.
that's what I'm bringing up. Yeah. So the larger goal, George, each one of those racks, those reconfigurable, those modular units, those are like 100K per because the larger goal, I think we need to set up our own. We'll set up our own cloud lab. So and Ginkgo actually they've got a whole bunch of warehouse space, so we obviously can host it anywhere. So they're happy to host it for us. But in the meantime, we can use Nebula basically for the next year and beyond. And so yeah, Jason, Tom, their whole team, they're stoked to work with us on that. So that's the goal for the summer, is to do some prototyping with their system and see what we can set up for next year.
That's great. Yeah.
David. So the next session is going until 4 p.m. Is that correct? And then I'll switch with Megan.
Yeah, you're gonna switch with Megan, but it's but we don't have a break actually, so we're gonna try to make it as smooth as possible.
Good. Okay.
We did a really good job. I mean, the last session, we were 3.5 hours, but we're only 10 minutes late out of that whole time. So hopefully we'll see what our latency is for this one, but pretty good. I'm gonna actually start the recording.
Recording in progress.
All right, everyone. Welcome back, folks. We are going to get started in 1 minute. So for this next 2 hours, I'm going to be handing over the MC duties to Dominika. So Dominika, thank you again for your leadership and for taking the reins for this next stretch. Qianlu, are you there, Qianlu? Just want to make sure that you are there and ready to present. Oh, I just saw Qianlu here. What happened? Did they disappear? Uh-oh. Okay, oh good, Qianlu, you are here. Are you able to turn on your video? Okay, there you go. Great. All right, Dominika, why don't we get started? You should full screen and we can get the show on the road. Yeah, go for it.
Amazing. Am I full screen? Can everyone see the screen?
You are not full screen yet. You need to hit the slideshow button.
I see.
Yeah, it's the upper right.
Yeah. There we go. Okay.
Just hits. Yeah, there. I think you did presenter view. You need to just hit the slideshow button.
I see. Oh, Sorry. One second. I'm so sorry. This is not full screen, right?
It's not quite. You
Give me a second. Sharing getting started. Sorry for the delay.
Dominique, are you able to do this, or should we?
Yeah. For some reason, I No, here we go. Okay, got it.
You're not sharing your screen right now, though. There's no
Okay.
nothing is being shared.
Okay. Okay. Sorry for the delay.
For anybody that missed the morning, we are now fast forwarding through all the presentations this morning. So if you missed it, you're getting an ultra high throughput right now. This
That's
is all of the
exactly
morning
right.
talks. Yeah, so you can brain scan everything that's popping up right now, because this is what we did for 3.5 hours. So
All
oh,
right,
there you
there
go.
we go. There we go. Sorry for the delay. Qian Liu, the stage is yours. 3 minutes for everyone and 2 minutes of Q&A session as just as a reminder. And there you go.
All right. Thanks everyone. My name is Qian, and I am an alumni of Brown University with background in interaction design and biodesign. And my final project is UV actuated carotenoid biosensors for natural UV protection, with a potential end product being a bioadaptive UV coating. Next, please. So currently, there are actually many UV coating products on the market, such as sunscreens and UV blocking paints. However, a lot of them are toxic and most of them are static, meaning they do not adjust based on UV intensity. So in this project, I aim to provide not just a visual signal of UV or a UV protection, but also a biodegradable alternative to current UV protective coating, which at the same time dynamically responds to UV exposure. So how do we do that? I look into how the nature solved this problem, and it turns out snow algae produce a red pigment called astaxanthin that protects them from harmful UV rays. And what's interesting about this is that as UV light gets stronger, snow algae produce more red pigments, and it gives them a better protection from UV damage. Next, please. So my first aim is to produce UV-resistant red pigment by utilizing a UV-inducible carotenoid biosynthesis pathway in E. coli. And to create this pathway, we have the PEG beta at GenSpace, which include the CRT E, B, I, and Y, as well as a PEG28 promoter. So the second plasmid with CRT Z and W is what I ordered from Twist. And while waiting for the Twist order, here are the steps I plan to work on once it arrives. So firstly, transform the plasmid into E. coli and pick single colonies and grow overnight in LB with antibiotics can at 37 Celsius. And then dilute and grow the culture at IPTG for induction and then incubate. And lastly, use the same culture for pigment extraction and UV resistance testing. Next, please. Next, please. And based on the last two steps and the data I expect to collect, this is a chart I anticipate to see, which supports the hypothesis that the more red pigment is produced, the greater the UV resistance. And then after this, we could start working on the second aim, which is develop UV responsive biosensor. And then this would require the change of promoter from PET28 to REC-A for UV inducible. And moving on to M3 with potential applications, I envision the biosensor being integrated into a UV responsive coating, which could potentially be applied to things like car windows or sunglasses. And it also has a potential to function as a UV sensitive canvas for artistic expression. And that said, this also comes with challenges. A lot of those things need to be taken into considerations, such as stability, durability, scaling up production, regulations, and also the sustainability of other additives and the overall sustainability. So my next step is to keep these challenges in mind while working on the UV resistance experiment. Thank you.
Amazing. Great work. Are there any questions like right off the bat? Because, okay, in that case, I have a question. So what is the approximate wavelength that those pigments can block?
Right now, my goal is
Range.
to have it between the 280 to 40, which is the UVB and UVA.
Are there any more questions?
Yeah, Ben's got a question. Ben, go ahead.
Great.
Yeah.
Yeah.
Thanks so much. Great project, Jen. I just have one question for you. I want to know if you have any information about the shadowing effect that components of cells can make for UV protection. Because when you're trying to use it as a shield or something like that, just the cells or just the components, they will make like a barrier itself. So have you checked something like that, perhaps? Thanks so much.
Sorry, could you repeat a little bit?
If you don't mind, let's do the question and answer in the chat. We're already 30 seconds over. But Chen, great job. Thank you so much.
That's great. Okay. Then for the next presenter, Syed Tawhid Ahmad. Did I pronounce it right? I'm sorry if not.
Close enough.
Okay, great. Okay, stage is yours.
All right, thank you so much. Assalamu alaikum, everyone. My name is Tawhid. I'm a research assistant here at the Precision Medicine Lab in Pakistan. Next slide, please. My idea is a bit more doom and gloom from what we've seen today. And it's basically to engineer a kind of yeast that can be used to produce food in a survival scenario. So the yeast would be engineered with an endoglucanase enzyme, which would convert plant waste, which is cellulose, into glucose, sugar that we can eat, and also ergosterol, which is already a product present inside yeast with a certain wavelength of UV light can be converted into vitamin D2, which obviously vitamin D, if people are living inside bunkers, is going to be one of the biggest kind of micronutrients that takes a hit. The inspiration actually comes from this really good book by David Deckenberger. It's called How to Feed Everyone No Matter What. And he kind of talks about these survival scenarios and, the different kinds of things that we can do. And one of the main kind of takeaway points is, if we're just prepared like a little bit, if we just have plans in place, billions of people could be saved. And that's kind of the theme of what I've been trying to go with here. Next slide, please. So my aim number one is just to kind of design a plasmid that can produce glucose from cellulose and also kind of, put it into a yeast where then exposed to UV light, it can then convert ergosterol into vitamin D2. So produce those two products. The aim two would then to kind of make this into like a product that can actually be stored long-term, lyophilized maybe, and can kind of be like a ready starter culture kind of situation where it can be used in that kind of situation. And aim three would be, so vitamin D check, glucose check, but Ideally, I wanted to also be able to produce other micronutrients that humans need to survive. So, if it's like a longer term bad scenario outside, maybe people can survive using this longer term, especially on cellulose, which is plant waste. So if it's a catastrophe scenario, that's probably going to be abundant anyways. The construct design I'm still kind of working on, but I especially want it to be, the DNA needs to be kind of UV resistant, right? So maybe lots of redundancy, or that's kind of what I'm trying to kind of put into place. Next slide, please. The ethics of this are complicated because it needs to be consumed by humans, so it needs to be super duper safe. And another big thing is not everybody understands English. So if this is like a no communication kind of situation, it needs to be, I really wanted to communicate in this package, which, so the script for English is left to right. The script for my native language, Urdu, is right to left. So I kind of went top down and symbols, so anybody and everybody can kind of understand how to use this. And on the left, actually, I have, when I've tried my best to, because it needs to be informed consent that they're going to be consuming a GMO. So I really wanted to kind of communicate that. I don't know if I've done a really good job, but I've been thinking a lot about how we can communicate that something is a GMO without the use of, language or English specifically. So yeah, that's kind of what I've been trying to do. And I just kind of also wanted to, this How to Grow journey has been kind of a wild ride. So I just kind of wanted to thank everyone at the end, especially David, George, Annie, and my my mentor, Austin. So yeah, I would love some questions.
Amazing work. I love the product design and the thought that went into it. Does anyone have questions? Yes, applause to you.
Yes, I have a question actually.
Yeah, go for it.
Yeah, please.
Cool project said. I was wondering if what the mechanism behind the project would be, like how would it work? How would the production of the food be triggered in a way?
So yeah, I think it kind of the way I've kind of imagined it is just kind of a suspension, and then you can then kind of maybe purify the yeast through a filter, which would then separate possibly the vitamin D and possibly the glucose from the cell bits, something like that. Or I mean, yeast is a consumable, so you could just eat the yeast or put it in some bread or bake it with something. So that's kind of what I was thinking.
Okay, cool. Do you have any idea of of the visuals of how that could look like, or how, I guess, or what the emotional side of it would be if you have to sustain yourself on this kind of like powder or something for a long time, or if there's something like, because humans need certain things, maybe it's weird to consume just like powder or something. I think that could be
100%. Food is very strongly linked to morale, but I guess in a survival scenario, anything is better than nothing, I guess. Yeah.
Okay, unfortunately, we will have to cut off here, but thank you so much for the great questions and the great presentation. We are going to move on. Any other questions can be asked in the chat. We're going to move on to Elvio Rodriguez. Are you here?
Yeah, I'm here.
All right. Okay, great. You're being, can someone highlight him? Okay, perfect. All right, the stage is yours. Go for it.
Perfect. Hello, everyone. My work is entitled Protein Complex Disruptors to Eradicate Chagas Disease. Next slide, please. So, Trypanosoma cruzi is this parasite on the left that is the causative of Chagas disease, which affects mainly the heart, and people often die from heart failure. There are around 7 million people in the America infected, and it's transmitted by that insect vector on the left. And during my PhD work, I found this essential epigenetic complex, which has these two proteins, the BDF5 and BDF5 binding protein in pink. And when I mutated the orange protein to interrupt this interaction between these two proteins, and I introduced it into the parasites, they will basically stop dividing, and there were a lot of nuclear defects in the lineages. So the idea is to take a small motif, this protein, this pink protein hat, scaffold many mini proteins to generate something like an inhibitor that can be used to inhibit the assembly of this complex. Next slide, please. So I designed these twist vectors to test the system if these mini proteins actually interact with the target by fusing the N-terminal half of GFP to the target and the C-terminal half of GFP to the mini proteins in order to, if these proteins interact, they will reassemble GFP and the bacteria that express these proteins will flow. Yes, to test this in an automatic manner using OpenTrons to mix the plasmids, perform the transformation, induction, and then take a microplate and measure it in a microplate reader. So the fluorescence will be directly proportional to the interaction strength. And the next slide, please. So as in 2, I want to be sure that these designs work. And if they do not work by themselves, I can, for example, fuse these designs to a delivery domain that targets some endocytic pathway or something. And for example, also fusing it to a nuclear localization signal to enrich it in the target compartment, which is the nucleus. And of course, I will be testing these designs in the parasites in vitro and with animal models to see if they cure the disease and if they kill the parasite. And as a long-term goal, I want to eradicate this illness. For example, by generating an immunity based on these mini proteins. For example, modifying a small amount of liver cells to express these disruptors and secrete it on the bloodstream. So when the parasite enters the bloodstream, they will be killed, hopefully. Another approach could be to modify genetically the insect vectors to be resistant to T. cruzi. For example, fusing the disruptor to an intestinal protein. So when the insect vector feeds on uninfected people, it will kill basically the parasite when they reach the gut. And of course, using gene drives to spread this trait in the wild population of these. And that's my work. I hope you liked it.
Okay, amazing work. We have 1 minute left for questions. Does anyone have an immediate question right now? Please speak up. In that case, did you have the opportunity yet to establish any of these in the wet lab yet? Do you have access to a lab?
Yeah, I'm working with the parasites in vitro and on some animal models infecting rats and mice and sometimes rabbits.
Okay. Oh, okay. So you have plenty of access. That's great.
I already asked for the twist orders. I hope they will be coming soon.
Yes. Amazing. Okay. Any other questions in the chat, perhaps? In that case, let's move on. The next presenter is Paula Rojas from Life Labs. Are you here?
Yeah. Hi, how are you? Can you hear me?
I'm great. How are you? Yes,
Good.
the stage is yours. Go for it.
Okay. So I'm Paula. I'm a designer and artist from Bogota, Colombia, exploring biodesign. So you can go to the next slide, please. My project is a low-cost lacase-based water decontamination system for a decentralized water treatment in rural and urban settings designed to empower communities and also support water sustainability and public health. In Colombia, the agricultural industry is responsible for over half of the water usage in the country, and over 1 million people lack access to clean water, mostly in rural areas. Rainwater and wastewater reuse is a sustainable solution, but only if treated properly, because contaminants such as endocrine disrupting compounds and pesticides can be commonly found in water that has not been treated correctly, and this can cause effects on humans and the environment. This project aims to make the reuse of rainwater safe in rural areas by using the lacase enzyme to break down contaminants as it does in nature, in a water decontamination system that is affordable, modular, and portable, and capable of floating on the water by passively cleaning it while also detecting pollutants. Next slide, please. As I told you before, laccase acts as a natural solution to degrade harmful compounds. It does it by oxidizing the pollutants and breaking down their structure through electron transfer, allowing the formation of less harmful products. So to achieve this project, it's essential to express and purify the laccase enzyme using E. coli as a host. I plan to do this at LifeUp this summer using a plasmid I designed and ordered from Twist. And this plasmid contains the laccase gene from a white rot fungi along with GFP to confirm the enzyme is being properly expressed in E. coli. It also has a histag to make the protein easier to purify and a thrombin site so that the histag can be removed afterward. And this is important because the laccase will later be immobilized for testing in water. So this will help to avoid the risk of metal ion leakage into the water. Next slide, please. So the continuation of aim 1 is to immobilize the laccase protein in calcium alginate beads to validate the enzymatic activity with BPA and common pesticides as atrazine. And the aim 2 is to test the GFP and the laccase protein for the biosensing component that can actually detect pollutants and say when the water is safe to use. Also, it's important to begin experimenting with mycelium-based materials for the floating structure, because the goal is to maintain a concept of circularity, enabling communities to grow the structure themselves and compost it after use. The broader objective is to transform this technology into a scalable, low-cost, modular water decontamination system, biomimetically inspired by fungi, and it will float on the water in tanks or ponds that are contaminated with BPA or pesticides, enabling passive and decentralized water treatment that empowers rural communities. So thank you very much, and I'm happy to answer any question.
Great work, Paula. Are there any questions? Yeah, David, go for it.
Yeah, great job. I really enjoyed your presentation. Really well done. So for bioremediation, one thing, one question I have is just on kind of the efficiency of the remediation. If you can comment on that, and then also, you showed it a little bit in your aim 3, but ultimately, what's the format, right? Like how, if the goal is, doing this at scale, yeah, like do you imagine this happening in plants that are nearby the water that kind of gets treated over there, or doing it kind of on demand at the location? So just those two questions.
Yeah. So the first question, the ability to bioremediate will depend of how successful the immobilization of the enzyme is, and also it's important to measure how long does this immobilization will last, because you will need like to replace them, so the alginate beads. So it's something I have to research a little bit more. And the second question, I wanted to be really decentralized. So I wanted to be like in each, I don't know, like house or agricultural farm, maybe in the same tanks where they sometimes capture rainwater or have like maybe the wastewater of the agricultural works. So yeah, but I want it to be totally decentralized, so more focused on each place. So it has to be something that's modular, but also portable. That's why I was planning like this structure could be from mycelium that they could grow themselves, and then the alginate beads could be something that can be integrated on this structure.
Great, thank you. Let's move on. So thank you so much, Paula.
Yes, let's move on. The next one is going to be Kurosh.
I'm here.
Okay, oh, great.
Hi.
All right. Yeah, from Life Labs. Yeah, we are looking forward to your
Can I begin?
presentation. Go for it. Yeah.
Okay. So hi everyone, I'm Kurosh, and the topic of my work is about simulating the diabetic wound microbiome and tracking its healing in silico. So next slide, please. Next slide, please. Oh yeah, thank you very much. So diabetic foot ulcers are one of the most common types of diabetic wounds. And as the name suggests, they are the direct result of diabetes, which is characterized by high blood glucose levels. So the interventions right now for diabetic foot ulcers are quite ineffective and at times expensive. And there has been this impetus to move on to probiotic alternatives and probiotic solutions. But I noticed that these probiotic solutions do require some degree of personalization in order to be maximally efficient. So that's why I structured my AIM-1 around constructing and implementing an in silico pipeline for validating and benchmarking different probiotic designs. So if you could move to the next slide, please. So yes, as you can see, my AIM-1 consists of 6 different stages. So far, I've implemented 4 of them. It all begins with quality control and model curation. This part is basically dealing with individual models. Then we'll get into the community scale by performing pairwise co-cultures of different probiotic and pathogenic metabolic models. These are all computational models that we are using. And then we would be using, let's say, strain design package to introduce entered DDROF or the ED pathway as a heterologous pathway to some of our lactic acid bacteria. And then we would be constructing the host model, which is responsible for giving out physiological responses and predictions. And then a final simulation would be, let's say, an assembled version of all of these models. So if you could please move to the next slide. Thank you very much. For now, I do not have most of my results. I only have my co-culture results, as you can see, the one at the top. The rest are the things that I anticipate to see from my strain design or from my final simulation. But generally speaking, as a part of my AIM-2, I aspire to enhance the scope and the accuracy of the model by integrating it with kinetic, let's say, data, or perhaps experimentally validate its parameters. As you can see, I've included a picture of my benchling. I'm going to choose one of those PSIP plasmids in order to benchmark experimentally benchmark my model. And the overarching goal of the whole project would be to deliver that integrated clinical solution to clinicians around the world to formulate effective therapeutic solutions for their diabetic patients, and possibly even integrating it with multi-omic data or using graphical user interfaces to do that. Or perhaps even creating, let's say, wound dressing solutions using novel polymers like bacteriocellulose polymers. So yes, that is the overarching goal of my project. Thank you very much.
Thank you so much, Kourosh. Are there any questions?
Yeah, I'm open to questions. Sorry, I had to wrap it up really quickly.
Oh, no. It's all good. It's all good.
But I would really like to hear more feedback on my project.
Yeah. Do you have like first plans for the experiments that you want to do, like wet lab experiments? And do you have access to actual clinical data for your model?
Unfortunately, I do not have. I haven't checked out whether I could actually access any clinical data here in Iran. I don't know. I have some friends, but I'm not sure whether they could grant access to some real-world clinical data. But the real concern would be really how the strains that I'm designing here as a part of my in silico strain design really close to what they would act or behave in a real-world culture medium. Okay, so the strains are my first concern. And of course, the host model has a lot of work to be done for. So yeah, that is also part of my concerns, how to scale up the host model. Currently, as you can see in my second slide, yeah, it only has one of the pathways integrated.
Yeah, okay. I will unfortunately have to cut you off here. Great response. If you have any more questions, then please address them in the chat. And we are going to move on to the next. Great presentation, Kourosh, again. Adelina Krusteva.
Yep, I'm here.
You're here. Great. Okay.
Can we move to the next slide?
Of course. There you go.
Thank you. So
I want to present my problem. My problem is actually my problem in my life. I'm diabetic, so just a little of a background how diabetes works. Can you see these beta cells? They are beta cells from our immune system. They produce autoantibodies against beta cells in our pancreas that produce insulin. And these proteins against our beta cells in the pancreas are like insulin 2, beta chain, or GAD65. So these autoantibodies aren't directly cytotoxic, but they help activate CD4 and CD8 autoreactive T cells, and then they destroy our pancreas. My project proposes a dual solution for this. So I would like to create an immune modulation with polylactic glycolic acid nanoparticles, which will encapsulate these autoantigens like insulin, beta chain, and GAD65 in order to reteach our immune system how to act towards our own body. And then at the same time, beta cell regeneration, which means that I'm going to send some of the beta cell proliferation genes into alpha cells, which are also part of our pancreas, and make our alpha cells become beta cells. Can we move to the next slide? Thank you. So the first step of this would be, I haven't done anything yet, is to encapsulate this beta chain autoantibody that our beta cell immune system does, in order to encapsulate it then into dendritic cells and present it to our immune system and be like, hey, this is a beta chain, nothing is happening to our organism, please don't kill beta cells, like pancreatic beta cells. However, I could not order the twist gene for beta chain. That's why I had to optimize my protocol and create it with the whole insulin gene, not just the beta chain. So what am I going to do about this is first dissolve PLGA in dichlorometane to form the organic phase. Then I would add my DNA solution and use a peroxonicator to make like a primary water oil emulsion. Then I'll add some 2% PVA to and then tipsonicate again to form the water oil water double emulsion. After that, I would stir it for 3 hours at room temperature in order for the solvent to evaporate and create and collect the nanoparticles in a centrifuge while washing it with PBS. So now after I create these nanoparticles with my DNA inside, I would characterize them with DLS to see the size, which would be around 50 to 100 nanomolar. I would see if there is DNA with nanodrop, and I would see if they're negatively charged for uptake with dendritic cells. Then I will use this protocol to actually encapsulate beta chain with overhanging primers that I'm going to create, and for the other co-delivery immunomodulators that I want to encapsulate in the PLGA nanoparticles. Can you move to the next slide? My second aim is the LNP-based alpha cell reprogramming. So this would be, as we can see here on the left slide, these are some mice which were made diabetic. They were injected with viral vectors with MAFA and PDX1, which were genes that make alpha cells go to beta cells, and this was efficient. So I decided to use the same thing, but with LNP particles, which are lipid-based, and they won't be injected directly into our pancreas, but they're a little bit more modulative, so we can modulate how long they last, where like we don't have to we can take them orally, we can take them as injection, and they don't have, because we can't really inject anything into our pancreas as people. And my third aim is to actually put these two things together and create an actual cure for diabetes without need for endogenous insulin anymore, and without our immune system rejecting any kind of transplants like beta cell transplants or Langerhans Islands transplants, which are like an agglomeration of like beta cells. Thank you. Sorry, it was very quick. Sorry.
No, amazing. Great. So we have only 30 seconds left. Is there like a quick question? Otherwise, just refer to the chat and please ask those. Please look out for these in the chat, and we will move on to the next one. Order Sacco. I don't know if I spelled it right. I'm so sorry.
No, that's right. Order Sacco, that's right.
Perfect. Live Fabs. Go for it.
Stage
Thank
yours.
you. Hi, everyone. I'm Auda Sacco, and I'm a educator in sustainable fashion and also a multidisciplinary artist here in London. And my project is Cellum Plus, We Are the Future. And can you move on to the next slide, please? Thank you. So really the problem that I'm addressing here is that over 60% of women's gymwear is made from polyester and elastan, petroleum-based material that traps sweat, irritate the skin as well, and shed harmful microplastic into the waterways. This fabric weren't designed really with women's wealth in mind or ecological repair in mind as well. And so the question that I'm asking here is, what if our clothes could actually do more than just cover us? What if they could actually actively support skin health, reduce waste, and regenerate the environment as well? So I've got 3 clear aims here, which one is to replace the fossil fuel-based gymwear with a biodegradable, regenerative alternative. My second aim here is to harness spirulina's natural bioactivities through hydrogel-based textile integration. And the third aim here is to develop a layered biomaterial system that is modular, breathable, and stretchable without plastic. So why spirulina? Well, spirulina is a microalgae that's vitamin-rich, antioxidant loaded, and naturally anti-inflammatory. It's already used in wellness supplements, and research shows that its compounds can benefit the skin barrier as well. But beyond health, really, spirulina is regenerative by nature, and it really does require minimal land or water to grow and actively captures carbon as well as it's photosynthesizes it. So using synthetic biology principle, I've developed a 2% alginate hydrogel infused with spirulina biomass, glycerol and pomegranate extract. And then a calcium chloride mist cross-links the gel, stabilizing it without harming spirulina viability. So this gel will be bioprinted onto the fabric in modular patterns, capsule, grids, or lattice, to allow the controlled bioactive release. Can you move on to the slide 3, please? So the way the layered system works and what it does include, so there is a first layer, which is a bamboo lyocell jersey for skin comfort and breathability. Then the second layer is a natural latex alginate stretched layer that avoids elastan. And then the third one is a spirulina hydrogel top layer designed to respond to moisture and deliver biobenefit during wear. So far, I've developed a stable skin-safe hydrogel formulation, demonstrated moisture reactivation with mist and stimulated sweat, validated compatibility between hydrogel and textile substrate, and build a lab protocol for spirulina hydrogel biofabrication at LAFLab. So really the next step for me will be to begin bioprinting these hydrogel patterns, conduct a fluorescence microscopy on spirulina viability, test the mechanical stretch and wearability across the layers, and then eventually launch an early garment prototype for pilot testing, really. And if time does allow me still, I did actually conducted a research survey. So it was a community-led data collection on microplastic impact in gemwear, and it was really positive. And I'm still working on the protocol development as well, lab-tested spirulina hydrogel workflows and prototyping, as I mentioned earlier. So then we can have the textile and hydrogel integration on the way. So that's really that for me. Thank you so much to LAFLab, really essentially Digby as well, and also to MIT. It's an amazing opportunity and I'm so grateful for it. Thank you.
Great, thanks so much, Auda. That was a fantastic presentation. Are there any questions
Yes,
in the
I
room? Yeah, go for it.
Thank you so much for the presentation. It was really nice. What would you say was the biggest challenge or is the biggest challenge in making this come to life?
Yeah, I think for me, the biggest challenge at the moment is at this stage, because we're not, well, I really want to, we're working with wild type spirulina platensis. So for me, I think the biggest challenge at the moment is to really come across the bioactivity of that spirulina and and really, apply that synthetic biology, so then we can actually, enhance the therapeutic trait of that project really, and really increasing the oxidant content, so then we can turn into the responsiveness of this pH or the temperature. So I think that is the biggest challenge at the moment.
Thank you so
All
much.
right, we will have to interrupt here and go to the next one. Great work. Oh, I'm so sorry. I'm afraid I didn't show the last slide. Here it is for everyone to see quickly. Alejandro Menas Castellanos, are you here?
Yes. Well,
Amazing. Yeah, live fabs again, so go for it.
okay. Well, hi everyone. I'm Alejandro, and I'm an undergrad student of biochemistry in Spain. My project focuses on engineering plants to detect and reduce mercury contaminations from the environment, which is a serious issue due to its toxicity. Mercury binds into the DNA and causes mutations. So these mercury come from mining, the water pollution, and so on. So what I want to do is to turn plants into sensors and cleaners of these pollution environments by editing their genome and turn them into super resistant plants. Could you pass this slide? Okay. To achieve this, I'm going for my first aim, that is using the MER operon, which naturally provides bacteria with mercury resistance. I'm especially using a MER-A enzyme, which reduces the toxic mercury ions into a safer volatile form. I'm also using a MER-R, which is a repressor, so when there's no mercury in it, it will not express my constructions. And also, obviously, when we work on plants, we use a GFP as a visual marker. These genes will be inserted inside a plasmid for plant expressions, and I modeled a version of the plasmid by gypsum assembly in benching. Could you pass the slide? Okay. To validate this, I'm going to, for my second aim, that is to actually see if these mercury ions have been reduced. I want to know if it's reducing and also how much this work has efficient on repairing the damage that mercury are in soil. So to do this, I'm using an enzymatic assay based on MER-A activity. Since MER-A using a cofactor, I could track the dependence of the cofactor by monitoring the absorbance of this cofactor. A faster drop in absorbance means more enzymatic activity and so on, so I can see how much mercury is the plant reducing and cleaning. And then a long-term idea, it's for my third aim, is to test this outside the laboratory. Since I don't know how these transgenic plants are going to develop in real contaminated soil. And I want to know if these plants will survive outside the lab, and also if they do, which impact they will have in the wild. And that's it.
Wow, that was really, really great in time. Are there any questions? Okay, then I have one. With regards to the expression of your construct, do you want to express it ubiquitously in a plant, or do you want to perhaps have it like expressed like in the roots in particular? Like, have you made any thoughts about that?
Well, at first I thought to only express it on roots, but since mercury can also be absorbed on air by the leaves, I want to have the whole plant resistance. So it can't like pollute air and soil and also water.
Okay, great. Are there any other questions? Yeah, Adrian, go for it.
Yeah, I was curious about this. So once the mercury is going to move to the plants, how are you going to dispose of the plants, or how are you going to deal with that part?
Like how the mercury ions are going to get inside the roots, you say? Well,
more what's going to happen to the plants after they collect this mercury.
Well,
because if you leave the plants in there, they're going to repollute the environment, right?
yeah, well, once they are exposed to the mercury are gonna enter the root plants by other ions receptors, and when it's reduced, and it's bound to the environment, this mercury can bind into solid particles that are naturally found on the wild. Mercury it's not usually oxidized like that in these polluted environments. I don't know if I'm answering your question.
Yeah, I think that's good. Thank you very much.
Okay, any remaining questions, address them in the chat. And thank you so much, Alejandro. That was a great presentation. We will now move on to the next one. Amy Chen, GenSpace, are you here?
Yes, hi.
Amy, perfect. Go.
Okay. Sorry. Hi, I'm Amy, and my project is about using the biosynthesis of bioindigo for copper detection with the end goal of turning it into a living responsive installation. Can you go to the next slide? My background is in architecture and materials research, so I'm fascinated by living systems responsive to the environment. I was inspired by Sense Biome 2, a color shifting sculpture by Dumo Lab and crafting plastics. And I wanted to create a piece of art that changes color in response to environmental pollution, specifically copper, which is safe to work with at GenSpace, a BSL-1 lab, sorry. Instead of working with traditional or synthetic indigo, I'm using bioindigo, an indigo blue pigment produced by engineered bacteria. It's more sustainable, has replicable protocol, and is living. My goals were to test how to optimize bioindigo production in the lab by testing out different concentrations of L-tryptophan, as well as different lighting conditions, to set the foundation for a copper detecting biosensor by doing a transformation, and then fabricate a copper sensitive sculpture that would show up in varying shades of bioindigo based on copper concentrations. Can you go to the next slide? So while waiting for our Twist Biosciences order, I tested how growth conditions impact bioindigo production using our existing E. coli strain that produces bioindigo. I experimented with 2 L-tryptophan levels, and the doubled amount worked the best, and then different lighting conditions in darkness at the incubator light, using the incubator's regular light, and as well as green lighting in the 37 incubator, and culturing it in complete darkness was the best. And the spectrophotometer results confirmed that the wavelengths in those were the true indigo at 420 nanometers. And then can you go to the next slide? And then my next steps, once the plasmid arrives, I'd like to transform the E. coli top 10 with a construct that includes a CUSC promoter, which activates in the presence of copper, the TRXA BFMO biosynthetic pathway for indigo production, all cloned into a PET21A vector, which is optimized for expression. And the goal would be to make color intensity directly proportional to copper concentration, essentially a visible biosensor where richer shades of bioindigo means more copper. And this could be tested with a simple assay to detect copper levels just by looking at color intensity. And then beyond that, this would become art, starting with a bioindigo dyed embroidery for a small scale piece, and then a hand-woven tapestry with indigo and bioindigo dyed yarn, bringing together craft material science and sustainability into one piece. And thank you so much for everyone for coming. Thank you.
Thank you for this amazing presentation. Does anyone have questions? David, go for it.
Yeah, so I think for the different biomaterials projects, I always wonder about how you intend to incorporate the biomolecules or the organisms into the textiles. Do you need them to be living as a part of the textile and so on? I think one of those interfaces challenges, I think, are some of the bigger ones when we want to think about textiles. So curious your thoughts on that.
Yeah, I was also trying to figure this out myself and thinking about how maybe Dumo Lab did it, but right now my 2 thoughts are to maybe I don't know if it would work. I mean, it has to be living. and it has to be embedded into the textile or whatever material. Because for Duma Labs, they used a PLA, and so maybe there are other materials other than textile that can be used that you can incorporate that biomolecule into, and that way it's still living while it's exposed to the environment. And then I'm also thinking about, like, would it be in soil, or would it be in water, or would it be in the air? Because theirs isn't the air, but maybe mine, because it's for copper detection, might be in soil. Yeah.
Great. Thank you
All
so much.
right. Amazing. So now we will
Thank
move
you.
on to Nina Kurowska, BioClub Tokyo. The stage is yours.
Thank you. So hi, everyone. My name is Nina. I am currently studying synthetic biology in Germany. And my project is about designing microbial fuel cell household devices for energy generation and water treatment. Can we move to the next slide, please? Yes, so as you all know, our household depends on electricity, which is predominantly coming from fossil fuels as of now. And despite advances in technologies, we are still not able to replace it fully with bio-based solutions. Microbial fuel cells offer us a dual benefit of both wastewater treatment and also power generation. So the main problem with them is their limited scalability and also low power outputs. And this stems from also the lack of a very robust chassis. One option is Bacillus subtilis, which can thrive in wastewater, but has problems with generating high power densities. The other one is Shewanella oneidensis, which can achieve that, but needs to feed on specialized substrates. So my aims are to develop chassis that can both thrive in wastewater and provide efficient power generation, create a prototype that is 3D printed, cost-effective, and can allow for testing this chassis. And the third one would be to scale it up. And let's go to the next slide. Yes, so my plan for this was to introduce a cellulose degrading gene into Shewanella oneidensis. That is because 1/3 of organic matter in municipal water is cellulose, which comes from toilet papers and other paper products. Another idea is also to introduce heavy metal resistance genes in order for this organism to thrive better in wastewater. To introduce the cellulose genes, I wanted to use the Integrate platform, which both uses the Cas technology and also transposase enzymes in order to incorporate the system into the chromosome for long-term expression. Let's go to the next slide. So my results that I obtained in silico, because I didn't have access to the lab for this, was that I first identified the sites on the genome of Shewanella where it was safe, because there were not coding to introduce the gene into. I then designed the crRNA for this reason. Then I encountered the problem because the backbones for this chassis weren't available as sequences, so it was very hard to find something as a backbone. So I used a novel synthetic biology approach that I found in the paper when they assembled modular plasmids from different components. So I used that to create a plasmid completely from scratch and then incorporate the gene of interest into that using the Integrate system. And I did that basically with Gibson assembly, but yeah, that was in silico playground, let's say. So future outlook would be to test this in a microbial fuel cell. You can see it here, basically how it would look like most likely. And here is an AI hallucination of how I would see the device looking in real life and be usable. So thank you very much.
Amazing. Thank you so much for this brilliant presentation. I love your cloning strategy. Does anyone have questions?
Yes, I do.
Yes. Yeah,
okay.
Go first.
Okay. Really cool presentation, a really cool project. Do you imagine this wastewater treatment system to be installed or be installable in single homes or more like in a bigger community?
So my idea was to do it decentralized, just like Paula said a couple of presentations earlier with her wastewater system. I wanted it to be completely decentralized, to be in households around the world. The problems here are vast. You can imagine with how are we going to test it? What is the emergency system for when this stops working, when people are cut off from water? So there are many bioethical considerations coming into this, but yeah, I would really love it to be in every house that wants to be self-sufficient.
Cool, thank you so
Great.
much.
There was another question.
Yeah, thank you very much
Go
for the
for
amazing presentation. And I used to work in microbial fuel cells as well. So I was just wondering where you have considered the opportunity of utilizing some chemical charge shuttles, like sodium acetate, in order to basically increase the transfer of electrons between the anode and the surface of the bacteria, basically the engineering. So usually when you're adding some, chemicals, it's easier to basically transfer the electrons as you basically artificially enhance the surface of contact.
In the coating of the anode in this case. To be honest, I'm not an engineer, so it is hard for me to answer. From my research, I think that cathodes were platinum coated, but that is not where the microbes are. They're around the anode, and the anode, from what I saw, the cheapest one for the 3D printing model of the device would be from carbon, but I don't know about coatings, to be honest, but great point to look into.
Okay. Sorry, I have to interrupt. Soham Patnaik, you're up next. Thank you so much, Nina, again. Address further questions in the chat. And Soham, are you here?
Yes, I'm here.
Okay, go for it.
Great. Hi, everyone. My name's Soham, and for my project, I focused on the future of food and how we can produce animal proteins we eat outside of an animal's body. This is similar to Spruce's project, which was about cultivated meat, but more specifically, this focuses on producing these proteins with yeast. So here I'll be producing ovalbumin, the primary protein in egg whites, with yeast, which in this case is Pichia pasteuris. Yeah, I guess in this slide, the formatting is a little different than I remember, but to start, let's go over why we should do this. While this project focuses on egg whites, this technology, in conjunction with the other pillars of alternative proteins, is meant to address the problems with industrial animal agriculture. Today, animal ag has a huge impact on climate change, public health, and animal welfare. For climate change, it contributes nearly 15% of greenhouse gas emissions and uses 80% of agricultural land, which is over a third of the world's arable land. For public health, 70% of the world's antibiotics are consumed by these animals, increasing the risk of antibiotic resistance. And for animal welfare, every year we kill over 80 billion land animals, 7 billion being from the egg industry, and over 1 trillion aquatic animals. Clearly, this causes immense suffering, and we have the opportunity to drastically improve this. With this project, I want to further research in the space by community biolabs by producing egg whites with yeast at GenSpace as a proof of concept, and I'll be sharing this protocol so others can too. Next slide, please. So how do we do this? This slide shows the exact steps using methods we learned in this class, but a rough overview is that we, 1, design and order the ovalbumin gene from Twist. The picture on the left is the plasmid map from Benchling with the gene insert. 2, we'll design and construct the plasmid. This involves digesting and purifying the plasmid, performing ligation, and then transforming E. coli to grow it. 3, we'll engineer and express the ovalbumin gene in Pichia pasteuris. This involves preparing the transformed plasmid and electrocompetent Pichia cells, transforming the Pichia, and then inducing ovalbumin expression with methanol. And lastly, we'll analyze and purify the protein. Since the plasmid has an alpha factor secretion signal, the protein will be secreted into the supernatant, making the step a lot easier. And this step includes quantifying the protein with a BCA assay, running an SDS page, confirming the ovalbumin with a mass spec, and purifying the protein using the HISTech. Next slide, please. So for this project, where are we now and where are we going? In the short term, I'm working on the proof of concept. So after we get our twist order, I'll produce and analyze the ovalbumin at GenSpace and document it on Notion as I go. In the medium term, the goal is to develop and share an open end-to-end protocol so others can build on this work. And in the long term, I'm hoping to increase decentralized research and production of animal proteins with microorganisms. Thank you.
Right on time. Yeah, I really like that you emphasized the importance of open science for your project. Does anyone have questions? If not, then I have a question. How exactly did you pick the promoters and the regulatory elements for your plasmids? Is it a plasmid that you found somewhere else, or how do you decide
Yeah.
on the expression levels?
Yeah, so I wanted to use materials that are commonly found in community biolabs that everyone should easily have access to. So I kind of just went with what we had at GenSpace already, which we're using for other projects as well. In this case, the yeast was Pichia pastoris, the plasmid was a PPIC 9K plasmid, so it already had the promoters and the alpha factor secretion signal. So I just wanted to like use readily available, commonly used materials.
Okay, great. Are there any other questions? Because we are a little bit behind, so I would probably move along. If you have any other questions, please address them in the chat. Next one is going to be David Castillo from Ottawa Bioscience. Are you here, David?
Oh yeah, hi, can you hear me?
Perfect, yes, go for it.
Okay, thank you so much. Well, my project is related to the production of antimicrobial plastics in a way to cope with many nosocomial infections that normally invade in many of these type of medical devices. Can we go to the first slide, please? So the problem, the majority of the nosocomial infections are due to the resistance that is created, not just by the abuse and constant exposure to this, whatever treatment on these bacteria, but also on the spaces, on the surface that are forming some biofilm. So the idea is creating a self-cleaning antimicrobial bioplastic. So for that, we are focused on the production of PHB, which PHB has been around for several years. Nevertheless, it has been expensive to produce them in a non-microbial setup. The majority of the PHB has been extensively studied in models like, for example, Ralstonia solarium, which is one of the images that I'm presenting on the panel below. So we're intending to jump the scientists into a different model, which is in a unicellular algae, which is a Reinhardti. We're using as a model for producing them. Chlamydia monocyte Reinhardti has also been observed to produce some antimicrobial peptides, which are called O-defensins. So we have already a couple of strains that are producing those proteins, and we're trying to merge them, like synthesizing one of the roots that we are trying to modify it for the production of PHBs, and also combine them with the expression of this O-defensin. Could we go to the next slide, please? So here is more or less what is the strategy is linking two metabolic pathways. One is for the sustainable production. Why the sustainable production? PHBs are at this moment, even for an industry level discussion, they are extremely expensive for scaling up. And one alternative for doing that, for producing them at large batches, is introducing the same operon into Chlamydia monocyte Reinhardti. So since Chlamydia monocyte already have many of the metabolic pathways that are combined for the production of the precursors of these bioplastic, which essentially are similar to polyesters, they're also biodegradable and they have thermal capabilities, like they can be 3D printed with the current technologies. So we are intended of cloning these full operon. Could we go to the next slide, please? Could we go to the next slide? Yeah, thank you. And this is the summarized work that we have done so far, which is already have a plasmid that can help us to produce these three enzymes. And also we have tested out several of these constructions in our model. So the next step is scaling up the production of PHB as soon as we get the synthesis of these three genes. So that's it.
Yeah, great. Making something antimicrobial while maintaining biodegradability is like truly a balance that is perhaps like hard to get, but this is like a really interesting attempt. So does someone have any questions here? Any? Okay, so I saw you had some chance of working in the lab. What are
Yes.
like your next steps?
Okay, well, what we're waiting at this moment is because we have already several plasmids from this Picambia that we have been working together with Sebastian Kuchova. But normally these plasmids are working in plant, but this can be easily well adapted for algae. So the next step is actually testing different versions of these operons, so we can optimize the production of these bioplastics. So as you can see in the bioreactor I'm presenting in the picture, it's just one little reactor, but probably if we intend to scale up the production, we can go up to several 100 liters. And the second thing is probably speaking with other people that is interested in implementing into medical devices. But I guess that requires a further collaboration with even doctors and specialists on applying these technologies to medical devices.
All right, fabulous. Okay, so if there are any other questions, please put them into the chat. And we will be moving on to the next presenter, Elizabeth Wink. Great work, David.
Hi, I'm here. Can we advance to the next slide, please?
Yes, Victoria Makerspace, let's go.
Thank you. I'm interested in developing new mechanisms to deliver therapeutic DNA to target cells. Traditional gene therapy vectors such as AAV are extremely well characterized. However, they're slightly limited in their low payload capacity. Non-conventional gene therapy vectors such as herpes simplex virus 1, due to their larger genome, can encode a much larger transgene within their genome, which allows us to target many different genetic diseases that have genes that won't necessarily fit into the AAV vector. One complication with working with herpes simplex virus 1 as a gene therapy vector is it has to be manufactured in a complementing cell line in the presence of virus proteins. Virus proteins are good and bad. They are essential for vector manufacturing, and in the context of a gene therapy vector, they sometimes stimulate transgene expression, which is good. However, virus proteins can be toxic to the mammalian cells that they're produced in, and obviously we don't want to express them in patients receiving therapies where they could be harmed. So I want to know, how can we balance, take advantage of virus proteins to balance high transgene expression with low cellular toxicity? Next slide, please. To do this, I want to re-engineer the HSV1 protein ICP0. ICP0 is kind of like a Swiss Army knife protein of herpes. It's a multifunctional protein that is, during a normal infection, supports productive replication. In the context of gene therapy vectors, ICP0 can stimulate transgene expression, but it's also pretty toxic. It can kill target cells. So I want to know if we can re-engineer ICP0 to keep this transgene expression benefit while lowering its cytotoxicity. So in order to do this, I want to make an ICP0 reporter library and then characterize it in mammalian cells to see if it can impact gene expression, cytotoxicity, and virus growth. And then for promising mutants, I want to actually clone them into a gene therapy vector and see if they could possibly be a useful tool. Next slide, please. This is just a basic rundown of my ideal workflow. First, I want to start by making a mutant ICP0 reporter library. I can make a series of ICP0 truncation mutants where I eliminate well-recognized domains within the protein, such as the ring finger domain or the nuclear localization signal. And I also want to take advantage of the fact that multiple strains of herpes exist, each expressing their own form of ICP0. By swapping around domains of ICP0 from different strains, I can make my own protein chimeras and hopefully play around with cytotoxicity versus transgene expression. I want to clone this into a reporter plasmid with EGFP, introduce this into mammalian cells, measure gene expression, cytotoxicity, virus growth, and then basically select mutants where ICP0 is able to stimulate GFP expression with minimal cytotoxicity. The most promising proteins I'll clone into a gene therapy vector and see if it's possibly a good tool in the context of gene therapy. And thank you.
Great presentation, Elizabeth. Do we have, yes, applause for you. And do we have any questions here? In that case, do you have like any plans, or have you thought already about how you want to measure the expression? Do you have any experiments already lined up that you want to do? How do your next steps look like?
I've measured transgene expression in the past. I'd probably want to perform Western blots to look at total protein level, or I guess in the case of GFP, I could just measure fluorescence over time. But I'm also interested in comparing both like the maximum amount of protein that's being expressed and how long it lasts too, because both are important for gene therapy.
Yeah, these are great avenues. Does anyone have any further questions? I bet there will be questions streaming in the chat. Just because we are a bit behind, I will move on to the next presentation. Great work, Elizabeth, again. M. Samudra.
Okay, that's
Ilham
me.
from BioClub. You're there? Okay, great. BioClub
Okay.
Tokyo. Let's go.
Yes. Okay. Thank you. Hello, everyone. My name is Muhammad Samudra Ilham. I come from West Sumatra, Indonesia, and I'm currently a second year undergraduate student majoring in biology. Next slide, please. So the final project title is Enhancing Bioadherent Binding Protein Specificity to Fluorescence. This work focuses on using computational approaches to improve how honeybees OBV5 proteins recognize fluorescence while reducing interference from air pollutants. How honeybees rely on a protein called OBV5 to detect floral volatiles for effective foraging. However, air pollutants like nitrogen oxide and nitrogen dioxide can bind to the same pocket in the OBV5, reducing their olfactory accuracy and potentially lowering pollination efficiency. To address this, I aim to improve the selectivity of OBV5 toward floral ligands using molecular modeling, machine learning, and in silico gene editing. I use a 5-step computational pipeline. First, I run a molecular dynamic simulation to generate stable protein conformers. And then I perform ensemble docking using 4 ligands, 2 floral linalool granule, and 2 pollutants, nitrogen oxide and nitrogen dioxide, and recorded binding affinities and key residue interaction. Next, I applied machine learning, that is random forest and SHAP, to rank residues contributing most to ligand selectivity. 3 top candidates, there is isoleucine 68, arginine 16, and leucine 72, were then mutated in silico using Pymol Wizard. And finally, I simulated CRISPR-based integration of the mutant gene, checking primer specificity and homology arms using Benchling. Next slide, please. Okay, the mutated OBV5 retains similar affinity to pollutants, but in this results, no increased sensitivity, but it shows stronger and more specific interaction with floral ligands, particularly through hydrophobic pocket reshaping. This suggests a promising route for enhancing floral specificity without compromising protein stability, as confirmed by RMSF analysis and docking consistency. This mutation, isoleucine 68 to leucine, arginine 16 to glutamine, and leucine 70 to valine, seem to redirect ligand binding toward beneficial volatiles while minimizing pollutant effects. In conclusion, this study demonstrates the potential of computational protein engineering to enhance olfactory precision in bees. In the future, I plan to validate this model experimentally, including proper cross-validation for machine learning and wet lab testing in vivo or in vitro. This approach could help preserve pollinator performance in polluted environments. That's all for me. Thank you very much.
Thank you so much. It's very interesting work. I'm curious if someone has a question.
Yeah, I have a question. And really fantastic presentation. Really great job. I'm sorry if I missed this, but what is your experimental plan to actually detect the ligand binding experimentally?
From experimentally, in silico experimentally, for docking, molecular docking, and then
No,
I
I mean
use
in real life, like not in silico, to actually perform the measurement.
maybe for the actually measurements, I haven't an idea from now, maybe.
Okay, I'm just because you've done some really great modeling, and I'm just wondering how you would actually test it in real life.
Maybe in the next plan is I'm to measure the activity in E. coli first, and then that is get the CRISPR gene editing in genome of bees after the E. coli activity.
Great, thank you.
Okay, thank you so much. Any other questions, please address them in the chat.
Very quick just question. I'm like, it could always be
Oh.
really interesting, like one look at assays for this, but I'm curious, like, what was your experience working with a bunch of these softwares, like, especially because there's a lot of like great work with these kinds of simulations. So I'd love to hear a bit more about that.
Okay, sorry.
Oh no, I just wanted to hear your experience on these. Like, how did you go about choosing these models for like predictions and so forth? Yeah.
Yeah, I'm using machine learning, random forest, and SHP. Using the interaction key before the molecular docking 3 snapshot and 4 ligands. So that is the input and output is the 3 targets side directional mutagenesis.
Gotcha. Cool.
Okay, thank you.
Cool work. We will have to move on. Thank you so much, Samudra. The next person to present is Alexander Proaño, Ottawa Bioscience. Are you here?
Hello, yes, I'm here.
Okay, go for it. Stage is.
Okay, thank you. Well, hello, my name is Alexander. I'm working with the Ottawa Bioscience node on a synthetic biology proposal. This is focused on sustainable soil restoration. So the project aims to develop a bio-based platform that transforms agricultural waste into functional soil inputs. Specifically, we propose to engineer enzymes that convert lignocellulosic residues into xylo-oligosaccharides or XOS. So basically, these are the residues, and this is what we're gonna do. We use synthetic biology, enzyme discovery, artificial engineering to improve enzymes that are already doing this. So after that, the end product is soil basic chemical building blocks. Please, could you turn to the next slide? Okay, but why do we are doing this? It's because a lot of farms are performed in this type of landscape. So traditional mechanical ways to restore the soil and avoid erosion and traditional chemical waste are leading to erosion and loss of benefits of the soil. And also because usually a lot of farmers have this practice to burn after each crop. So this kill all the microbiota of the soil. So this leads to erosion. So here we propose the option that is promoting the restoration of microbiome with soil prebiotics in conjunction with microbial ecology and soil health. Please, could you go to the next slide? Okay, so how are we doing this? For this particular presentation and project, we focused on the sugarcane bagasse as a source of xylose. After that, we engineered a vector with several advantages. They have thermostability, activity, and the novel thing here is that we are using several mutations that have been previously studied, but we mixed them in this construct. So after this, we produce the xylo-oligosaccharides, and in the future we're gonna produce other type of this compound. Another thing that is important is that we add thermostability because of the purification method to just use the whole cell lysate. Thank you.
Okay, great. That was right on time. Does anyone have questions? Okay, so great work, Alexander. Were you able to do any of this, or like do any work in the lab, or do you plan on doing anything in the lab? As of now, like any experiments?
until now, not because the tree starter doesn't come to the node yet, but after I want to try some basic enzyme characterization. So that is going to be the first step to do before doing the whole project. I don't know if you have other question or that was the question.
That was the question. Yeah. I hope
sorry.
you get to do some of the work in an actual wet lab. Does anyone have any questions? Because else I would move on to the next presenter. Great work again, Alexander. Please address any further questions to Alexander in the chat. Anastasia Bernas from GenSpace. You're next. Are you here?
Yes, hello.
All right, let's go.
My name is Anastasia. I am UBC alumnus, and my project is about sustainable production of renewable diesel. Next slide, please. It probably wouldn't surprise you if I told you that the energy sector, and particularly fossil fuel combustion for transportation, is one of the major contributors for greenhouse gas emissions. That is why we thought of biofuels. But while the idea of biofuels is already a step forward towards saving the environment, it is still far from ideal. In fact, all biofuel generations share one issue. The chemical processes involved in the conversion of the feedstock into the fuel are complex and energy intensive, which often offsets the potential benefits of the biofuel. Now, imagine that biofuels like renewable diesel, which is chemically identical to petroleum diesel, can be produced from waste materials in a much greener way with just 3 enzymes, a lipase, a decarboxylase, and a reductase. Next slide, please. The first aim of my project was to design more catalytically efficient mutants for my 3 enzymes of interest using a Python script that combines several publicly available machine learning tools. As a result, for all 3 enzymes, I was able to increase the turnover number, KCAT, which represents the number of substrate molecules that can be converted into a product per second. For the lipase, it was a 22% increase. For the decarboxylase, it was a 63% increase. And last but not least, one of the reductase mutants showed a 4,023% increase compared to the wild-type enzyme. It is important to note that the original reductase substrate is structurally quite different from the substrate that I wish to use for the production of renewable diesel. And since I used the unnatural substrate to characterize the reductase mutants, most of the KCAT values were quite low to begin with, including the wild-type KCAT value. Nevertheless, the mutant with the significantly higher turnover number indicated a mutation that was able to expand a substrate specificity of the reductase, which makes me quite optimistic regarding the possible future results. Next slide, please. Moving on to my second aim, I would like to run in vitro assays with the successful mutants and analyze their products, as well as develop a novel immobilization method using a more hydrophobic, biodegradable polymer called cellulose acetate butyrate. Finally, my visionary aim is to introduce enzymatically produced renewable diesel to the market for public consumption. There are a few challenges that I came across while working on the project, and one of them is the fact that the machine learning tools like EvolvePro require an in-lab validation of the suggested mutations, which I wasn't able to do since I had no access to the lab. Also, in the future, I would like to immobilize my enzymes for large-scale production, and it's a bit tricky for the decarboxylase because it has to be immobilized in a specific orientation. And the reductase is NADH dependent, so I will have to add a cofactor regeneration system to my bioreactor. Thank you for your attention. And I would also like to thank the HGGA community, David, George, Joseph, and especially GenSpace TAs for their support and guidance.
Amazing presentation, great work. Does someone have questions? Yes, Suvan,
Yeah, I
please
would
go.
like to have a question. So basically, have you considered the processivity rates of each of the 3 different enzymes. So basically, in order to undertake a chemical process on a large scale, you basically have to synchronize the processivity of the 3 enzymes so that you will only be able to convert an equal number of molecules and substrates. And also, I was wondering what would be the substrate utilized for this biodiesel? Would it be like something which is widely available, or would it give rise to the dilemma food against fuel, as it used to be the case for the first and second generation biofuels?
Right. So the renewable diesel as a second generation biofuel. So its feedstock is mostly vegetable oils that are not good for food consumption, as well as animal fats, and also used cooking oil. So that would be the initial feedstock, kind of after it's prepared, purified. The triglycerides would be the substrate for the first reaction for the lipase, which would turn the triglycerides into the fatty acids, and then they would be decarboxylized by the SIP, and then turned into
Unfortunately, I have to interrupt you. You can perhaps write it out in the chat, but we have to move on to the next one. I'm so sorry.
Sure.
Suvan, also address your question in the chat if you want to. We're going to the next presenter, who is Marlene Tarja, and great work, and again, Anastasia. Victoria Makerspace. Are you here, Marlene?
Yes, hello.
Yes, please go for it.
Hi there, my name is Marlene Tarja. Today I'm going to take you through the looking glass to explore the design of a mirror image version of Gyras. Next, please. Gyras is a bacterial type 2 topoisomerase which untangles knots from DNA, facilitating its replication. As most proteins in nature, Gyras is made of L-amino acids, which are chiral molecules that polarize light to the left. My aim was to convert the Gyras A subunit, GIR-A structure, to its mirror image composed of right-handed D amino acids. Using a tool called Finder, the first step was to remove the DNA atoms that were present in the original GIR-A structure. Then, just by flipping the atomic coordinates along the x-axis of the PDB file, an in silico mirror GIR-A was generated. Why is this important? Mirror proteins and the peptides have gained attention as potential candidates for novel drug discovery, since they present low immunogenicity and are safe from protease degradation. These enzymes won't recognize them as targets because of their different chirality. In the future, I would like to synthesize a mirror GIR-A, which could add to the repertoire of key enzymes needed to generate a mirror bacteria. However, we are all aware of the potential risk of generating mirror life. So my next step was to identify a peptide which could function as an inhibitor of these mirror gyras. I targeted the dimeric C-gate GIR-A region at position 386, which maps to the homodimer contact required for gate closure. The C-gate is the exit of the DNA from this enzyme. So it is a proven target for killing bacteria. Then I performed a mirror image virtual screening, and the top binding candidate was this short 12 amino acid residue peptide, which has an alpha helical conformation. It is important to notice that for simulating this mirror GIR-A ligand interaction, direction of alpha helix rotation was taken into account to avoid missing any key binding features. Therefore, it's not just swapping left-handed for right-handed amino acids. When there is the presence of alpha helices in the structure, keeping correct backbone side chain connectivity and geometry is crucial. Next, please. Overall, I may have provided both the poison and the cure, thanks for your attention. And I want to give a special thanks to my final project TA, Steven, and also to Derek.
You can continue. Oh, unfortunately, you cut off, unless I cut off. But that was a fantastic presentation. You went into such detail on mirrored amino acids. Does someone have questions here?
Thank you. And I hope I didn't cut for everybody. Sometimes my internet connection is terrible.
No worries at all. No worries at all. Does someone have any questions here?
I have a quick question. Why did you pick gyrase of all the proteins you could have picked?
Because it's a very fascinating enzyme. It's involved in many important biological processes such as replication and transcription and keeping genome stability. So right now they have already synthesized mirror proteins such as ligase and a DNA polymerase. So I think that also having this mirror gyrase could help for the replication process in a putative mirror bacteria. And because it unentangles DNA, so I think it's an amazing molecular machinery. Like in the top right, you can see a model of this incredible protein.
Thank you. Nice presentation.
Thanks a lot. I really appreciate your question.
All right. Great presentation. I also used to work on establishing alternative deamino acids and the incorporation into alternative proteins. But let's move on to the next presenter. Any other questions, please address them in the chat. Kale Harrison from Life Labs, are you here?
Hello, I'm here.
Okay, perfect. Let's go.
If we could go to the next slide, please. Hi, everyone. My name is Kale Harrison. For my project, I am looking at using synthetic biology for the biocontrol of black mold. The UK is a very damp island, despite the background behind me. What this means is that there is a cost of £1.4 billion a year on the healthcare system due to cold and mold. And dampened mold causes nearly 15,000 respiratory illnesses a year. However, black mold is famously stubborn to remove from properties, and it often impacts people who are least empowered to fix it. Therefore, this project envisages using either bacteriophage or microviruses to remediate black mold. I've covered my aims there, but in a subsequent part presentation, we'll go over in further detail, so I'll just skip over those for now. I have here a photo of Aspergillus niger or brasiliensis, as it has been renamed to. I'm going to use Niger throughout because I can pronounce that more easily. And then I also have a benchling of the codon-optimized microcryvirus that I decided to use. And then if we could go to the next slide, please. Many microviruses have no impact on fitness. There's a few that do, so I found one which does, and then impacts Aspergillus niger, mainly via suppressing RNA silencing. However, microviruses don't really spread like a conventional virus. They spread when the mycelium splits, so it kind of has to be there already. So to overcome this, there's something that many of us have in our house, the humble spray bottle, which you can use to apply dsRNA, and then that can actually biocontrol. This has been demonstrated in myrtle rust, but it has not yet been demonstrated for A. niger. So my plan is to cultivate A. niger, then take the twist order, turn it into RNA, and then thermocycle it so it becomes double-stranded RNA. Then I will use gel electrophoresis to verify that that has happened, and then suspend it as a solution in water, basically just stir it in some water and then spray it over the petri dish of A. niger and see if that impacts the growth. And then we'll also, as you can see here, have a control, which is just plain water, just to make sure that we're not capturing some other effect as a control. If we could go to the next slide, please. And then in terms of the expected outcomes, a clear result will demonstrate whether or not the proposed solution is effective. And then also making sure for AIM2 that we have demonstrated the solution would be both safe and responsible in real-world usage. So things such as checking for homologs, which I have already done to an extent, but of course you can keep going, check other microviruses, and also decide how to test strains, because Aspergillus niger is not the only one, it's just one of the most predominant. And then also keen to roll out a solution and reduce the impact of black mold in the UK and rest of the world. Very keen for that to be not-for-profit, inspired by particularly the paper microscope presentation. I'd like to thank the professors, the various labs, especially Life Labs, all the fellow students and the presenters. And if there's any questions, please.
Amazing work, Kale. Yeah, the question stands. Are there any, is there anyone who wants to ask a question? So did I understand it correctly that you were already able to materialize some of these ideas in real life? Like, have you already done some experimentation?
So we're looking to develop the experimentation now. The key rate limiting step is the twist order, and also the Aspergillus niger. So there's two things that we need. One is to order the Aspergillus niger, which I think we're making progress on. Had some great conversations with various people in the HCGA community in the US. But then for the twist order, it would be great if there was any sort of update on timelines there, because that's kind of the other thing needed. But everything else we've got mapped out and have budget attached to.
All right. So people might be just shy, so look up for the chats. There are going to pop up a lot of questions, I'm sure. So I'm, in that case, going to move on to the next presenter.
So I have a question for Kale, actually. So how do you engineer the system?
Let's do it in the chat if it's okay, Federico. So I'm so sorry. Let's do it in the chat, and we'll keep. We're still a few minutes behind, so let's keep it in the chat. Thank you so much.
Yeah, I'm so sorry. Great work, Kale. Laszlo.
Yes, I'm here. Can you hear me?
Yeah. Yes, I can hear you. Life Labs, go for it.
Perfect.
Sorry.
Okay, so my name is Laszlo, and I'm an architect and multidisciplinary designer based in Lisbon. And we can go to the next slide. So I was wondering if we could measure and visualize the level of stress in different urban areas, because I think mapping this could be useful for urban planners and policymakers. Other sensors detecting like individual stress, like based on heart rate or skin temperature, they already exist. But because I'm interested in the population level, I turned towards volatile organic compounds in exhaled breath. And these are products of human metabolic processes. So there's a chance that the right receptors already exist in humans, or similar receptors could be designed based on these. So I envisioned a biosensor, which could be constructed in a yeast cell, because they successfully expressed a G protein coupled receptors in yeast cells already. So I envisioned this genetic circuit which could process the signal and create an output. And in the next slide, I go through the steps of this design, starting with identifying the volatile organic compound. So several studies already prove that there's a detectable volatile organic compound footprint in the human exhaled breath of stress. This is a mix of compounds. They act together, and also some of them are still unidentified, and most of them are actually regulated. So in a future design, I would like to include the possibility to sense the absence of certain molecules relative to a baseline, for example, CO2. And then as an AIM2, I would like to then run like an in silico GPCR finding. I will work on the steps, and then I can use a yeast plasmid to to express these genes and for testing and fine tuning before it's integrated into to the chromosome for more stability. And then the same applies for my output design. I designed this oscillator of free chromoproteins, which results in a dynamic color change using these operators and genes inhibiting each other in a circle. Because if we go to the next slide, apart from collecting data from these individual sites and merging them on an online map, I would like to display it also on the site. So I would like to scale up the image of the yeast bioreactor and using camera and screen. And then I would use mirrors to make this effect so that if there's no stress, only the base color of yeast is visible. But if stress is sensed, it creates this mandala image, which mandala is associated to focus and mindfulness. So in a way, it creates this kind of feedback loop for reducing stress. Thank you very much.
Thanks, Laszlo. Any immediate questions? Because, oh, Suvan, okay, quick.
Yeah. Quick question on the GPCRs. So how do you plan on testing those? Because I know those can be very tricky to test sometimes, especially the interactions. So I'd love to hear more.
Yeah, so a couple of options. I was thinking first to do a directed evolution in lab, but I haven't researched because I didn't have any access to any kind of lab. So, but it would fascinate me, and like to optimize basically the like already existing binding sites more. But then in silico, I imagine like using some machine learning to already existing known binding and GPCR reactions. I would make a database, and then I could maybe, yeah.
Okay, great. Thank you.
A lot to
So
research.
unfortunately, we will have to move on. Any other questions, please address them in the chat. Hanna Urukalovic, like this. Are you here?
Yes. Hi.
Okay, go for it.
Of benzyl acetate from PET, and I'm a second-year biomedical sciences student at King's College London. Next slide, please. So a bit of background. Plastic pollution is a significant global problem. And to date, 9,200 million metric tons of plastic have been produced, 91% of which has not undergone any recycling. Of the 9% that is recycled, mechanical recycling is the most common today, but an unexplored area of development is secondary recycling opportunities. So my solution here is using PET as a substrate for bioproduction. PET is usually used in closed loop recycling, meaning that it becomes another plastic product like a bottle, or it's used in textiles to make polyester. But using it for bioproduction would eliminate it from the ecosystem. My aim one was to create a pathway going from PET to benzyl acetate, which is the bulk of the project. And benzyl acetate is a fragrance compound which smells like jasmine, and it's usually extracted from plants or synthesized chemically, but these processes are both difficult and use harmful chemicals. However, this approach would minimize the use of potentially harmful substances and would also help combat plastic pollution along the way. So using the previous work that has been done on this topic, I made a 3-phase system. So far, the work has been done on PETase and metase systems, which digest plastics. There has also been work using ethylene glycol to make amino acids and using glucose as a substrate for benzyl acetate production. So I connected these 3 metabolic pathways and found DNA sequences for all the enzymes and codon optimized them for expression in E. coli. But for the 3rd phase of the pathway, I also used SynBioCAD
to model the third phase of the pathway using the retrosynthesis workflow, and it came up with a 6-enzyme pathway for phase 3. Next slide, please. For AIM1, I also made a theoretical bioproduction pipeline, which is illustrated here on the slide. And the plan is to use transfected E. coli for chassis or cell-free systems, and to ultimately use high-pressure liquid chromatography to isolate the benzyl acetate. For AIM2, my goal was to validate the end steps of the pathway, since there's been less work exploring these particular steps. And I made and ordered a PET28 plasmid with an insert for alcohol O-acetyltransferase 1 fused to GFP with the linker, which is the last enzyme of the pathway. So the experimental protocol will be testing the last 2 steps of the pathway. Next slide, please. So the protocol is here. So I would start by testing the toxicity of benzaldehyde to E. coli, which is a substrate that I would need for the last 2 steps of the pathway. And then I would grow the transfected E. coli with LB and with benzaldehyde, and validate by using HPLC to see if benzyl acetate was produced, and the GFP will indicate if the protein was expressed. For AIM3, that's future work, and it's mainly to do with optimization of the reaction conditions, protein sequences, and maybe changing the chassis organism. And the next step would be to upscale using bioreactors and use genomic integration instead of plasmids, and automate using Opentrons or the systems that Ginkgo Bioworks uses, and to potentially create pathways for different useful molecules. Thank you. I'm sorry I ran over time.
Oh, that's absolutely fine. Perhaps we will.
I have a quick question.
Quick.
So do you think that the phenylalanine or benzyl acetate is more valuable in the market?
Yeah, that's an important question I've been thinking about recently. My initial inspiration for the project was just based on my passion for perfumery. But along the way, I did also realize that perhaps just making phenylalanine would be useful enough. And it is, yeah, it's also valuable, and it would require less steps to make it. So perhaps that's a better route in the long term.
Thank you.
Thank you.
Nice presentation.
Okay. Fantastic, and fantastic discussion. For any further questions, address them in the chat. And we will move on to the next presenter, David Chow, GenSpace. Are you here, David?
Yes,
Yeah,
I'm here.
go-
Can you hear me?
Yes, go for it.
Well, thank you. So hi, everyone. I'm David. And for my project, I wanted to make bacterial cells talk to each other. So let's go to the first slide. Okay, so this project came about because a classmate pointed me to this thesis by J. Padmakumar, who was a PhD student at Christopher Voigt's lab at MIT. In this thesis, he engineered E. coli to compute the MT5 cryptographic hash function. And when I saw this, I knew I wanted to do a project based on this thesis because it combined a lot of my own interests in computer science and cryptography. So in this class, when we first heard about genetic circuits, I thought you can just take any genetic circuit, put it in the cell, grow it, and read out the results. Turns out it's not so simple. You can only put maybe 3 or 4 gates into a cell. Any more than that, you have to distribute your computation across multiple cells, and then you need to make the cells talk to each other. So that's what I wanted to focus on for my final project, cell-to-cell communication. So to make cells to talk to each other, you need one cell to make some sort of signaling molecule, and you need another cell to receive it. Making the signaling molecule is just bioproduction, which we've talked about in this class. Responding to that molecule is a biosensor, essentially, which we've also talked about. The challenge is that you need to pick a signaling molecule that you can both make and detect, and that's a little tricky. Fortunately, in his thesis, Padmakumar identified 4 different signaling molecules that worked well for him. I decided to pick 2 of those and try to make them because I thought they'd be feasible. So let's go on to the next slide. So how does this work? Let's take one of these signaling molecules. I picked this one because it's simple to produce. You just need a single protein called LuxI, and that protein can produce the signaling molecule. So You just need to take that protein and you can order it from Twist. Twist has vectors that already have the promoter and RBS, so you just need to tell them what protein you want to make. And you can take that plasmid, put it in a bacteria, and it'll express the signaling molecule. Detecting that signaling molecule is more interesting. In your receiver cell, you can make this protein called LuxR. When LuxR binds to the signaling molecule, it changes shape, and then LuxR travels over down to this Lux box promoter, PLuxB on this diagram. And that promoter is normally turned off. When LuxR binds to it, it turns it on, and then you start expressing whatever that promoter is promoting. So to recap, this all works because we have this special promoter that you can turn on and off. And I mentioned before, I picked two signaling molecules to implement. The other one is this other one you can see on the screen on the right. The mechanism is very similar, so I won't have to go into all the details, except that it's a different protein and a different special promoter. And for one of these signaling molecules, I chose to make the bacteria turn blue if it's detected. For the other one, I made it turn red. So I will say, in this project, I'm not really discovering anything new. Everything here comes from existing research, but the variant of the components I'm using were all things that were published fairly recently in the past few years. So one of the cool things about this project is that I'll get to replicate some pretty recent research. Let's go to the last slide. Yep. So I was really hoping that I'd be able to show you this all working, and you can see bacteria changing color. Unfortunately, the orders from Twist haven't arrived yet, but when they do, I'll transform them, grow the bacteria, collect the signaling molecules in the supernatant and put in the receiver, and hopefully the receiver will change color. There are some measurements I want to make, and I also have some ideas for extensions of this project that are on this slide. But overall, it's been a fun process. Before this class, I had very little idea of how genetic circuits work. Having gone through this project, I now understand a lot of this thesis and could conceivably even replicate a lot of it if I had more time and equipment and a much larger Twist budget. So that's been really fun. And that's all I've got.
Okay, great, David. Thank you so much. Unfortunately, the timer is a little bit weird, so I'm assuming we are at 4 minutes 40 now. So we will perhaps delegate the questions to the chat. But amazing project. Sorry for the time confusion. Yeah, let's restart the timer. And please ask questions, chat, to David. Next one, Oscar Schrack from GenSpace. Are you here?
Yeah.
Yes, please go ahead.
Hello, how's everybody? My name's Oscar. I am at GenSpace, and I'm working to make PHA biopolymers out of agricultural waste. And so there's about 400 million tons of plastic waste created each year, and then over 80% ends up in landfills. And so that's like a truckload every 3 seconds, which is kind of wild. Next slide, please. So this project, we're trying to optimize my creation of these PHA biopolymers. And so the method of doing that is to create more NADPH in their environment so that they produce longer and more chains of PHA carbon. And so we're doing this through like a growth optimization where it's co-growing with E. coli that's producing this NADPH, which was what we ordered from Twist through this NAD synthase codon. And so this is just some process work that I'm trying to show here in kind of our circular economy process and what PHA is and some of the lab work we've done so far. Next slide, please. Yeah, so that's the codon. This is some bacteria streaking I did. This was one of the ligation checks. And then this is the general experimental design where I want to grow them separately and then co-grow them and then see if I can get a greater PHA outlet through that or output through that. And so that would be checked through FTIR. Yeah. And so next slide. So sadly, the controls I was running got moldy while I was waiting to prep them for FTIR. So I'm rerunning them again this week. So we should have a baseline to go off of. And then I want to redo my plating of the bacteria just because it got a little overgrown with the lawns. And then, yeah, kind of establishing that co-culture and separate cultures, protocols, so that we can have something to work off of. And then FTIR, all of those. Yeah, thank you.
Okay, fantastic. Does someone have questions? Yeah, sorry for the mold. Do you have any plans for perhaps scaling the process up?
Yeah, if it works, I would love to get it into larger and larger bioreactors. Right now, I guess we're still at the shake flask and kind of up into volumes right now. But yeah, that's the plan.
Okay. Are there any further questions? Because if not, I bet people are cooking up some questions right now.
Yeah, there are questions in the chat, but we can keep moving on.
Yeah, then let's keep moving on. Seems like we have a 5-minute break. Okay, no, this has been a great session, extremely great discussion from what I've seen in the chat. So thank you to everyone who has presented thus far. We will keep the stride going in 5 minutes with Megan. Megan, are you here?
Hey, yeah, I'm here.
Okay,
So,
great.
yeah.
Then no worries. I will stop the screen share in that case and head it over to you.
Sounds good. Yeah, let's make sure that the screen sharing is working. I think someone needs to approve it.
Recording stopped.
Yeah, go ahead, Megan. Try putting the screen share.
Yeah. Again, brilliant presentations. Great work. I'm
Megan,
really
you're
looking
sharing-
forward to the next one.
Megan, you're sharing notes right now, not
1 second. Yeah, let me just
And TAs, who's doing the timekeeping for the next 2 hours?
That's me, David. Yeah.
Oh, Digby. Okay, great, good. And you have the right sound to play for the
Yeah, I was worried about that last one because I had it set on another one, but luckily they were.
Good. Yeah, there's been a lot of good discussion about the noise to play. We've landed on a good, gentle one, so
yeah,
let's keep that one. Great. And then okay, yeah, perfect. Megan, you want to try to share on your just full screening the slideshow? Make sure that
yeah, sure. And then if we could also just make sure that the timer is working. Is it working now, the screen share?
Yeah, the timer's working. Yeah.
Timer's working. Okay, and can you see the full screen?
We can, yeah. Can you see the timer? It's very important that you see the timer.
Yeah, so let me see. I see it.
Right. Okay,
yeah.
perfect.
Hold on.
It'll be in the top right and on my screen as well if I get my camera on.
Megan, are
Okay.
you
Yeah.
good?
Yeah, I'm good now. So let me just actually, the way I have everything set. Okay, so we have 2 more minutes. And I guess that we actually are technically still on schedule, so we can still keep with the normal time for everyone's presentations.
Oh yeah, we've been remarkably on time this whole day. And actually, maybe just to pause for a second here, I'll start the recording again.
Recording in progress.
We're now, how much time do we have left? We've got 3.5 hours left. How many hours? We've been doing this for like 7 hours. I don't even know how long. We started this at 9 in the morning, I guess. So it's been who has been here this whole time? I know there's at least a few of you. I know Dominica has, Adrian.
I have.
Oh, Anastasia, nice, great. Yeah, we were saying this yesterday, but for any of the students that have been here for the whole time, we're gonna give a special recognition to you all at the graduation. So this is you all get the How to Grow a Marathon Award. So we'll give you a special acknowledgement for everybody that's been hanging out this entire time. It's been a crazy crazy day, but just again, so impressed by all of the diversity and depth and creative thinking and hard work that y'all have been showing. Really impressed. And just note too, I know we've mentioned Twist a few times. Now that we've gotten all the MIT orders in, we are prioritizing all of your orders right now, and so Suvin's been processing all those. Emily has been so fantastic and has been working to get those orders turned out to you guys. So just know that all of those orders are coming your way. If not now, very soon. So stay tuned. And I'll turn it back over to Megan. Megan, thank you for being MC for the next 2 hours. And we can get started, actually, if Allison is here and ready.
Of course, yeah, if Allison Cabrera from SynBioGenetics is here, yeah, we'll kick off the rest of these presentations. So we still have more than 3 hours to go. So yeah, without further delay, Allison, go for it.
Yeah, okay. Can you hear me?
Yeah.
Yeah, thank you. So, hey everyone, I'm Allison, I'm from Ecuador, and my project is the production of recombinant dispersin B to trade biofilms in a cell-free system. So please next. Yeah, so first of all, let's talk about biofilms. So these are structures created by bacteria in order to protect themselves from the external environment. So this is a big issue because it makes bacteria harder to get removed and causes like billionaire losses all around the world and in many areas. And it's even worse in developing countries like mine that lack of these technologies or emerging strategies to prevent. So this is how the idea comes in. And I propose the use of these dispersal enzymes, specifically dispersin B, that attacks directly on the biofilm matrix. And I work on the 3 following aims. So let's start with number 1. Next, please. So aim number 1 was based on the construction of this plasmid, of this genetic construct in order to produce the dispersin B. So for this, I selected a truncated version of the gene. I did like the codon optimization for E. coli, and I also choose a plasmid and the BBT7 as a backbone that luckily was available here in my note as well. And yeah, this is the diagram that I did in Benchling with the plasmid and the many different parts. And those are in more detail in table number 1. And I wanted to highlight the use of this signal peptide because it's going to be helpful in further steps for the recovery of the enzyme. And well, this was done mostly in silico, of course, but I also added like the laboratory workflow that you need to follow. From the sample preparation to the bacterial transformation, and I expect to obtain like a good quantity of this enzyme. Next, please. So this led us to aim number 2, which is basically produce this enzyme, but also use a CF system, and this in order to increase like the production, the scalability, and also to have more control and experiments. And this led us to aim number 3 that is focused more on the future. And for this, I wanted to evaluate like the enzyme activity directly on biofilms. And I don't have like the results yet, but I expect to obtain results similar as they're shown on the images on the screen. And that shows the degradation of biofilms using different stains. And this led us to the last part, which is the design of these products for the market. And I did like two prototypes, and one that is a ready-to-use solution, another one with the lyophilized enzyme. So in conclusion, I think this is a great affordable strategy to combat biofilms. And I think in the future, it will be interesting to study different mutations around the dispersin B gene, or maybe add another compounds to increase the enzyme properties. That's it. Thank you.
Thank you so much, Allison. Does anyone have any questions? So yeah, it's a really great project. And if anyone has any questions, you can put them in the chat. If we don't have anything right now, I guess we'll move on to the next presenter. So Paul Romero from Ottawa Bioscience, are you here?
Sure, I'm here.
Perfect. Go ahead.
Thanks. Hello, everybody. My name is Paul Romero from Ecuador, and my final project is bioproduction of the compound 3N-butylsalide MVP. Please, next slide. The compound MVP has various therapeutic properties, one of which it is an action has a antihypertensive agent. Is an active compound found in plants of the APC family. One of the biosynthetic pathways in plants involves the conversion of the substrate zinc-quinolate to MVP through the action of one enzyme. The DNA sequence of this enzyme was discovered by me and colleagues in 2024. They named it LC2-OGD2. Please, next slide. Why carry out this project? Because according to the World Health Organization, the global incidence of hypertension in adults has doubled over a 30-year period. In Ecuador, according to the STEP survey, 19.8% of adult population has been diagnosed with hypertension. Additionally, according to the Pan American Health Organization, 1 in 5 Ecuadorians suffer from these conditions. Although there are various pharmacological treatments, many patients do not follow them due to side effects. Therefore, the bioproduction of the MVP compound could represent a promising solution to this problem. Please, next. About the methodology. To achieve AIM-1, which involves optimizations, the conditions for the production of the MVP compound, we plan to develop the heterologous expression of the LC2-OGD2 enzyme, which is responsible for its biosynthesis. Therefore, steps such as cloning, transfection, and induction will be carried out. Subsequently, the MVP production step, a feeding assay will conducted where different concentration of zinc-unolite will be added to the L medium so that the E. coli cells can assimilate this substrate and transform it to MVP. Finally, the MVP product will be extracted with 3 times of this volume ethyl acetylate and its quantity by determined of GCMS analysis to achieve AIM-2, which involves the scaling up the production of this compound. A small scale feed batch bioreactor will be used. The stages of the production process for the feed batch bioreactor are shown in this figure. Each of these stages has been developed based on parameters used by Glaser and college in 2020. This methodology is expected to achieve high density, high cell density, and high yield of MVP. And the AIM-3 is to explore the potential biotechnological MVP production as an alternative to traditional methods. To achieve this first literature review can be
so sorry. If you could just wrap up like your final point, because we're starting to be over time.
Okay.
You can still say like one last thing, but if you could just I guess wrap it up, and then we'll probably just move the questions to the chat for your project. Do you have something final to say about it? Paul, are you on the line?
No, sorry. I would like to mention that the AIM-3, to achieve the AIM-3, is necessary a literature review and analysis of biological activity of MVP compound, and finally a theoretical design of an industrial scale bioreactor model. That's all. Thanks.
Thank you so much. Okay, I think we're going to move on to the next presentation. So we'll move those questions to the chat. So Yusuf Greati from Designer Cells, are you here?
Okay, hi everyone. I'm Yusuf. I am a pharmacy student. I'm a sophomore, and my project focuses on the computational design of de novo peptide inhibitors for the histone methyltransferase SETD8. It's a bit hard to say. So next slide, please. Okay, so let's begin with some biological context. Inside the nucleus, you pretty much have 2 meters worth of DNA. And in order to fit that into a nucleus that is around 15 micrometers, you have these positively charged proteins called histones. And they form an octamer, and the negatively charged DNA basically wraps around them tightly to form what is known as a nucleosome. And this packaging is essential because without it, we wouldn't be able to fit our DNA inside our cells. And it's very important for transcriptional regulation. And these histones have tails that are not necessarily bound to DNA, and they are able to undergo chemical modifications. Some of these modifications could be either methylation or acetylation. These modifications are central to epigenetic regulation, which is any gene regulation that doesn't alter the DNA sequence. And one group of enzymes that mediate these modifications are the histone lysine methyltransferases. They basically add methyl groups to the histones, and this plays a crucial role in gene silencing and transcriptional regulation and DNA repair. They basically control how tightly the DNA wraps around the histones. And among these, if you look in the bottom left corner, SETD8 is unique because it's the only enzyme that is known to monomethylate lysine 20 on histone H4 in vivo. And it also monomethylates P53, a tumor suppressor, and PCNA. And this enzyme is known to be overexpressed in many different cancer cell lines and tissues. And while several small molecule inhibitors have been developed, most notably UNC 0379, only one potent inhibitor has been reported so far. And that's where my project comes in, where I aim to computationally design de novo high affinity binders. Next slide, please. Next slide, please. Oh, thank you. The workflow begins with obtaining the crystal structure and knowing the active site of SETD8 as well as the sequence from databases. And then I design candidate peptide binders using both structure-based models like RF diffusion and non-structure based and non-diffusion models such as PEP-MLM and PEP-PR-CLIP, which we saw in week 5 of protein design. And after generating peptide sequences, I compare outputs across models, and I see if any of those models have similar results, because that can help in the filtration step. And I'll try to finish up. I narrow it down to about 100 peptides or sequences, and then I express those sequences in an expression vector. I isolate these peptides and I test for their affinity in vitro, and then hopefully move on to in vivo. And so that's basically the methodology for AIM-1. AIM-2 would be to hopefully test those on cell lines. AIM-3 would potentially be using these as a research tool for fundamental research as well as potential therapeutics. And finally, AIM-4, why not target all the methyltransferases? Because they are a lot of them are overexpressed in many diseases. That's it. Thank you.
Thank you so much, Yusuf. Yeah, I really love the project. We don't have that much time, but I just have a quick question. So you mentioned that you do want to actually validate these in the lab. Do you actually have access to a lab?
Actually, no. And I even had problems with computational power. Like I said, I didn't mention that actually. I'm a student, so I'm always running around everywhere, and I tried to use the collabs, but a lot of the time they took ages to run. And so I had connectivity issues, and I actually got in touch with a lab in Turkey through a connection, and I used their supercomputers to run some of these simulations. They are still running, but in order to express and test these peptides, I don't actually have access to that, unfortunately. Maybe I'll send it to a lab and propose it.
Yeah, it could be good. Yeah, I was just wondering because you said you were studying right now, so I didn't know if you actually were going to incorporate that into your school program as well. But yeah, I really love the project, and yeah, looking forward to actually like looking at it in more detail. I think we'll have to move to the next presentation.
Thank you.
So Aryan Chowdhury from BioClub Tokyo.
Hello, am I audible?
Go ahead.
Yeah. Listen. Hello everyone, I'm Aryan Chaudhary, a BTech student from IIT Delhi. I have been into technology since I was very young. So here you will see how my project is also a fusion of computer science and synthetic biology. Next slide, please. Yeah, so currently the issue in silicon-based data storage is like silicon is not that great in terms of durability and the amount of data it can store in a given volume. So my project is basically a website which can convert data like text, images, videos, etc. to DNA sequences, but with a few more bells and whistles. Why DNA, you might ask? Because it has a high data density and durability. Next slide, please. Yeah, so my algorithms pipeline is basically this. It has 5 steps. First step, it does compression. It uses the Huffman encoding algorithm. To compress the data it receives to a smaller size. It then encrypts the data using AES, which is the industry standard. You can provide it a password for this encryption. It does error correction using the Reed-Sullivan algorithms. So this is essential for basically future-proofing the DNA, and in case of mutations, the data will still be recoverable. It then converts the binary data to DNA as shown in the mapping, and then it finally appends the start and end codon. You can see this diagram. Next slide, please. This is the first like homepage of the website. In future, I would like to like currently this is just in silica. So in future, I would like to do this actually in a lab and implement this in living organisms. I would also like to simulate the aging and mutations process. Like currently, if you try to change a codon, if you try to change a nucleotide to something else, it will still give you the correct data because of the error correction. But like we don't know exactly how much data we can corrupt or error exactly, after which it will stop working. And using this, we can build databases, we can build storage modules and things which can be very efficient in storing large amounts of data for archival purposes and all. So yeah, and I believe in future when silicon is outdated, DNA could be a possible solution for storing data. Thank you.
Thanks so much. This is always like a fascinating paradigm to look forward to. Does anyone have any questions? Anyone? Okay, well, I guess we'll move on to the next project, and feel free to reach out to Aryan over the chat. Oh, is that a question?
Megan, just really quick
so
then. Oh yeah, Federico, ask your question. Also, Megan, I sent you some messages in the chat. I hope you didn't.
Okay, thank you.
Oh, sorry.
Federico, ask your question.
Yeah, sure. So basically, I was just wondering, what is the advantage of actually encoding a message into a DNA system? I mean, is this the, is it actually that it takes less space, or is it like harder to decrypt?
Arian, that's a question for you.
Oh, sorry, I couldn't hear you. Can you repeat?
Yeah,
Let's
sure.
do it in the chat. Let's do it in the chat. Federico, ask your question in the chat. We'll move on. Thank you so much.
Oh, sorry.
No, you're doing great. Thank you.
So I guess Ava, are you here?
Yeah, just want to check if my mic's working all right, because I saw there's a note saying that the network is unstable. So
Yeah, I can hear you just fine. Do you want to turn your camera on, or do you not have enough connection?
I guess I
that?
don't have enough bandwidth, unfortunately.
No problem. Yeah, go ahead.
Yeah, so I'm Ava from LifeFabs, and I am a lab biologist by training. Next slide, please. Antibiotic pollution is the leading cause of antimicrobial resistance, a global One Health threat costing 1.5 billion euros every year. 200,000 tons of antibiotics are being consumed by humans and livestock, with up to 139,000 tons being excreted. To give you a scale of the problem, this equals the rate of 3 million people. And current wastewater treatment plants are decades old and designed back in the 19th century. They can only break down less than 30% of the antibiotics. This leads to up to 2.7 million human weight of antibiotics being released to the environment every year, causing ecotoxicity, superbug proliferation, and biodiversity loss. Next slide, please. And my solution to this is an enzyme-based solution to convert antibiotic waste to valuable byproducts, and with highly efficient enzymes with enhanced stability by immobilizing them in membrane bioreactors. And the first aim is doing the rationale protein design in silico and establishing a pipeline for designing, filtering, and ranking enzyme variants, starting with lactase. And my second aim would be validating this in the lab by synthesizing the lactase variants through cell-free methods and also performing biochemical assays to test for efficiency and stability with and without membrane bioreactors. And for my final aim, I aim to produce a range of highly efficient enzymes targeting different antibiotics in wastewater and conducting a pilot in industrial scale wastewater treatment plants. Next slide, please. And this is the pipeline I use for the rationale protein design, which I started with selecting the host and also the enzyme variants, as well as looking into the biochemical pathways and also cofactors and the materials required for the membrane bioreactors. Then I look into the literature to identify the key amino acids for mutations in lactase, as well as analyzing the active site for mutational targets. And currently, I'm still in the process of generating mutant libraries using PyMO mutagenesis plugin. And later on, I'll be doing the filtering and shortlisting, for example, using AlphaFold to see if the variants will affect the folding of the protein, and also doing mutation modeling and also high throughput docking to look at the catalytic efficiency and also substrate binding. So currently I'm looking for a mentorship in protein design, as like that's not really my background, and also computational resources as well. And I would also like to take this opportunity to thank all the speakers and also TAs in HTJA as well as LifeFabs for their support throughout the project. Thank you.
Thanks so much, Ava. Really like your project. So what would you say was one of the biggest challenges that you faced coming up with this?
So I guess one of the biggest challenge is like designing like the biochemical pathway. So for example, like I noticed for lactase, they can degrade a range of antibiotics, like ranging from tetracycline, ciprofloxacin, and etc. And it is like important to look for what kind of products that is valuable after breaking down, so that the waste to value bioconversion makes sense. So for example, ciprofloxacin can be degraded and convert into piperacin, which can be used as like a corrosive inhibitor. So for example, looking into the biochemical pathways is one of the challenging part. And I guess the other part is really because there's a lot of different computational tools that is available, and really finding the right tools and the right method is another like challenging bit.
Thanks for sharing. I think Dominika had a question.
Yes. So with regards to aim 3, the degrading enzymes that you want to expose to the wastewater. So enzymes can be like a bit finicky, and I was curious, how do we ensure that they function properly in contact with wastewater? Because some of them need like very specific condition in order to function.
Yeah. So this is also like one of my later aims, because like it is basically first looking into efficiency and also like stability later on. And by immobilizing enzymes, there's actually like a few ways of doing that. You can either immobilize enzymes, or like you can do encapsulation as well, where we're pretty much like providing more stability, and the range is really depending on like the enzyme variants, because like, for example, for lacase, it can be found in like plants, fungi, bacteria, and they have like different pH range and also temperature range as well. So like finding the correct range of enzymes, because like different wastewater treatment plants have different temperature range and pH range. So finding the correct enzyme variants, and also like maybe potentially can do some like directed evolution as well to see, because we're now not only selecting for efficiency, but also for stability and like different criteria. So yeah, there's actually a few things that we have to consider, as you have mentioned.
Great, thanks so much. I think we'll move on to the next presenter. So Federico Brunello from LifeLab.
Hi there. Yeah, sure. Hi. Thank you very much for this opportunity. So my name is Federico Brunello, and I'm a master's graduate in cellular molecular biotechnology at the University of Trento. I'm currently undertaking the Pharmac Academy post-master specialization program in RNA pharmaceuticals and gene therapy at the University of Naples. And I'm currently undertaking an internship at the Talenton Institute of Genomic Medicine and the Department of Industrial Engineering at the University of Naples. I have a passion for studying biology, and I'd like to thank everybody for this opportunity. And I would like to ideally, keep working on this in the future. So my project involves the design and blueprinting of a microfluidic super evolver chip as an AI-assisted in silico evolution tool exploiting the CRISPR-Cas13 mRNA randomized mutation and downstream cell-free hits system in order to be able to do a parallelized particle evolution for the development of biotherapeutics that are based on monoclonal antibodies and ADCs in order to make them able to escape to basically not be blocked by the escape of the cancer cells. Next slide, please. So the background here, as we know, immunotherapy is utilized quite widely as a fourth-line approach against cancer. However, due to genetic instability, tumors are always able to evade immune response in one way or the other, as they dynamically mutate their epitopes, which are recognized by the monoclonal antibodies and the ADCs, ADS. So tumor homeostasis, as we know, are complex systems which are made by multiple cell types, which are constantly evolving in order to evade the immune system. And unfortunately, this means that after each round of therapy, surviving cells may acquire competitive advantage, and therefore they might not be able to be treated again with the same round of ADCs and monoclonal antibodies. So the vision in order to avoid these problems is to build a parallelized multi-chamber microfluidic chips, which is able to synthesize based on random CRISPR-Cas13 induced mutations of in vitro transcribed sequences generated from biopharmaceutical power tubes, an array of basically systems which are able to adapt to the dynamic evolution of the cancer. Next slide, please. So this is how the system works. So this is basically how the system works. So as the first step, we have either de novo power tube sequence laser printing, dark synthesis, or the utilization of an existing ADC power tube as a template. In this case, you can have like bevacizumab. So we take here its epitope binding site, and we reverse translate it into cDNA. cDNA is then transcribed into RNA, and then the RNA is mutated in a base mode by the CRISPR-Cas13. This mutated RNA is associated with the mRNA of a GFP protein, which in turn is downstream coupled to a qubit-associated immune cell. This immune cell is basically bearing both the green and the red fluorescent parts. And basically, you put them into chamber 5. And in chamber 5, you basically are able to find out which cancer cells have responded to this newly mutated, de novo mutated therapy. So by then fact sorting the cells in order to be able to find out which cells have been able to be bound by the antibody, you're able then to find out an evolved therapeutics which hasn't responded successfully to an inherent mutation. As we said before, tumoral cells are inherently unstable, and therefore we must be able to find a continuous evolutionary way in order to tackle the evolution of the cancer mass. So this is a way to basically continuously generate evolved monoclonal antibodies and ADCs that are able to dynamically adapt to the mutations that cancer uses in order to escape our ADC and monoclonal antibody treatments. So the first aim of the project is to basically design the macrofluidic chips, and to test its hits in vitro translation. And we also want to assess the quantum dots and the feasibility. Aim 2 would be to generate a library of paratopes from de novo predicted sequences DNA, and to successfully generate quantum dots bearing antibodies and GFP paratope assembly. And then the aim 3 would be the final integration of this system, this loop, so that instead of basing our design on existing paratopes and evolving them progressively in a random way in order to tackle mutations, we are able to feed these successful hits into an AI system, which then might be able to allow us to generate de novo sequences based on Cambrian Genomics direct printing and AI.
Great. Federico,
So,
I think we're running
yeah.
over time now. Do you have any more?
So that's the, just this last slide, basically, the conclusion.
Perfect. Okay. So, do you have any more final points or?
Yeah, this last slide, yeah.
We got to move on for 20 seconds over.
Yeah, I think we're
So
pretty
these are
far.
future directions. Yeah.
Just one last sentence, and then I think
Future direction is to basically be able to build this chip and then have it tested so that it can be used in a diagnostic tool and also an artificial evolving tool to basically predict and follow the inherent mutations of cancer by evolving artificial and dynamically artificial ADCs and monoclonal antibodies for different cancer types.
Perfect. Thank you so much, Federico. Unfortunately, we're actually running over time. So if
No
we could
problem.
actually put the questions into the chat, I'm sure there's a lot of people who have some.
Maybe it wasn't really, it was quite a bit complicated.
We got to go, Federico. So sorry. Onto the chat. So sorry. We'll continue the discussion in the chat. Thank you.
Thank you. So Richard Machoka from Life Labs.
Yes. Can you hear me?
Yes.
Great. So my name is Richard from Nairobi, Kenya. My project is on Dendrospis polylepis snake venom diagnostics using cell-free system. Next slide, please. So one of the biggest takeaways from this course is that you should be able to take synthetic biology and apply it to what is the problem locally. Have we moved to the next slide? Maybe it's mine that is taking long. Yeah, so the problem that we're trying to address is snakebite, which causes about 81 to about 138 deaths globally, which is a huge underreporting of the numbers because the numbers are a bit higher than that, and this is with about 300 to 400,000 permanent disabilities. So the reason why I picked this specific project is because this snakebite disease affects the most marginalized communities in the rural areas in Africa and the poorest of them who are not able to access medical services for them to do that. So what you see in the middle is the composition of the snake, the venom composition of the black mamba, the Dendrospis polylepis, and the yellow one is the commit type proteins, which then is the largest amount of proteins which the snake uses to actually mobilize victims by causing paralysis by interfering with the channels, the ion channels. So the general aims of this project was the first one to develop a cell-free system that can be able to identify these specific venoms that are specific to this snake as a model for the others, and then use the cell-free system to be able to use to identify those. Then the second one was to develop a panel of peptides that bind and degrade snake venom protein components, and this was highly inspired by the talk that was given by Professor Pranam, because it was possible from the discussion there that we are actually able to degrade some of these proteins. And then we should also be able to evaluate the effectiveness of these neutralizing effects peptides as potential treatment for snakebite environment. The reason for this is because current methods of treatment of snakebite is you're using antivenom, which is a very long process that involves injecting animals with venom and then harvesting the antivenoms, purifying, and then which takes more than 6 months to even a year to be able to produce, which is a very long process, and we require an easier using, especially the various methods that we have been taught in this class to be able to deal with this problem. Next slide, please. So therefore with the problem of little computational power with where I am, we're able to just use PepMLM to be able to develop a peptide that actually bind to dendrotoxin. Dendrotoxin is a specific type of Kunitz type reagent that works on that is for this specific snake, and we're able to develop that. And these are the results of the attachments of those. And then when you look at the next slide, so we will be able to take these attachments and then put them on a cell-free system. Next slide, please. So that we can be able to then combine them in a luciferase reaction to be able to emit light. So the next step involves.
I just want to quickly just so we're mindful of time. We just have a little bit of time left, so maybe just give your final point, and then so we can have a question.
Okay, yeah. So the next point will involve validating this work experimentally. Then we repeat this for the various snakes. For the importance of diagnosis, because antivenoms actually given to snake to victims based on the species of snakes, and then to develop this as a potential therapeutics for snakebite victims across the country. Thank you. I think I can take some questions.
Thanks so much, Richard. We would have time for, I guess, a really quick question, if anyone has one.
Yeah, quick question. The synthetic peptide system with the light emission, is this something that's from the literature, or is this something that you've designed?
So yes, I got it from the literature. The luciferin C and S, which when combined together, they actually emit light. But now we up to when they bind to the target, then they can be able to emit light as a way of being acting as a signal.
Great, thank you.
Yeah.
Thank you so much. Next up is Mehmet.
Yes. Good afternoon. It's Mehmet. My project is a dual trigger CRISPR kill switch and Van-Modulin system for rare earth element recovery from e-waste. Next slide, please. The problem is globally less than 1% of rare earth elements like neodymium and dysprosium are recycled from e-waste. The challenge is developing an efficient and selective extraction method using biotechnology. My solution can be summarized in 3-step process: dismantling, bioleaching, and separating. So dismantling part is a physical pretreatment. Bioleaching part contains engineered A ferrooxidants with a CRISPR kill switch. This bacteria produces sulfuric acid to dissolve metals. I will explain it in the next slide. The separation part facilitated by Van-Modulin protein. So this protein binds the rare earth elements at pH 5 and release it at pH 2. My works done are increasing binding affinity with 4p2a mutation, fusion with isonucleation protein for surface display. A gene fragment was ordered from Twist Bio, and freeze-dried whole cell bioabsorption for separation. Next slide, please. For aim number 1, the gene fragment was designed and ordered. Cloning work will be done using shown in the figure with BMH1 and the Him3 enzymes. Final plasmid will be transferred into E. coli BL21. Bacteria will be cultured, then centrifuged, and the pellet part will be lyophilized. So the binding affinity for lanthanide metal will be measured by colorimetric assay. A special dye called alizarin changes color from orange to purple when rare earth elements are bounded. For aim number 2, I designed a CRISPR kill switch for A ferrooxidants. So the first cassette contained a p-ATP2 promoter, which is triggered when the pH is more than 5 and transcribe a TET-R protein. The second cassette contained a peak hold promoter, which is triggered when the temperature is below room temperature and transcribed like I protein. If both conditions are set at the same time, then AND gate works and the third cassette is initiated. This one transcribes guide RNA and the Cas9 protein. So the guide RNA targets the FTSZ gene, which is a vital protein for cell division, and the mutation caused by CRISPR-Cas9 kills the cells. Next slide, please. For validation purpose, I prepared a table showing various pH and temperature conditions and its expected outcomes. For aim number 3, I would like to replace a conventional rare earth recycling with fully biological closed-loop systems. I also want to demonstrate the scalable integration of biocontainment, extremophile engineering, and protein design. Just lastly, the biosafety consideration. Even if I pay attention to biosafety, there are more questions I have in mind, like how reliable does the kill switch? So what happens when the mutation disables kill switch or acid or temperature sensitivity? And I also thank you for your time and attention. I truly appreciate the opportunity to share my project. I'm happy to take any questions you might have.
Thanks so much, Mehmet. Really fascinating presentation. Does anyone have any questions? If not, I just want to ask you, how did you come up with the idea? It's a really fascinating one.
Yeah, actually, I just read a couple of papers recently published about the land modulin, and the CRISPR kill switch also interesting for me. Yeah.
Yeah, I'm really looking forward to seeing if you continue working on this project, what comes of it.
Mm-hmm.
Really
So
exciting.
lastly, I just thank you for the Life Labs for the opportunity.
Okay, so if anyone has any more questions, feel free to reach out to Mehmet in the chat. And I think we're going to move on to our next presentation so we can keep moving because we still have, I guess, more than 2 hours of presentations. So Escalalita, are you here?
Yeah, okay. Am I audible? Sorry.
Go ahead.
Yes. Is my background so disturbing?
I can hear you okay.
Okay, very good. Okay, hello guys. My name is Eska. I'm from Indonesia. Good early morning from Indonesia. It's very early. And it's my first time to learn about synthetic biology. So I would like to present about engineered plant growth promoting results for bacteria in saline soil. Okay, next please. Yeah, saline soils currently threaten more than 20% of the world's agricultural land, posing a major challenge to global food security. High salt concentration disrupt water uptake in plants by creating osmotic stress and significantly inhibiting growth. However, nature offers promising, it's called rhizosphere bacteria. These microbes, especially producing SEC deaminase, which can support plant growth under stress by modulating ethylene levels. Unfortunately, their effectiveness is often limited by the specificity of plant-bacteria interactions. And this project aims to go a step further by engineering Bacillus subtilis to express SEC deaminase and designing a synthetic gene circuit that can respond to the saline conditions, being adaptable to a wider range of plant hosts. Okay, next slide. Yeah, to achieve this, I will design a plasmid called PSALT-X PR5, which I know is a novel plasmid and I will optimize it via benchling and GenScript tools. The engineered genetic circuit integrates with multiple parameters responsive to salt and environmental signals, especially root exudase from plants, which I need several gene components then related to root exudase components you can see on my slide. And I designed it through a logic gate, which is AND gate. And I assemble it using Golden Gate assembly, which I got it from a course. Then I plan to using Bacillus subtilis as a chassis, which is a well-known grass organism. And the key component is SCDS gene, which will be produced SEC deaminase gene, SCC deaminase, the product, and will be validated through mass spectrometry to confirm SCC deaminase activity. And okay, the next slide. Yeah, for the future outlooks, looking ahead, this innovation has the potential to be developed into bioinoculant formulation, beneficial, which is it is beneficial microbes applied to soil or seeds to support plant growth, and ultimately, this approach contributes to climate resilient agriculture, reinforcing food security, reducing poverty, and aligning with global sustainable development goals by harnessing synthetic biology and plant microbes interaction, which we can cultivate not just plants but a more resilient future for our planet. Okay, thank you.
Thank you so much. Does anyone have any questions for Escalita?
Yeah,
David.
so
Yes.
great work. Yeah, so great work. Plants are notoriously slow to work with in engineering. They're obviously they grow much more slowly, and or they're just different challenges to engineer them versus like microorganisms. So I'm curious, number one, do you have thoughts on potentially being able to experimentally test these your genetic engineering someplace? Like, do you have a lab or a place you can do some of that work? And then also, are there ways that you've thought about how to actually work with genetically engineered plants in the lab?
Yeah, actually, now I haven't pursuing my master degree yet. Probably if I will enroll on my master degree, probably I will have a lab to do it. And sorry about the second question, I didn't get it.
In working with plants for doing genetic engineering, they're just challenging to work with because the design time is very slow. So I was wondering if you had approaches and thinking about how to optimize your genetic engineering with plants.
Yeah, actually, I just want to not. It's actually not engineering with plant, but I try to engineer with bacteria. Did you get? I mean, my idea. Yeah, and this engineered with bacteria I will put it on rhizosphere. I mean the in the rhizosphere, and then the bacteria will have communicate with the plant bacteria and responding the salt stress situation on plant. Okay, probably that's all. Yeah,
Perfect,
probably I can answer it on the chat probably for the further answer.
Perfect. Yeah, let's move on. Thank you so much.
Yeah, that sounds like a great idea. Thank you. So next up is Yusuf. Actually, I think maybe this was put in a different place in the slide deck. Yusuf, are you here? I think maybe we'll move on actually to the next one. So it looks like it's something. So
Can you hear me?
yeah, I can hear
Yusuf
you.
is not there.
Okay, yeah. So we'll move on to is it Toma?
Yes, yes.
Okay, perfect. Go ahead.
Yeah, we can begin. So next slide, please. I'd assume that all of you have seen or heard about the news coming out of plastics in humans, especially biomedical news. It had been found that brain plaques in symptomatic patients contain 51 times more plastic particles, or that E. coli forms more drug-resistant biofilms on microplastics than on glass. And also, plastic was found.
So sorry to interrupt, but your volume is very quiet. It's really hard for us to hear you. Is there a way to get closer to the microphone, or?
Yeah, I'll do it. Yeah.
Yeah,
It has
it's still-
also been found that microplastics reduce plant photosynthesis by up to 1.14 times, and countries with high microplastic levels have higher cognitive disability rates. Now, to the next slide, we can see that I'm trying to come up with a solution known as shy plastic. Thankfully, Dr. Bertoncini's lab has found special enzymes in the saliva of Galleria melonella, specifically Ceres and Demetris. And compared to other enzymes, these are the first known animal enzymes capable of degrading polyethylene without needing pre-treatment. and this greatly improves degradation strategies. On the next slide, we can see what I did about this project. Now, while we don't exactly know how does Ceres and Demetra degrade the plastic so well, we know that it happens through the oxidation process. For oxidation, it has not been fully elucidated, but they have found repeating patterns within the enzymes that are similar to that of hemocyanins. Now, the way hemocyanins perform redox reactions is through their copper core. And while copper core is good for redox reactions, we've seen that through evolution, iron is more redox active. So to the next slide, we can see what I try to do. So there are other proteins that also contain iron, and these proteins have different roles in the bacteria, for example. I took the one known as NTRC from Shevanella oneidensis, and I targeted the amino acids that are able to capture the heme. Now here on the right-hand side with a lot of labels on it, you can see the amino acids that are in 5 angstrom to the heme. And I basically took these amino acids and I implanted it inside the region where putative hemocyanin-like copper-binding amino acids are supposed to be in the Demetris. I also added a special signal peptide, which is meant to simplify the purification so that Adrian in Ottawa can purify it through centrifugation. And of course, added a fluorescent tag for visualization. So that's all my presentation. Thank you for listening.
Thank you so much. So yeah, I really like the idea, and a lot of the computational work is fantastic. So as far as like validating this, what have you thought about that at all? And if I'm not sure.
So the plasmid had been ordered from Twist, and Adrian, my group lead, is waiting for it. And once it arrives, it has everything to be tested. So he can directly purify it by centrifugation because it has a signal peptide that throws the protein outside the membrane. So he can just centrifuge and take the lysate, take the supernatant. And basically, it's then all he has to do is put the sample on the plastic and check if it works and if it's better, and measure the time, how fast the degradation occurs.
Perfect. Yeah, I'm really looking forward to seeing what the results will be for that. Yeah, it'd be great if you keep the class posted on it. Any other questions? I think we have time for one more.
Keep it moving.
Okay, yeah, we can keep moving. We were actually somehow a little bit ahead of the schedule because we skipped one, but we will keep moving on to Falasewa. Are you here from Life Labs? Falasewa? I'm not sure if I'm pronouncing that correctly.
Hi.
Hello. Yeah, so go ahead.
Hello, everyone. My name is Falashawa, representing Life Labs. I will be talking about engineering HLA-G protein for enhanced immune tolerance and reduced graft rejection. Can you move to the next slide? Okay, so according to research, about 40% of graft failures result from acute rejection of grafts. For successful transplants, organ transplant recipients are forced to a lifelong use of immunosuppressive drugs. So this poses a significant challenge on how we can reduce graft rejection and also the over-reliance on immunosuppressive drugs. In a pregnant woman, the fetus is technically a foreign object. However, there is no maternal fetal rejection due to the natural function of the placenta. This leads us to a question that is it possible to engineer a membrane that is inspired by the immunomodulatory function of the placenta and that can allow the transplanted organ to not be seen as a foreign object, just like the fetus in the womb. This leads us to the protein called the HLA-G, which according to the research paper, an increased expression of this protein in lung transplant recipients correlates with better graft acceptance. Therefore, my research question is, can engineering HLA-G protein variants enhance immune tolerance and reduce graft rejection? Next slide, please. So my first aim now is to get a finalized protein design candidate that meets the desired criteria for immunomodulatory function. So this is my experimental workflow. I started with literature review, then I went on to isoform selection. There are 7 major isoforms of HLA-G, but from the recent research paper, there are 2 that I can work with for this research. So I chose the HLA-G1 because of the fact that it binds to key inhibitory receptors involved in immune responses. Then I went on to identify some single nucleotide polymorphism, and this is to ensure that to guide me in selecting my HLA-G variants. There were no known SNPs in the database that were both missense variants and also clinically significant. So I had to make use of some criteria to select 5 SNPs and then made use of the PIMO to check the structural model and to see the potential receptor binding points. Currently at the point of using the SNPs to get my HLA-G variants and then make use of the Rosetta and some other molecular docking platforms. Next slide, please. My aim 2 and aim 3 is if I've gotten my top 3 protein candidates, I would check for in vitro experimentation to confirm if my desired protein can modulate immune responses. These are my top 3 focus areas to check for immune activation, to check if the protein is stable, and to also check if it's functional. And the final aim is now to initiate the multi-stage clinical trials, and this is when we'll be making use of rats, because according to the last paper I read, they made use of rats to check if it's possible. And the future vision is to check for strategic partnerships, funding pathways, and some other things. Thank you.
Thank you so much. This is really an incredible project. Does anybody, okay, Dominica, go ahead.
Oh, my apologies. I actually meant to have the clap emoji. But now that we are talking about it, is there any way that you can put this into like a wet lab experiment?
Yes,
or
that's my aim too. I feel because I'm not I don't have access to a wet lab here, so I just want to try to see if I can make use of computational tools to check my top 3 protein candidates. And then if I have access to a wet lab, then I can move forward with it by culturing some systems that can impact HLA-G on effector cells. I want to thank Digby for helping me to review my slides and also my mentor for giving me the opportunity to present today. Thank you.
Thank you so much. Yeah, really looking forward to seeing how this evolves. Any more questions? If not, feel free to reach out to her over the chat and we can move on to the next presenter. Berenice, are you here from Ottawa Bioscience?
Can you hear me?
Okay, yeah, the stage is yours.
Okay, so my project is TDT, or terminal deoxynucleotide transferase, or mRNA analog synthesis. The next one, please.
I'm so sorry. Can everybody make sure that their microphone is muted? Okay, go ahead, Bernice.
Well, my project is TDT, or terminal deoxynucleotide transferase, for mRNA analog synthesis. And you change to the next slide, please. Well, currently, clinical DNA synthesis is centralized by the bigger companies, and it represents high costs depending on the length of your DNA sequence. Because the longer it is, the more cycles and the more reagents are needed. And also you need to preserve a cold chain. So it makes these assays restricted to laboratories with the required specifications. But on the other hand, we have enzymatic synthesis that is performing aqueous solution, which results in a greener process since it needs less activators, oxidants, and it also can be performed in situ. And this allows us to perform this DNA synthesis in places with limited resources. And well, based on that, we found TDT, or that stands for terminal deoxynucleotide transferase, which is a DNA polymerase enzyme. And it has the particularity of indiscriminately adding DNTP to the 3' hydroxyl group of a single-stranded DNA creating material de novo. Can you change to the next slide, please? Well, based on this mechanism of action, we can use this TDT for the controlled addition of the entities to come up with the desired DNA sequence that we want to create. And well, my aims, the first one is to express this protein based on the bibliography that I found in the vector PET28A, and will let it be expressed on E. coli. And the second aim is to synthesize the primers depending on the therapeutic target that you want to use them. The TDT, for the TDT to start synthesizing these entities, it needs at least a primer with 3 nucleotides long. And my third aim, well, is to create this mRNA analog synthesis. And once we have our primer, we can start adding the DNTP in order analogs, and they can bind to a target mRNA, to target mRNA in which it can block the function of, for example, oncogenic microRNA. And the next slide, please. Well, this is the vector with the new TDT gene that we ordered to twist. And we can also observe that we added the CAR9 patch, which confers the ability for the protein to adhere to glass or plastic surface. And well, this was added because in the future, maybe we can perform these microfluidic assays in which we adhere the protein and start the solution, the DNTP solution, and start constructing our oligo sequence. Thank you.
Thank you so much. Does anyone have any questions?
Yeah, one question about the microfluidics that you proposed. Can you talk a little bit more about why microfluidics for this application? Like, what is the advantage of using microfluidics here?
As I described, like in the comparison between chemical and enzymatic DNA synthesis, the enzymatic synthesis can be done in situ, and you can move it like wherever you are. You can do the experiments there, and you don't need a lot of chemicals. So that's why I chose microfluidics, and also because we can recycle the DNTP pools. Like you can use, I don't know, maybe a pool with a DNTPs, and you can recycle along the microfluidic device and start creating bridging. That's why I chose the microfluidic device.
Great, thank you.
Thank you. So next up is Anandita from BioClub Tokyo.
Okay, this is my audible. Okay, thank you for the chance. My name is Anandita Amalia, and I'm from Gadjah Mada University. I will present about structural instability of HBB gene mutation. Maybe you can next slide. Okay, we started from hemoglobin. Hemoglobin is protein found in the red blood cell that carries oxygen, and then it's and has several genes. It's alpha genes and also beta genes. And the mutation in the genes, in particular beta genes, can cause several diseases, sickle cell disease, and then also beta thalassemia. And the dominant types of the mutation in beta thalassemia include missense mutations, insertion and deletion, and also frame shift, and so on. And in Indonesia, the beta thalassemia also had prevalence because 3 until 10% of the population carries the beta thalassemia genes. So the aims of the final project is understand the mutation of beta thalassemia and understand the structural genes and develop the experimental design in the beta thalassemia. Maybe you can next slide. Okay, so this is the methods and result. First is a collect high HBB gene mutation using ClinVar, and this is a 7 missense mutation. But I think there's several more missense mutation in beta thalassemia. And then we predict with the several tools. First is using MCSM, predict the mutation effect, and based on the delta free free energy, the result that protein number 127 is high destabilized, and then predict again with using deep sequencing by using mutation tester. And based on this result, that protein feature like genes domain globin helix is probably lost, and then structural and functional impact of amino acid substitution also by using polypen2 is probably damaging. But in the computational prediction of chemical and also physical parameter of the protein based on param from valine and glycine at that sequence is that instability index is decreased to the glycine, and then but in the structural prediction by using AlphaFold, it's an insignificant difference. And then the next slide. So the validation, another validation is superposition of protein by using TMA element is the file 127, and this is superposition, and the TM score is 0.99, it's almost 1 bit. Then the result is that both of the protein has the same fold, and then the experimental find is a gene therapy by using CRISPR. And so because of this is a gene therapy, the bioethical consideration is safety and unintended consequence, informed consent, equity and access, regulatory oversight, and also therapy and versus enhancements, public perceptions. And thank you for all global data and committed listener for the hard work. And maybe this is my presentation. Thank you.
Thank you. Great presentation. You use such a variety of computational tools, and this is a really good example of like really robust analysis. Anyone have any questions? Otherwise, I have one.
Maybe just again, really great work. Very impressive. All of the effort. Maybe you can say a little bit more about the computational approaches that you took, and what did you find challenging? What were the parts that you're most excited about and worked well?
Okay, first is the computational. I using several computational because they have several different results. First, the protein param for chemical, and also the polypen to resolve that the damaging of these substitutions, and also mutation tester. I can see the probably result of this mutation, and then for the TM alignment, I can see the superposition, how much they different to the others. Yes, I think.
Great, thank you so much.
I think we'll move on to the next presenter. So yeah, feel free to reach out to Ananita over the chat. So Anahi from SynBioGenetics. Anahi, are you here? Otherwise, maybe we'll move on and we can come back. Okay, so Maria Jose Mendez.
Yes,
Also
I'm
from
here.
Okay, perfect. Go ahead.
Thank you. Can you go to the next slide, please? Okay. Ever wonder why your mom always made you eat your grains? Turns out she was onto something. Vegetable, fruits, and whole grains are rich in fiber that fuel your gut microbes. When these microbes ferment fiber, they produce short-chain fatty acids, and among them is butyrate, that is a true health superstar. This strengthens the intestinal barrier, reduces inflammation, supports immunity, and even helps regulate metabolism. So how can we deliver the benefits of butyrate without depending on diets people don't actually follow? My name is Majo from Cali, Colombia, and this is the motivation behind my project. My goal is to create butyrate, a safe, flavorful probiotic seasoning for daily use using a genetically engineered strain of Saccharomyces boulardii that produces butyrate directly in the gut. Can you move to the next slide, please? The heart of my work is aim number 1, which here I design, build, and test the strain using CRISPR-Cas9 genome editing. I will start telling you like the steps I will follow to create this aim. The first is to design an operon with 5 enzymes needed to for the butyrate synthesis, a constitutive promoter, self-cleaving peptides, and a terminator and an ERA marker, along with the CRISPR-Cas9 plasmid targeting the HO locus. Then order clonal genes to twist into fragments, maybe 2 or 3 fragments, that will then be assembled all together in the expression cassette using Gibson assembly. Then I will transform the S. boulardii using electroporation or lithium acetate and introducing the linear donor and the CRISPR-Cas9 plasmid. Next, I will select the transformants with the ERA marker and verify the genome integration through PCR. To confirm that everything is correct, I will sequence the genome using the next generation sequencing. After all of that, I will do like fermentations, like a wild type versus the modified yeast to see how the genomes, the genes are operating in the strain, and to further optimize the strain. It could be like any operon, promoter, or even the enzymes. Finally, I will do the in vivo test using mouse models. Here, the goal is to administer the strain orally and quantify the butyrate in feces using liquid chromatography. And here, I would like to analyze like the beneficial metabolites like acetate, propionate, and other secondary bile acids. Can you go to the next slide, please? My aim number 2 focuses on developing a stable lyophilized seasoning. Here, I will test the strain survival through freeze drying and its activity after rehydration and its compatibility with typical seasoning ingredients, because my aim is that it is tasty. And my aim number 3 is to explore safety, consumer perception, and regulatory feasibility. Here, I will conduct further in vivo studies to assess any long-term effects, evaluate consumer response to the product, and map the pathway towards a food-grade approval. Now, to sum up, my ultimate goal is to bring synthetic biology to kitchen, creating a probiotic product that is delicious, safe, and delivers the clinically proven benefits of butyrate in a form people can enjoy every day. Thank you very much.
Thank you so much, Maria. I love it. What's not to love about this project? Does anybody have any questions off the bat?
Yeah, so just I love this project. Short chain fatty acids are near and dear to my heart from a project I worked on many years ago. Can you talk a little bit about kind of the amounts of production that you would need or want of butyrate? And then you mentioned also a couple of the other short chain fatty acids. I think you mentioned like propionate and some of the other ones. Can you talk a little bit about your approach to try to make some other short chain fatty acids that may also be nutritionally interesting?
Yeah, okay, so I may not make myself clear. The idea is to only produce butyrate, but when this strain that is beneficial for your gut is present, it can increase like the feasibility of other strains that produce these other short chain fatty acids. So in the papers I took as reference, they did some of similar procedure, and they found out that when introducing another molecule that is 3-HB, these other short chain fatty acids increase. I cannot remember right now, like the threshold that the butyrate has to be in the gut, but for sure it will be like something to test, that it will be beneficial for your gut, because if it is not in the correct concentration, it will not be like beneficial at all.
Thank you.
Thank you.
Thanks, Maria. So next up is Mariana from Ottawa Bioscience. Mariana, are you here? We're actually running a little bit early right now, so.
Shocking.
Yeah. Okay,
So
I
we
will
missed
move on.
Mariana. There's one other presentation that I'm not sure TAs if we remember. There's one other person we skipped. I'm trying to remember who that person was. Did we track that? Make sure. they
Yeah.
come back.
I think it's actually a high. Yeah, so right
Okay.
before Maria. So from biogenetics, yeah, so I'll keep track of that. So if Mariana's not here, we'll also move on. We might even want to think about, I don't know, taking a break early though if we start skipping a lot of them, but we can check in about that so that we don't get too far ahead because then like people might not be on the call
Yeah,
yet.
we're gonna take a break until 6, so in like half an hour. So let's keep
Okay.
going until we get to that point.
Sounds good. Okay, so Kelly Zunika.
Yes,
Are
I'm
you
here. Yes.
Great.
So, did you know that the deadliest toxin known to science is also the same substance people inject into their faces to smooth wrinkles? Botulinum toxin A is so potent that just 1 gram could kill over 1 million of people. Yet, it is daily used in science and medicine. Hi, my name is Kelly, and I'm going to present my project. Next slide, please. Botulinum toxin A produced by Clostridium botulinum is one of the most potent neurotoxin, causing paralysis by blocking the acetylcholine release at the neuromuscular junction. According to the CDC in 2019, toxin A was the cause of 33, 90, and 68% of infant, gout, and football botulism cases, respectively. Current antitoxin treatment rely on egg-derived antibodies, which has some limitations such as high production costs, immunogenicity risk, limited tissue penetration, and dependence on cold chain logistics. That's why in this project, using bioinformatic tools, inhibitory peptides with high binding affinity to this toxin can be designed and also evaluated in silico, in vitro, and also in vivo to demonstrate their therapeutic potential, low cost, and accessibility for this type of botulism. Next slide, please. So for this part of these aims was to design and engineer inhibitory peptides. So for this, I use PEMLM to design the peptides. I also use AlphaFold server to see the interaction between the toxin and the peptide. And I also use the score to select the best candidates. So from the 29 peptides analyzed, I choose the best one, which I call PepKC. So here is my genetic construct, and also here is my insert, which has the sequence that is going to codify for the inhibitory peptide, and also the resulting plasmid after the enzymatic digestion assay. So for the second part of this, it was optimize the genetic engineering and expression. So codon optimization was performed in order to express this peptide in E. coli. So the next steps could be the purification of the peptide. That's why I select NEA protein purification and also the protein cleavage because my insert has these parts. So for the last part of this aim was evaluate efficacy and stability. So for this, in vitro binding assays and also functional inhibition assays would be performed in order to have a peptide with good binding, good percentage of binding specificity, stability, good percentage of inhibition, and also stability of inhibition, etc. Next slide, please. So the second aim was to evaluate in vivo efficacy. So for that, I designed this experiment to be done in mouse models. And the third aim, it would be the delivery of the peptide efficiently. That's why I designed this tool that I call Botate to deliver this peptide intravenously. My project also has some bioethical considerations, such as this toxin is extremely potent, so strict biosafety is needed. Also, I need a license and approvals to do that, reduce animal use in research, and also promote social responsibility and equitable healthcare benefits. So for more visionary aims, I would like to optimize these peptides, making a cocktail, scaling up the production, extend to the other botulinum toxin serotypes. And my message for this project is that making inhibitory peptides to this toxin could be life-saving and in the future the best option to treat this type of botulism. Thank you.
Thank you. Really fantastic presentation. Does anybody have any questions? So what are the major challenges that you, oh, hold on. Actually, yeah, let's go to Ben because I've been talking a lot.
Great. Thanks so much, Megan. Thanks, Kelly. Amazing presentation. Just want to ask you about your delivery system. Do you know maybe how much of your peptide you should be putting in the needle or in the device that you just proposed to create?
No, because first I have to do the experiments and see which is the best amount of solution that I needed. But I think that when I do the experiments, I'm gonna have the real amount of peptides that I can use to do that.
Okay, great. Thanks so much.
Thank you.
Any other questions? So actually what, here, Patrick, go ahead.
Really cool idea, and I like that it has such a wide impact. I was wondering if peptides made from bacteria or from other organisms like yeast or mammalian cells have different glycosylation, which might affect binding to botulism toxin.
Yes, the first idea is to make that in E. coli because this toxin is like an exotoxin. So I think that if I produce a peptide just in E. coli, it could be enough to get the objective.
Do you know if they have different glycosylation patterns?
No, about the patterns of what?
Of glycosylation,
No,
like
because
little sugars that are attached to, yeah.
yes, everything of all of this is just like an idea. So I need to do the experiments of all to try to do it.
Okay, thank you.
Thanks again. So I think we're going to move on to the next presentation. So just feel free to reach out over the chat. So Aparajita, are you here?
Yeah, I am. Can you hear me?
Okay. Yeah, I can hear you. Go ahead.
So hello everyone, I'm Aparajita and I'm doing my first year MBBS from KMC Manipal, India. My project Bioscribe, can we move on to the next slide, please? Yeah, so my project Bioscribe is about revolutionizing sustainable design by creating a self-recharging, biologically powered ink pen, enabling continuous color production so that the bacteria keeps multiplying. Almost, it's like an infinity pen. This can help reducing waste and reliance on synthetic chemicals. So it's like a natural glowing ink made by genetically modified bacteria like E. coli to produce GFP, which is green fluorescent protein. So usually GFP makes the jellyfish glow, but now we're making it to have the bacteria glow. Next slide, please. So for an overview, I took a PG yellow plasmid for the vector. I chose this because it naturally has a GFP UV gene. So it's easier for it to produce and express fluorescence when it's genetically modified. Using Benchlink, I integrated the GFP insert in the PG yellow vector. This can be inserted in the E. coli in a wet lab by first making the bacteria competent and then giving a heat shock. After our bacteria is ready, we fill this in a pen cartridge along with cultivating media. Now this cultivating medium, it has LB broth for promoting faster growth, glucose for energy needs, and to ensure that the ink isn't toxic to the environment, we can use a non-pathogenic strain of E. coli like DH5 alpha, and also we can modify the bacteria to be auxotrophic so that it doesn't multiply outside the medium. Now, yes, proteins degrade, but in my formulation, I plan to use cross-linkers and additives like glycerol or silicone nanoparticles. And I can also encapsulate my ink to slow down the heat or air exposure impact. This keeps the ink from fading and makes Bioscribe useful. Next slide, please. For my project, I used GFP, but in the nature, several colors like RFP, YFP, CFP are also found. I'd compared the structure of GFP and RFP chromophore on PyMol, and the beta barrel structure was pretty similar, with the central structure producing the fluorescence. Now, my project works under UV, but we can also use that proteins to work under visible light. For example, AE blue or AML-CP that can be used to create colored pens in visible light. Next, I don't want to limit it just to ink pens. I can also use it in 3D printers to form live organism models. And lastly, we can integrate it with IoT using systems like Raspberry Pi, which can be used to detect signals and log toxins in the environment. An interesting paradox like that I would like to mention in my project, it may seem like I'm replacing one synthetic, that is ink, with another synthetic, that's bacteria, but the key difference here is control and sustainability. My engineered bacteria are biodegradable, unlike the petrochemical inks that persist in the environment. So my BioScribe doesn't leave a toxic legacy and can be tailored for utility and smart functions like sensing. That's it. Thank you.
Thank you so much. Really, really cool idea. Anyone have any questions? Otherwise, I'd like to ask more about this 3D printing idea. If you could just like talk about it just a little bit more detail.
We could code how the printer works, and instead of the normal pen cartridge, we could use this formulation instead of the normal ink. And that having already the bacteria, which is live, it can create live models, not just stationary models, maybe.
Yeah, I think that would be a really cool use case in addition to it. There's a lot of potential.
Thank you so much.
Any other questions? Really creative. I really hope you actually go through with this. It would be really exciting to see this come to market. I think we can move on to the next one. Damaris. Damaris, are you on the line? Okay, so okay, perfect. Go ahead.
I have to share my screen. Give me a second.
Actually, I should still be sharing my screen. Can everyone see it? I can just
Yeah,
if you just let me
Yeah.
know when to advance to the next slide, I can do that. Oh, do you
Okay.
mean share your camera?
No, it's fine. It's fine. Yeah. Okay,
Oh.
then.
Yeah, but feel free to show have your camera on too if you have a strong enough connection. I think there may have been a misunderstanding.
I'm trying, but I can't connect my video. I don't know what.
Don't worry about it. Yeah. Okay.
I mean,
So-
I'm pressing the button, but there's no, I can see myself, but I'm
It's
pressing
okay.
the
I
button.
think we can just go ahead with the audio. So stage is
Okay.
yours.
Okay, thank you. Hi, my name is Amari Hanchoso, and my presentation is based on in vivo monitoring of hydrocarbons contaminations in a coastal ecosystem. Next, please. So this image you're seeing on the left, this happened just on March 15, 2025 in Ecuador. So there was an a damage to an oil pipeline caused by a landslide. This contaminated the river in the Ecuadorian province of Esmeraldas. Oil spills in Ecuador, at least, are not isolated or recent events. They have been occurring for decades due to corporate malpractice. So just to have an idea, on average, there are 22.5 spills per month. And 97% of these spills happen because of the Ecuadorian state-owned companies. So there's in the Constitution an article that established the obligation to restore ecosystems and compensate affected individuals. However, there are no official reports or documents published on governance companies that where the contamination and remediation actions can be verified. So the protocol when this happens is that they send satellite images, and aerial images can help identify the presence of oils in the surface. But there have been limitations in determining the thickness of the layer, maybe the surface contaminations, or the presence of dissolved hydrocarbons. So again, not all contamination is visible to the surface. So my idea relies in the creation of a biosensor module powered by bioluminescent bacteria, engineering bacteria, with intent of monitoring and mapping these environmental hazards. So this is just an AI generate image of how this could look, but my objective with this is to go from the root, and maybe I can be able to help with this process, which is the first part in how to fix it, if that's my idea. Next, please. So this is a 3-aim project for me. That's how I divide it. So the first one is the creation of a functional sensing genetic circuit able to sense TPH traces. So crude oil is a mixture of different components of different elements. So the best idea will be able to identify all of them, but just for the sake of the project, I choose one. So this how should work. You should definitely select a chassis, and then you should add a riboswitch which is able to detect toluene. In this case, there are different. That's how in the left how a riboswitch could work. So if you have no toluene, you don't have fluorescence, but if you have the riboswitch on, you get a signal. So again, there are different bioinformatical options where they can help you design how it should look. And then again, you create a genetic circuit construction based on what you want to detect. Next, please. So my aim tool will be to find a matrix-based material that allows that modified bacteria to remain alive, because my project is based on doing in situ. You don't have to take a sample to the laboratory because it's too expensive and it takes time. So one of the two most nice ideas I found is sodium alginate hydrogels and chitosan cellulose hydrogel. So the first one is highly biocompatible, is inexpensive, and it's good for file applications. And the second one is stable with my microbial growth environments and its sensitivity. So that's the two options that I found. And the second and the third aim is more on long term. That is a prototype, a basic electronic module that captures this bioluminescent signal and then to be transferred to a database. So this is just something that I did, a prototype model of how maybe should look like. So you can have, maybe you can put it on a voice and the sea, and then you can create a signal. And whenever all of this is power and you have a signal, then you can send it to a database, how you see it here, when the information can be stored. So for me, this is one of the most important parts, because then you can make this information accessible to everyone, because if we're talking about ecosystem conservation and restoration, this is an issue affecting to all the Ecuadorians. So that's where my idea comes from. We want to make sure that this is happening right now as we talk. So, and it has happened since I was born. So basically, for me, this is how you should fix it, or you can start working, you're doing something, because while we're talking, oil is being spillage and nobody's doing anything. And
Hey,
this
Tamara,
is
so sorry. I think we have to really wrap it up now. This is a really fantastic no, so thank you so much.
Yes, it's no problem.
Does anyone have a really quick question? It was a really, really great project. I want to give the chance to have someone give a quick question. If not, I actually have a question about, so could you just quickly talk about, so for the hydrogel, how you would test like compatibility with like survival with the prolonged environmental exposure? I'm not sure if you mentioned that.
Yeah, so this will be probably to test error and good. I mean, I select a chassis, but there's some different options. So it will be probably to test which one combines the best with the material. I mean, these two are not the only ones. Basically, there are a lot of hydrogels. So I would think that is just to probe. And one of the first things is to make sure that the prototype is working, so you don't get a false positive. So for me, it will be just testing and trying to get a good signal, and that is working, right now.
Yeah, I think it's a really fantastic project, and I hope you continue with it. I think we have to move on to the next
Thank you so
project.
much.
Thank you. So Jenny Wang.
Hi, I'm here. Hi.
All right, go ahead.
Sure.
Thanks so much, Jenny. Yeah, really fantastic project. Does anyone have any questions? David, do you have a question? I saw you unmuted yourself.
Go to Adrian.
Okay, Adrian, go ahead.
Yeah, just very bad. So when the viral particles are going to hit the mask, you're going to trigger a signal. How are you going to take that signal and move it to such a signal that will go to your app on the phone there? How are we going to make that transition? You are muted. You are muted.
Yeah, sorry. So basically, once the virus the concept is that once the virus gets detected, it triggers the bioluminescence via the LuxCDABE operon. So that's the concept, and I'll be like doing further research.
Okay,
So
thank you.
basically, that light is detected by a small e-textile sensor, like a biosensor, and logged via a Bluetooth connected app.
Thank you.
Yeah, no problem.
So are you actually implementing this, Jenny?
I'm doing further research. So because I'm personally interested in biodesign. That's why I'm involved in this course to like learn further, and I've learned so much. Thank you, David, for offering this opportunity to global students. I really appreciate. So I'll be definitely doing like further and further research. So I've actually read a paper earlier. It's also like it's regarding like a mask that can monitor glucose level in real time. So that's really cool. So it's very similar to this concept. So I'm like learning more and more and finding like more interesting stuff. I really enjoy the process.
Yeah, it seems like it would be a really great application to bring to market. And yeah, that's a cool source of inspiration to the glucose monitors. Any other questions? Otherwise, feel free to reach out to Jenny in the chat and we can move on to Ethan.
Hey everyone, I'm Ethan, calling from Montreal, Canada. And yeah, this is my project. The goal is to create new enzymes for the Reclone Open DNA collection. You can go to the next slide, please. Yeah, next slide, please. Oh, sorry. Yeah, so for some context, Reclone is an organization that aims to make biology more accessible to the public. So they have this repository that you can check out where they have multiple open collections for enzymes, bacteria, yeast, etc. And then they have multiple reagent hubs for making it easy to share the DNA in parts and the collections. And so while this is a fantastic initiative, they're missing some crucial pieces to make this truly accessible to the public, and specifically T4 ligase and BSAI. So these enzymes are crucial for cutting DNA and sticking it back together. And it's really important because BSAI, like all these parts, use the MOCLO standard. So every part is flanked with BSAI restriction sites. And so you really need this to kind of cut and put things back together. And then actually the reason it's not available is because there was a patent on BSAI, which recently expired. So there's a great opportunity to make this available for everyone. So yeah, the goal is to synthesize T4 ligase and BSAI. Next slide, please. So T4 ligase is pretty straightforward. We found the DNA sequence online. I inserted the sequence into the PET28A vector, and then the protocol is to then order that from Twist, transform that into E. coli, induce it, lyse cells, and then since PET28A contains the 6X his tag, we can extract using IMAC. Yeah, next slide, please. And then BSAI is a little more complicated, and it's because since it's a restriction enzyme, if we start producing this inside E. coli, it's going to start cutting its own DNA, which is obviously a problem. And this is what was described in the patent. And their solution was to produce BSAI in addition to two methylase enzymes, so BSAI M1 and BSAI M2. And so what these do is they methylate the sequences that BSAI would cut, and then protecting it so you can produce the BSAI without worrying about like cutting your own DNA. And it's the same protocol as T4 ligase, but only the BSAI has the 6X his tag, so you can extract it without getting the two methylase enzymes. And so I have a screenshot there of the sequence. So you can see there's three enzymes there. There's the blue one, which is the BSAI, and then there's the two methylase in green and red. And if you see that only one of them has the 6X his tag. Yeah, and so the idea would be to produce these two enzymes and then distribute them out so that everyone can take full advantage of the Reclone repository. And yeah, that's my presentation. Thanks to everyone who helped out.
Thanks so much, Ethan.
Cool.
I think Ben has a question.
Yeah, what's up, Ben?
Yeah. Thanks so much, Ethan, for your project. Amazing idea. Do you think perhaps on using cell-free for the production of your enzymes? Because in that case, you don't have the genomic DNA, so maybe that will help for your reproduction.
That's an excellent idea. I wish I thought of that sooner.
Yeah, but great idea. I love it. Thanks so much.
Thank you. Appreciate it. Cool. Well, I guess I'll just pass it to the next person if there's no more questions.
Yeah, so yeah, feel free to talk to Ethan over the chat. Really nice project though, and I really like the open science angle again. So Divya from Celsius, are you here?
Yes, can you hear me?
Mm-hmm.
Okay, cool. We can go to the next slide. It's okay. So my project is similar to some of the ones that we've seen in the past 2 days around living materials. My background is in bioplastics and biomaterials, and I'm interested in how we can augment these materials with living cells and biology. And there's definitely a big wave of biomaterials that are being incorporated into the built environment or developed for the built environment. So if we go to the next slide, something I'm curious about, so I didn't mention this previous slide, all those materials are actually made out of agar, which I guess in the biology world is mostly used for gel electrophoresis or testing, but you know, it makes pretty cool bioplastics as well that I've experimented with. And I think something that could be interesting is as we talk about sensing, biosensing on larger scales, like on fabrics and textiles, on surfaces, on walls, there's an opportunity for us to actually design what that looks like and perhaps even develop a living materials design language. And I think that is because, you know, as we want patterns like green fluorescent proteins or other visible proteins to show up based on certain inputs and outputs, we can actually design perhaps what that patterning looks like. So that's kind of my first step. I won't go through the full protocol because it's a bit basic. I'm trying to figure out what I can do in the next 2 weeks, but I'm basically getting a plasmid that's already in E. coli that is inducible with QMate, and planning to do a few steps with the Opentrons to kind of combine Opentrons and the Agar art projects and basically try out different patterns and perhaps develop some kind of protocol around what are the design affordances of printing with bacteria versus, you know, it's different from inks and paints and things like that. There's probably different minimum feature sizes and perhaps even different ways of patterning that enhance the inherent qualities that it has. And doing some sorts of qualitative, perhaps through user studies as well as quantitative measurements with this patterning. And then on the next slide, I kind of have the bigger vision around, so yeah, AIM2 is kind of an intermediate, make it realistic, switching to visible chromoproteins and bacteria that can live in a dormant state or activated through or in cell-free systems. And as I said, you know, that picture of the fabric with all the circuits on it, that was kind of my vision of sensing textiles, but I think it can be redefined with biodesign. And then obviously the super big vision is, you know, instead of large LCD displays that are interactive in like buildings, like what if those were powered by bacteria? Or what if internal, you know, in architecture, a sweat can trigger an internal system that will start cooling rather than, you know, wasting power. I don't know. That's kind of the bigger vision. But yeah, would love to hear any questions.
Thanks a lot, Divya. I really like the project and also really beautiful slides, by the way. Anyone have any questions? I think I just saw a hand go up. David, go ahead.
Yeah, awesome project and yeah, beautiful design. You know, I think one challenge always with these types of projects is, you know, if you've got this kind of you know, bacterial powered system, you know, how do you keep the microorganisms alive and doing their thing in your system?
Yeah, I think that's where the aim to make it realistic thing comes in around maybe transferring it to a different type of bacteria that can enter a spore state. Or the harder challenge, which I won't be attempting this time around, is doing it in a cell-free system. And perhaps there's environments where it makes sense to keep things alive. Like I've seen some projects where, like a project by an artist where there's a light bulb that's powered by bacteria, and the intention is that someone comes and keeps feeding it and keeps it alive. And, you know, maybe that's part of our future living is we got to feed the systems that are providing electricity to us. And that's just kind of a behavioral change that we have to think about.
Okay, thank you very much.
Thank you.
Thanks a lot, Divya. I think we'll move on to the next presenter, Rodrigo. And this is the last.
I'm here. Could you hear me?
Go ahead. Yeah.
Okay. Well, I'm Rodrigo Tamayo, and I'm gonna present my project called Bisbal. Next, please. First thing, I want to tackle baldness. You can see here the cycle of hair growth and the proteins involved. So I chose the second one and the third one that are related to apoptosis of dermal papillary cells and inhibition of the anagen phase. Both of them results in follicular miniaturization. So we have a current therapeutic gap that I want to fill. So I want to propose a dual peptide therapy and use plant-based exosomes as carriers for this to make a regenerative strategy. So my aim number one is to design those peptides to target these proteins, then to optimize the system to put these peptides inside the plant-derived exosomes, and finally to evaluate the efficacy in vitro and in vivo to make a topical formulation. Next, please. So what I did in the beginning was to make those peptides using a PEP-MLM, then to use a AlphaFold2 multimer to see the molecular docking of this peptide with the protein. Then the idea is to put the peptides inside one vector and the proteins inside another vector and make a BIFC assay where the protein binds with the peptide, and if they bind, they emit fluorescence. So when I have a candidate, the idea is to put it in another vector, and this time with a HISTAG and a TEV site. The idea is to catch the protein and then to purify it. Next, please. Well, the next step is to make a sonication to put these peptides inside the exosome. The exosome are gonna be from native berries from Chile. It's like make a juice and pass it to a lot of filters to make the exosome. We're already trying to finance that project. We asked for fund, $200,000, so fingers crossed. We're allied with a company called Flare Nation. Then the idea is to, in the beginning, see with qPCR, and the expected results are to see a downregulation of the SMAD gene and also an upregulation of beta-catenin. Then the idea is to test it in mice, and finally to test it in humans to hopefully restore hair. Thanks.
Thank you, Rodrigo. Really cool project. Does anyone have any questions? So what are the main challenges that you foresee having in your project, actually, and which ones you've had so far?
Sorry, can you repeat the question?
What are the main challenges that you had coming up with the project and coming up with the aims, and then also what you might foresee?
Well, in the beginning, I never worked with designing proteins before, so I think that's the first one. The second one is that there is no exosomes from these berries, like there is no protocol yet. And the regulation to test it in humans, I don't know how it is in Chile, at least.
Okay, yeah.
I don't know if I can make a topic cream with that.
Yeah, so I guess you have some things to think about then for the future, but a really cool project, and I hope you continue with it.
Thank you.
So I don't know if we have any other questions, but if feel free to reach out to Rodrigo in the chat. So I think we are going to have a break soon, but I wanted to see if there were two people who were unable to go during their time. So is Anahi here now? I just wanted to give them a chance. Otherwise, we can try to move them to the back of the slide deck.
Yes, that's me.
Okay, perfect. Yeah, so I can move back to your slide. 1 second.
So after Anahi's presentation, we'll do a quick group photo. So find an interesting object you want to take a picture of it. So we'll do that right after this.
Perfect. Is everyone able to see my screen again?
We are.
Okay, great. So go ahead, Anahi.
Yeah. Hello, everyone. My name is Anahi, and today I will present my project. I call it NeuroLuma. Next. To start, I was looking for Parkinson's disease because it's a progressive neurodegenerative disorder with a silent early stages. And I have found two biomarkers that are strongly associated with DC, which is MIR153 and a misfolded alpha-synuclein. And the problem here is that there is no portable fast test that detects both markers. Actually, there are no tests for this. So I propose to have a rapid test for this disease based on two models. Two-hole switch, which detects the MIR153, which will express the GFP like a green, and a parment used to detect the properly folded alpha-synuclein. And next, I decided to take these two. Next slide. Yeah. And I worked here for when the MIR53 binds to the holes, I will express the GFP, and so it's gonna be a green color. And if we detecting the misfolded alpha-synuclein, it's gonna be like the logic is the contrary, because we have when is the well-folded alpha-synuclein binding, the program is going to be off, and when it is well-folded, it's going to be on. So here I put the presentation how the output and input and the interpretation going to be. Like if the output is green, is partially positive, and it's blue is partially positive. But if we have green and blue, it's fully positive, so it's very likely you will have Parkinson DC. Next. I will go to adapt this in a small rectangular device with two parts. We're calling this sample. The sample will be saliva, and it's going to find the part for the GFP and for the VFP. And I think this is very useful because it's gonna help us to find an early detection for the Parkinson disease, which is not another test like this now in the market. And I think it's going to be very useful in the future. Thank you.
Thank you. Great project. Does anyone have any questions? I'm perfectly on time. Ben, go ahead.
Thanks so much, Anaí, for your project. Amazing idea. I really like the final part of the prototype of the medical device. Can you maybe elaborate a little further on the device? Because you mentioned it that you will be using saliva mainly. Do you know that these proteins were being inside of the saliva, and how the sensing will work inside of the device? Yeah, only that part. Thanks so much.
Yeah, actually, I was looking for that information, and yeah, and most of these two biomarkers are present on the saliva.
Okay. I think Anaí, thank you so much again. And let's do a big round of applause next to Megan for a great job facilitating over this past 2 hours, and keep that applause going for Dominika, who did the first 2 hours. We've been going for 4 hours again. I feel like we're in like some other like dimensional time warp. Like this whole day, I feel like my brain is just like, I don't even know what I'm processing anymore. All of this epic how to grow synthetic biology all day. This has been truly, truly epic. Anaí, if you don't mind, stop sharing your screen. So what we're actually going to do now is we're going to pause and take a quick group photo. Okay, I know some people need to go to bed, especially our colleagues in Europe. So grab an object, any type of object you want. You can bring it in that you think is going to be fun to have with you. And then, oh man, I love the cat. Anastasia's got, I miss my cat. My cat's actually at a neighbor's house right now. Otherwise, I would bring my cat. I feel very jealous. But everybody, get your object ready, and then when you're ready, we're gonna do a picture together. Okay. Adrian's moving. People are running around. I don't know what happened to Dominika. Federico's leaving. Oh my God. All right. And turn your videos on, folks, if you're able to. Okay, here we go, everyone. 3, 2, 1, and oh, so good. Keep holding that. We're gonna do one more for the other folks. Oh, this is so good. And then last one. Oh, so good, everyone. That was beautiful. We love it. Before we go to the last hour and a half, I just wanted to say as well, again, thank you again, just to all of the nodes, all of the TAs. Y'all have been so amazing. For everybody that gets through the full documentation period, again, you've got 2 weeks to finish your documentation, okay? We have a whole process where we're going to do some grading and basically figure out who is gets a certificate, and I hope all of you are going to be able to complete your documentation for that process, and then we'll have a little graduation ceremony in June. So we've got a bunch more work. And just to say too, I'm going to send out a survey later today, okay? And so for everyone, How to Grow does not end even today or in 2 weeks or even a month. All of the TAs that have been working with you working so hard and doing such a great job, all of them were former students like you, okay? So believe it or not, you have the opportunity to go even deeper down this wormhole with us. Patrick, who is a TA, is now running one of our nodes and on our faculty. Same thing with Lisa. Thross was a student turned into a TA, is also a faculty member. Pranam was a TA and now is a faculty member. So we would love for you, if you're interested, to join our teaching team. That's how this all moves and runs and grows. We need you actually to help continue to scale all this amazing work. So that's one part. Also, just again, the projects have been so amazing. We've been ramping up our social media presence over the past stretch, and so we'd love to actually showcase a number of projects. So this will be something that we'll put out in the survey as well. But if you're interested in sharing your work as a part of our social media, we'll be reaching out to you and giving you an opportunity to do so with us as well. So anyways, those are just a few quick opportunities to engage with us. So this is not the end. Today is not the end. 2 weeks is not even the end. A month may not even be the end. And who knows, maybe you'll just keep doing this forever. We'll see. So anyways, that is my little ramble for now. It is 6 p.m. I am going to take back control of the Zoom deck, or of the deck here. And who is my timekeeper for the last hour and a half? I'm forgetting.
Georg.
Georg is back.
I'm
Georg,
back.
you got some sleep? You took a little nap? I love that, like, our Asia colleagues, like, they went to bed, they get up, like, we're all delirious here. Like, I'm pretty delirious, to be totally honest. I'm like, I feel like I need to like eat some of the How to Grow projects that somebody made. Give myself some energy for this last stretch. But all right, we are so close. And actually, Megan, there's one other person that we missed, right? One other person did not present. That's in your section. Is that right?
Yeah, I was just about to message you, but so her name is Mariana, and actually I believe she's now on the call. She just had some connection issues before. So
Oh, great. Actually, why
yeah.
don't we start with Mariana then? Mariana, are you here?
Hi, David. Yeah, can you hear me?
I can hear you. What's Mariana's slide number?
1720. That's it.
What is it? What's the slide?
318, I think. 318.
Okay, great. Okay, then Mariana, are you just audio or do you have video as well? I don't see you. Mariana, are you there?
She has some video, but it's kind of shaky. You can see her.
Oh, Mariana, I see you. Okay. You're muted, Mariana, but go ahead.
Oh, my slides are not bad ones. Are the ones from ALS.
Which slide number? I was told 318.
I believe 303. Oh, the blue ones.
What? Can somebody give me a slide number, please? Is it this one?
No.
I thought it was right after that.
318.
Yeah, there. Yeah.
Okay,
Thank
great.
you.
Okay, go for it.
Okay, thank you very much. Well, good afternoon, everybody. My name is Mariana Valdez, and my project for this section is rebuilding the motor neuron network in ELSE. So the next one, please. Okay, why else needs regenerative solutions? This project actually is born because of one of my familiars was diagnosed with this disease, so I could like really understand and experience how this disease is. So let me tell you about it. ALS is about the degeneration in motor neurons, which leads to muscle weakness, paralysis, and eventual respiratory failure. So it's about a progressive and fatal disease. It also causes this motor neural loss, which is a challenge for now to overcome. So with this project, I would like to overcome this challenge. And so the current treatments only like modestly extend the survival. So these are our current challenges that I could perceive. The diagnosis also, it's kind of tricky. So when it's already diagnosed, it really like advanced the disease. So treatments are also only for extending the survival and not regenerating the neurons, nor even helping that neurons to have this cure. So it doesn't have a cure. So it has a lot of causes. Anyways, any cause leads to this irreversible motor neuron. So the core idea of this project is to promote this regeneration and of course the remyelination of these neurons using genetic circuits to upregulate progenerative and neuroprotective factors. The next one, please. So here I present you two aims. The first one, of course, is to design the genetic circuit. We could use therapeutic genes, of course, anti-inflammatory genes. This also stands for the in silico design, the plasmid construction, and finishing the first part of this project with the cell line transfection, by transfecting these genetic circuits into neural stem cells or glial progenitor cells. The second aim is to test this functionality under ALS-like stress conditions, so we can assay the neuroprotection and remyelinization assays. The next one, please. And so this is a very general view about the project. I invite you all to visit my Notion page so you can know more about it. And well, these are my vision and my next steps for future work, and of course, my expected outcomes. Thank you very much.
Hey, great job. Questions for Mariana? I have a question. So again, I love the ambition of your project. Can you talk a little bit more about some of the experimental validation for the myelin regeneration? Like, how are you going to test that and measure that?
Yeah, it's quite ambitious. I know. It's just that I believe that we need this medicine, regenerative medicine in here. Mexico is also a good target for doing this. I believe that we need a lot of experimentations before like proceeding to clinical trials. So part of the experimentation, can I go slide before, just like to have the idea clear? Yeah, thank you. It's to propose, first of all, the genetic circuit. This would be in silico design, just like using a SnapGene bench-link and having this DNA assembly. And before having the in vitro assays, we could make this assays for the dynamics of how it could react, or how can the genetic circuit is working, like before having this step in vitro. And if the results are the ones that we are expecting, which are this treatment is having the results that we believe, which are the neuroprotective factors, then we can go to the plasmid construction and of course the transfection to these cell lines. Also, I believe that for having here these pharmaceutical projects and medicine projects, they have to be tested like really specifically, because I mean, we're talking about people's lives, people's health. So for me, it's really important to have these previous experimentations, of course, in silico and also in vitro.
Wonderful, Mariana. Thank you so much. Any other questions, please refer to the chat. Let's go next to
Thank you.
Melanie. Melanie,
Yeah,
are you
hi.
there?
Yeah,
Oh,
I'm
there
here.
you are. Melanie, the floor is yours. Go for it.
Okay, next slide, please. Yeah. Good afternoon to all listeners. I'm Melanie Jumbo from Quito, Ecuador, and this afternoon I want to tell you an open secret about an industry that contributes significant revenue to the country. This is small-scale mining, especially those that extract gold through a process called cyanide leaching. All the waste from this mining flows into the river, whose water is used by Ecuador's Amazonian communities. That's why I set 3 aims. First one, create a genetic circuit that gives Bacillus subtili 168 cyanide degrading properties and resistance to an alkaline pH. Why this bacteria? Because subtili is known for being safe, being robust, and easy to modify. This also has a high adaptability. Next slide, please. So I started searching the literature for a plasmid that is compatible with Bacillus subtili and has essential components. Then I found BGL0 under code 9. So at NCBI, I searched for genes that are involved in 2 important things. First, cyanide degradation, the NEAT gene, which produces a nitrolysis protein and has a cyanide degrading function. The second one, resistance to an alkaline environment. For this, the NH8A and C genes produce sodium and hydrogen antiporters. Now comes the magic. Using SNAP gene, the constructs were built. So the first one is with the NEAT gene, and the second one with the other genes, which are regulated by an alkaline inducible PATP2 promoter. Everything was inserted in the measurement vector, and this new plasmid was obtained. Then we have to introduce into the bacteria through like electrocompetition and other process. Next slide, please. So after all this process, my second aim is to evaluate the functioning of the genetic circuit and verify the removal of the cyanide using an SCR bioreactor to ensure that we can see the presence of the chloramphenicol marker gene by cultivating in nutrient-borne medium with one with CMR and the other medium without antibiotic. It's incubated and its presence is confirmed by growth curves. Similarly, an SDS page can be performed from the nitrolysis protein. Finally, to assess the degradation effectiveness, an SCR bioreactor can be built in a flask, and the initial and final amounts of cyanide can be calculated to obtain a removal percentage. Now we move on the final aim, bringing all of this to the mine tailings. To achieve this, we seek to design a biological reactor prototype that degrades cyanide. This can be achieved with internal modules of perforate steel trays to house the Bacillus bacteria biomass. This contaminated wastewater passes through the compartments, and the cyanide degrades upon contact with the modified bacteria. From this process, we can determine the retention time and the removal percentage. Finally, the ethical considerations and future perspectives that are important to address are preventing the accidental releases into the environment and strengthening a genetic contingency plan, and using auxotrophy in a system like can be in a future plan. So that's all. Thank you.
Melanie, that was awesome. This is really great. I appreciate your thoughtfulness across the board, from your design all the way to your implementation. Any questions for Melanie? One of my follow-up questions. So I really like one of the big challenges for a lot of these bioremediation projects. You've got a really great aim 3 here. We actually have a system you're showing where you kind of input kind of contaminated water and then filter it out through your system, which is great. Are you envisioning, how do you get to scale though, right? Like, is the idea that you'd have, like massive reactors like the one that you've shown here, or is there some version where this can go float out in a contaminated body of water? Or maybe you can say a little bit more about, if you're wildly successful, what this would look like.
Yeah, yeah. I like vision to put like this biological reactor in situ place where are all the tailings. So yeah, it's to like connect it with PVC contacts and like make all the wastewater pass through it, and then avoid that all these wastewaters go through the river and all the nature with the cyanide that is the contamination. So yeah, I want to put, if everything works well, I want to put this bioreactor in situ there next to the tailings.
Great. Awesome. Awesome job. Thank you so much for the great work and the great presentation. And you can, again, continue to ask Melanie questions in the chat. Let's go to Isabella. Isabella, are you there?
Yes, I'm here.
Okay, Isabella. Let me see, did I spotlight you? Okay, yeah, go ahead. The floor is yours.
Okay, can you go to the next slide, please?
Oh,
Oh,
there's no slides here.
I don't know
Is
what's
this
wrong. No, my slides are on the presentation.
Okay, I think I need to reload. Just give me 1 second here.
Thank you.
Okay. Here we go. Uh-oh. My favorite thing to eat is chocolate, so this is gonna be a problem. I could use some chocolate right now. Okay. Oh. I'm so sorry. Give me 1 second here. Okay. And let me go full screen. Okay, go ahead, Isabella.
Okay, thank you, David. So today I'm presenting about chocolate flavored yeast. Agriculture has been significantly impacted by climate change in the past years. Certain crops, not only staple crops, have been affected by this, and some of the affected plants are those who produce popular flavors like vanilla and chocolate. Synthetic versions of these flavors exist, but they often fall short to the real deal. So, and also the nutritional benefits, they lack the nutritional benefits, and often their production contributes to pollution. So scientists and researchers have been trying to use microbial platforms to produce these compounds. It has successfully been done for vanilla, so I tried to do it for chocolate this time. The two main components that give chocolate its qualities are theobromine, which is an alkaloid and provides a bitter taste, and linalool, which is a terpene alcohol that gives a floral aroma. The objective of my project is the expression of the biosynthetic pathway for theobromine and linalool in Saccharomyces cerevisiae to replicate the chocolate flavor. Next slide, please. My first aim is to see if the metabolism of Saccharomyces was able to support such big metabolic pathways. Theobromine needs 2 enzymes, and linalool needs 5 different enzymes to be able to work. So for this, I designed 3 different cassettes that will be integrated into different chromosomes of the Saccharomyces genome. This integration will be done using CRISPR, and the guides listed there, they will induce double-stranded cuts, and through homology-directed repair, these sites will be included into the genome and expressed. The evaluation of the results will be done with an OD600 for the cell growth, and the liquid chromatography for theobromine, and gas chromatography for the evaluation of the production of linalool. Next slide, please. So the integration is expected to have a 100% success rate, since we're using guide RNAs, which make precise cuts. And then the production of linalool is thought to be bigger than the production of theobromine from previous studies. So this leads to my second aim, that is to optimize these pathways and be able to grow them more, and be able to grow these strains in a batch system. My final aim and the end goal of this project is to establish a protocol for freeze drying and being able to sell this yeast as a supplement with an endogenous chocolate flavor. This, by achieving this, it will open up the possibilities to the different flavors that can be produced within a yeast vessel. Thank you so much.
Great job, Isabella. I'm a massive chocolate fanatic, so thank you for working on this project. Dominica, a question to you.
Yeah, with any kind of biosynthetic pathway that you introduce into like a novel organism, there is like always the question of stoichiochemistry. So what thought have you put into like ensuring that the components are expressed in a manner that they are also like in the host organism?
Yes, so as we saw in the last slide, the theobromine production will be lower than the linalool. So I think that's my limiting, the enzymes that produce these are my limiting components. So to solve this, maybe there could be multiple clones of this pathway to express more of the enzymes that produce the theobromine. And for the genetic charge, I think about trying different promoters that will have different effects on the expression levels and will not interfere with the metabolism of the yeast.
Yeah, that was the answer I was looking for, right? Promoter and ribosome binding site variations would be great.
Great. Thank you so much, Isabella. Appreciate you. Tanisha,
Thank you so
are
much.
you there? Great job. Tanisha, are you there?
Yes, I'm there. Can you hear me?
Okay. I can. Tanisha Shah, the floor is yours.
Thank you. Next slide. So the idea for my project sparked from when I was trying to do like an animation using the Pikachu. I guess some of you would have seen. And quick shout out to Ronan for like running these in the MIT's lab. But I was trying to do like a frame by frame animation using the Pikachu, and it was like quite difficult because they're not equally centered. Sometimes the level of the agar board gets disturbed. So then I started researching into this, and my question was, how about we make like a screen using biology, like a display where the visuals change by themselves kind of thing. And I found this one project that one of my classmates led me towards, where they use like E. coli as biopixels and they got a still picture. But I wanted to generate a system where it's like a video, it keeps changing, not just a still picture. But then there were some problems with that. At first, I thought, how about I use reprislators? But over time, they go out of phase and not the frames would not be at the same time anymore. So then I thought, hey, okay, if we use fluorescent protein, then they can't go away. So how do I degrade it? But chromoproteins are not bright enough. So while studying about all of this, I found this hybrid E. coli protein called MRFP, which has this fitness cost test that they did, and they realized that it's much more efficient to produce at higher qualities, and it still does not have like a high metabolic cell load. So I used those colors, and then I wanted it to be light sensitive. And there's this constructs of P-dawn, P-dusk plasmids where the colors change in response to light. So I thought, how about if I combine both of these and make like these chromoprotein and hybrid fluorescent protein colors to be the response to the promoter, which is light sensitive. I could potentially have a system where when you change the colors, the screens change over time. So to design this, next slide. Basically, I made the system of like 5 plasmids. I initially chose many different colors, but then that would be too many plasmids to put in a single E. coli cell. So I shortlisted it down to 3 colors. It could be any 3. But for my idea, I wanted to like recreate the Starry Night painting in life. So I took like yellow and blue and lime. And then there are the control plasmids, which have like the light sensitive parts of it, which the promoters would react to. An important aspect was that each one needs to have like a different resistance and a different backbone so that they do not compete with each other. And also there are like degradation tags added to the plasmids with the generic color so that after like a phase of 2 to 3 hours, the whole thing changes. So if you go to the next slide, basically the design would be such that there would be like an LED matrix under the Petri dish, and there would be an agar grid of E. coli cells printed with the opentrons, and the LED matrix would change the color projected in every few hours, and the E. coli cells would like mimic that, and we would get the picture. So the first aim is to test this out. And concept is like first you have to test if the light system is working in one of the colors, and then you expand it to more colors and see if the cell can still handle all of that mechanism and all of those plasmids in one. Once you get that working, of course, then you have to see if the timings are right, the transitions right. Then you need to see the time lapses over time to see that the phases are being maintained and there's no cross-contamination. And of course, the last aim is that, hey, we could just make this open source and give like an online interface where people can upload a video, it gets translated into frames, and they can have like this translated into their digital bio screen. So yeah, and it's pretty biosafe because it's all E. coli and it's all going to be degraded pretty soon as well. So there's no like risks. So yeah, that's all. Thank you.
Anisha, this is beautiful. Again, are these some of your renderings, or so are these some of your drawings here of your system?
Oh, I use Sora for that. Like I detailed what I wanted to. I basically did hand-drawn drawings as reference, and I made it create the images back. So yeah.
They're great. They're really, really great. Thank you. This is awesome. Adrian.
Yeah, Tanisha, that's a beautiful project there. This is more like a suggestion than other things, because I was actually, it just happened to correspond with Andreas Moglik, the person that created PDAWN and PDUSK. So he has some more constructs. He has some that work much better. They have like a 250 times more expression when it's on than when it's off. And they also respond to red light. So you can even use like a multiplexing using 2 lights, or actually you get 4 switches if you turn on and off, because now you have two bits, and you can use that too. And they are on some of them are on AdGene, and I was talking to him and he said that he's going to have some published very, very, very soon, some other ones very, very soon. So that's what I want.
That's kind of exactly what I did. Like I do have my light sensing. It senses one is red, one is green, one is blue. And I did take those constructs, the ones you mentioned only. So yeah, but thank you.
Right.
Adrian, if you can share that resource on the Slack in the general channel for everyone, that's great.
Okay, I will do that.
Fantastic. Thank you so much, Tanisha. Awesome job. Up next, Kate Melanson. There you are, Kate. Oh, Kate. Hey, we just saw you in person. Kate was with us at MIT yesterday.
Yeah.
Great to see you, Kate.
Thank you. Happy to be here. So for everyone else, I'm going into my second year in Carleton, and I'm studying health sciences, and this is my project, Haroma. So next slide, please. So when shadowing a surgeon in Barbados, I watched a hysterectomy as a result of overgrown uterine fibroids, non-cancerous solid tumors that had grown on the uterus and led to heavy menstrual bleeding, anemia, pelvic pain, and infertility. The surgeon explained that women in Barbados have an increased likelihood of uterine fibroids as a result of risk factors such as increased diabetes and a prominent African lineage. With 18.7% of Barbados being diagnosed with diabetes and 92.4% of Barbados descending from Africa, uterine fibroids are a burden to Bajan women and the island's healthcare system. The Caribbean's lack of early screening and specialized physician training leaves many women living with undiagnosed, untreated uterine fibroids, impacting their quality of life. My final project looks at a way to overcome these technical and socioeconomic barriers through the development of an early screening modality, Haroma. Next slide, please. Haroma is a biosynthetically smart menstrual cup that analyzes the concentration of uterine fibroid biomarkers from the comfort of the patient's own home. The menstrual cup synthesizes technology from a Bluetooth menstrual cup already on market and biomarker sensing electrode technology. This creates an information flow from the patient's menstrual blood to the physician's analysis. Biomarkers present in the blood bind to an aptamer, changing the aptamer's conformation and thus the rate of electron transfer. A change in the rate of electron transfer changes the magnitude of an electrochemical signal produced, reflecting the biomarker's concentration. This is then shared via a Bluetooth module to the patient's phone and then via Wi-Fi to the physician's phone, allowing the physician to identify at-risk patients. Next slide, please. The two biomarkers chosen are cytokines TNF-alpha and IL-6, both pro-inflammatory cytokines involved in the local environment of fibroids. TNF-alpha is consistently elevated in women with fibroids and closely linked to disease severity, whereas IL-6's role is more localized to tissue inflammation and remodeling. Crucially, both proteins are released into the blood, allowing the aptamers to bind directly. As I set out to design the aptamers, a literature review revealed that researchers had already developed aptamers using the CellX method. I then visualized the TNF-alpha and IL-6 with AlphaFold as seen on screen. Once the aptamers bind to their respective biomarkers, the conformational change alters the distance between the methylene blue group and the electrode, which is why the rate of electron transfer changes. Actively, I'm trying to visualize this distance change using GROMACS. Understanding this distance provides insight on whether the signal's magnitude would increase or decrease when presented with high biomarker concentration. This is important as an increase in the concentration of TNF-alpha and IL-6 increases the risk of uterine fibroids. Therefore, measuring biomarker concentration allows physicians to determine which patients are at risk, allowing for early intervention. This would alleviate unnecessary suffering from the women of Barbados and allow for the reallocation of funds within the Bajan health system, helping the women and the island thrive. Thank you for listening, and if anyone has any questions, please ask.
Okay, Kate, great job. Questions for Kate? One question I have is when you want to actually, if you imagine the assay works and this was a success, what's the kind of form factor and the interaction for the women that are using this? How would that look?
I'm not sure I completely understand.
Like, how would you actually test this? How would you actually use this system? Is it like a like a pregnancy test, like a flow strip? Like, what's the
Of course.
mechanism?
Yeah, I was actually thinking about this a lot. I changed my original plan. I was thinking maybe a watch, but I want it to be almost like a pee in a cup test, but you can't go and just like bleed in a cup because that's just going to take far too long. So it's something that a physician would be able to give women, and then they would be able to go home like at a certain age, especially because there's an earlier onset for fibroids in women in Barbados. So it'd be like a timely age situation because to see if they're at risk or they have uterine fibroids. So I see it as something that they would take home. And that's why there's a Bluetooth and Wi-Fi component because they'll be in very far, they won't be in the clinic when their biomarkers are being scanned.
Great. Thank you so much. Any other questions for Kate? All right. Great. Thank you so much again. Oh yeah, Federico, let's ask in the chat. We're at time, but why don't you go ahead and ask in the chat? Thank you so much. And Federico, I appreciate all your questions today. You've been great. Thank you so much for all the great questions. Kate, thank you. Great job. Let's go on to the next presentation. Oh, Hyungseo.
Yes,
Great
I'm
to
here.
see you. And thank you for all of your leadership in South Korea with Patrick's group.
Thank you. So yeah, for me, I'm Hyun-Suk Kwon and I'm studying in Designer Cell Labs in Yonsei University. And today I want to present about like gut soil axis specifically in Korea. Thank you. So the background information and what the subject brings me is that the relationship between soil and gut microbiome was really interested to me, and it seems like an emerging topic in microbiome area. So I tried to study whether the soil has been studied in Korea, but it hasn't been really interact with the guts because it was only there's interactions based on dietary, not soil. So I found some synthetic biology soil microbiome study. So to use universal primers called V3V4 region of 16S RNA, which is used for valuable like providing good region resolution of for identifying bacteria taxa. So by having the middle point of the point, I want to find some alpha and beta diversity of Korean soil microbiome. Next slide, please. So this is a protocol I set up. So I want to collect 10 samples from Korea and then 10 samples from United States, which is uploaded on the internet. And I want to extract the DNA with the soil extraction kits and then do PCR with the V3V4 region using the universal primers, which is I explained it before. and then we'll do gel electrolysis, and then after I'll do fish for pre-test assay. And there's like positive control, which is only in Korean microbiome, and then negative control, which is only found in like United States. And then after checking, there's pre-test, there's what we want. I'm hoping we are going to have some next generation sequencing and get some data and know what microbiome we are having. Next slide, please. So this is an example of the results I want to get. I really want to find valuable results from soil gut access of especially in Korea. So this will gives us to understand the interaction between the microbiome, to understand the significance of pathogenesis and progression of human disease. Yeah, thank you for listening.
Okay, great job. Questions for Hyunseo? Ben.
Yeah, thanks so much, Hyunseo, for your presentation. Really interesting. Do you know which kind of sequence technology you will be using? Perhaps what would be the best? Maybe making a long or short reads. You know something about it? Just that. Thanks.
Sorry, but could you repeat the last word?
Oh yeah. You have different kinds of the sequencing technology. You can do long reads for maybe making a read T2T, telomere to telomere, or maybe just making reads like small fragments. Do you know which one will be the best for you, for your case?
I think I have to really study more about what topic, what like method I have to choose. Yeah.
Thanks, Ben. One quick question too. So again, I love this soil gut access. This is the first I've heard of it. There's microbiome off as like gut brain access, soil gut access. Can you say, I know you're studying soil gut access in Korea. Are there other soil gut access studies in other parts of the world and that you can reference or learn from?
Yeah, actually, there is like 2 papers only about saying the soil gut access. So those papers really got me interested in this topic. So from that papers, those were not in Korean, so those were from US.
Okay. Yeah. Okay. Very cool. We'll leave it there for now. Hyunseo, thanks so much. And thank you again for your leadership. All right. And again, let's continue the questions in the chat for Hyunseo or any of the speakers. Eduarda, are you there, Eduarda?
Yes, I'm here.
Is your video on, or are you not able to do a video? I do not see you. Oh, there you are. Great. Okay, Eduarda, the floor is yours.
Okay. So good afternoon. I'm Eduarda, and in this project, I designed a synthetic long non-coding RNA to target the GP82 messenger RNA from Trypanosoma cruzi. The next slide, please. So, like a little of background, the non-coding RNAs, which are not translated into proteins, include short, medium, and also long RNAs. In this case, I focus on the long ones that have over 500 nucleotides and that regulate gene expression across organisms, including kinetoplastids like Trypanosoma. One known example of long RNA in these organisms is the Grumpy from Trypanosoma brucei that promotes differentiation into a stumpy form. This stumpy form is non-replicative and is less pathogenic. So Trypanosoma cruzi has like a cousin apparent that is Trypanosoma cruzi, and this parasite causes the Chagas disease. But the role of the long RNAs in this species is still unknown. So because of their evolutionary proximity, I hypothesized that Grumpy-inspired long no-coding RNA could similarly modulate this differentiation in Trypanosoma cruzi. So because of that, my aim is to use the synthetic RNA Grumpy to interfere with the GP82 gene from Trypanosoma cruzi, which is essential for host cell invasion. Next slide, please. So the first thing I did was to analyze the original Grumpy structure and its binding to the messenger RNA of GP82. Then I retrieved the sequence from TripDB and simulated co-folding, which showed a good interaction. Then to improve specificity, I modified the region of Grumpy called SNOR that is responsible for the binding, and then replace it with an antisense sequence complementary to the GP82 messenger RNA. This region was inserted into a hairpin to stabilize the interaction, and then I integrated into the original Grumpy backbone. So then I co-fold and fold the new RNA designer, and it has a good stability. Then I inserted it into the plasmid that you can see on the screen. I used 2 restriction sites, and this is a plasmid that is specifically used for Trypanosoma cruzi. The next slide, please. So the next steps would be that I will transfect Trypanosoma cruzi epimastogotes with the plasmid and select the ones that are transformed. Then I will evaluate the expression of my RNA following the steps of a previous article. And if this all is successful, I hope that the synthetic RNA could become a basis for RNA-based therapies against Chagas disease. So basically that would be. Thank you.
Hey, great job. Thank you so much. Questions for Eduarda? So one question I have, again, great job in thinking through your whole process. Can you talk a little bit more about your functional assays?
Yes, the functional assays would be like proliferation, differentiation, and GP82 expression. These assays were done on the article that reported the Grumpy RNA on Trypanosoma cruzi. And basically, they are also associated like with the parasitemia, with the time that infected mouses could have when they have like the RNA and when they does not have it.
Okay, great. And you haven't been able to do anything in the lab yet, right? This is your hope is to be able to do those experiments, right?
I mean, I hope to. I think it would be wonderful, but I don't know if it is possible because unless here in Ecuador, because we need to work like directly with the Trypanosoma cruzi, and I don't know if the labs are like well established for that to do that. But I know that in Uruguay it is possible, and I think it also in Chile.
Okay, great. Thank you so much, Eduardo. I appreciate you very much. Cesar Rubio, are you here?
Yeah, yes, hello. Can you hear me?
Cesar. Yes, we can. The floor is yours.
Okay, thank you. Well, my name is Cesar Rubio. I'm very happy to present to you my final project, which I named RisoAlert, a fluorescent biosensor for monitoring the health of your houseplants. Next slide, please. Okay, so many people around the globe, and maybe some of you have tried to to grow indoor plants in your houses or your apartments. But maybe you'll have found yourself seeing your plants, your beloved plants dying, and without really knowing why. And what happens is that many times our plants are stressed, and we don't really, and we can't really know until it's too late and we see our plants wither and die. So I had this idea to engineer soil bacterium, specifically this species Pseudomonas fluorescens, and make it able by a genetic circuit to detect ABA, which is a stress hormone in plants, and then express a signal that we can see, and that way know if the plant is under stress. So the background of this is there have been already made different synthetic biosensors in this rhizobacteria. And next slide, please. Thank you. And so here I show the genetic circuit that I like to create, which basically consists of an expressing an ABA receptor, which is this hormone. So we express this receptor, making the bacteria sensitive to ABA, and then it will create a signaling pathway that then activates a specific promoter, then promotes the expression of RFP in this case, which is a protein, a fluorescent protein. And so the idea of this, as you can imagine, is that we can detect the fluorescent signal in the plant. Okay, so going rapid with this, I would clone this DNA into a plasmid, integrative plasmid, then transform it into the bacteria, select the bacteria transformed, and then I would evaluate if this bacteria express these genes, and also if they're effectively if they emit a red fluorescent signal when excited with UV light. Okay, so okay, so basically, next slide, please. So basically, the final part of this project would be to really evaluate if the plant, these bacteria send these fluorescent signals whenever the plant is under stress, like heat, salt, or drought stresses. And so determine if the system is working, and then assess that the system is safe, that the bacteria isn't harmful to plants or humans, and develop a customer-friendly kit to help people grow their plants. Okay, sorry, I passed on the time, but thank you for your attention.
Great job, Cesar. Any quick questions for Cesar? Ben, go ahead, very quick.
Yeah, thanks so much, Cesar, for the presentation. Really interesting, your project. Did you know maybe if the same molecule will alert you from different stressors, or there will be a way, like, for example, to know if it is a stress for cold or for water or different things? Just that. Thanks so much.
Thank you. Yes, so this plant hormone ABA isn't really that specific as far as I know. It is secreted by the plant under different stress conditions, but I think this project could be like expanded and maybe to create different biosensor for more specific kinds of stress. But this is like general to know that your plant is in a general condition of stress.
We'll have to leave it there. Cesar, thank you very, very much. I know Georg
Okay,
has a
thank you
question
so
for you.
much.
You're welcome. Thank you. Great job. Okay, Jorge,
Hi, I'm
you're
here.
next. Go for it.
Well, good afternoon, everyone. My final project is a molecular modulating and a in silico validation of a peptide inhibition and a target MDM2 p53 interaction for a cancer therapy. Next, please. Well, in this project, we address the interaction as between a p53 is a K-tumoral suppressor and MDM2 is a main negative regulator under normal conditions. MDM2 controls p53 by a promotorin in degradation. But in many cancers, MDM2 is overexpressed in activity p53 and favoring tumor growth. As a solution, we propose to design a peptide that blocks this interaction, restoring p53 functions. We will use our artificial intelligence tools to design faster and more accurate inhibitors. The objectives, the aim, one, to design a destructive prediction, these peptides, two, validate then marine models, and then develop a system for the early detection and a personality application of cancer therapies. The next, please. Well, the experimental design considered developing a peptide capable of inhibiting the MDM2 protein with the aim of reactivating the supracellular fusion of p53. For this purpose, the crystallographic structure of the MDM2 p53 complex was analyzed and K interaction residues are identified. From the information p53 derived peptide variants were generated.
Oh no. Did we lose our speaker? I think we did. Wow, that was highly unfortunate. Let's see. They come back. I don't have visibility in the Zoom. Are they not in the Zoom room at all anymore? Georg, Danny,
No, it's not in the Zoom anymore. He was, maybe his connection get lost or something.
Yeah, he was kidnapped by the internet. Okay, well, why don't we come back? Because I know, yeah, I'm sure he'll be back in a bit. But why don't we move on to the next project for now, just to keep things going. Is Gustavo here?
Yep.
All right, Gustavo.
Sure.
You're on.
So hi everyone, I'm Gustavo, and we can go to the next slide. So a very new bioluminescence has been elucidated the whole pathway in 2018, and you're probably seeing around this bioluminescent fungi that they produce this green light. It was elucidated very recently, and we know the whole pathway. It has many really cool things about it. First of all is that we know the whole pathway, so we are able to produce de novo the luciferin in vivo. Also, the luciferin and precursors are not toxic at all, and it also has a high level of orthogonality, meaning that basically, if you have different bioluminescent systems in a single cell, you'll be able to have different signals, and they have different substrates, and it's also ATP independent. The reaction basically goes from caffeic acid, requires two more enzymes, and you produce the luciferin, which then gets converted into light by an oxidation reaction by the Lus enzyme in the fungi. This is a very promising technology, and it's something that is being growing, especially in the plant industry, in the plant research community, because plants produce endogenously the caffeic acid, meaning that you can have a bioluminescent plant, and it's being super useful for this kind of research. The problem here is that the ideal activity for this enzyme is around 10 to 18 Celsius. Maybe around 20 Celsius could work, but then it loses its activity. This is fine for the fungi that lives in the forest, but if we want to use it in research or even biomedical research, like in mammals, the physiological temperature, it loses reactivity and it's not that useful anymore. So the goal with this is to try to make it as stable, as robust as we can, and also make it as small as we can to make it even more useful. This will open the doors to make better biosensors, better biomedical research, and even art applications. Next one. So to do that, I decided to go for a protein engineering route. I looked for the most up-to-date version of the enzyme, which in this case it will be last before they added some mutations to it. And then what I did is that I decided to go for a homology type of analysis. I went into literature and also blasted the protein sequence and looked for several sequences from different fungi that in literature has been cited that they are bioluminescent, meaning that the whole pathway should be working. And by doing that, I also did a multiple sequence alignment and with Classical Omega be able to see the different sections. And I updated all of this into Camera X by looking into the different structures and also labeling it by the level of similarity. I was able to track which sections are conserved, therefore try to keep in them. I looked for the different cavities, different sections that I could mutate, and I was looking for the different amino acids that I could change to try to fill this up, make it more compact, and also try to trim some sections of it. Next one. So with this done, basically what I did is that I generated 2 mutants, one with a total of 13 extra mutations from the wild type, one that is truncated. And the idea is to try to express this in GIST. In this case, I'm going to use Comatobella pathy, previously known as Pichia, and try to use this with the OpenGIST section. These are a little bit of the logic behind the mutations that I did to try to add the different amino acids, to try to fill these gaps, to try to increase the stability of the different loops. And the idea is to produce the whole system that I talked before, to produce the light inside of the yeast, be able to see if there's any difference and vary the temperature, being able to use cytobioluminescent assays and the bioluminescent output as the way to look if it worked or not. Yeah.
Okay. Thank you so much. Gustavo, this is a really cool project. You should definitely talk to Ren. Ren for her whole doctoral thesis is working on fungal engineering. Very, very challenging work. So this is a really exciting project area. Questions for Gustavo? One question that I have is for your thermostability part against another great challenge area. Everybody's trying to figure out how to make thermostable enzymes. Can you talk a little bit more about say a little bit more about the computational approaches for how you're trying to enhance thermostability?
Yeah, I had 2 main sections. So I look for the different holes that were inside of the different cavities that were inside of the enzyme. And the goal will be to try to fill them up, like you see there, to try to fill them up with these different amino acid versions. So by looking into the homologs of the other taxonomically close fungi, I decided to do a more conservative way of doing that and choosing between them who will fill it up to try to make it more compact, more resistant to just falling apart with more energy. And the other one is just adding prolines to it because it just adds more rigidity to the general structure. We'll have to test in vitro, well, to be able to see if it worked.
Gustavo, this is really awesome. I hope you can do these tests. This is really phenomenal. Great job. Great work.
Thank you.
Thank you so much. Okay, Jorge, you are back. You got sucked up by the internet there. We lost you.
Yeah, my computer have a problem. I
Oh,
don't
it's
know what
okay.
to say.
All right, so we were on this slide, right? This experimental slide,
Yeah.
method slide. Okay, so why don't you
Yes.
pick it up right here? Go for it.
Thanks. Well, the front end is information P53 different peptide variants were generated and their three-dimensional structures were modeling a product their stability and affinity for MDM2 protein. After select the most promising candidate as an expressing plasmid named PEPT MDM2, scan peptide fused to a STIP tag was designed for subsequent purification. This plasmid include essentially genetic element for this production in a bacterial system. Overall, this provide a combined computation and molecular biology tools to develop a target inhibitor strategy against MDM2. Next, please. Then the next step of the project is preclinical validation of an inhibitor of this peptide. Step this includes evaluating its efficacy in mouse cancer models and cellular lines using techniques such as RNA sequence. Well optimized is a delivery by liposomes and localized injections. And additional is stability to cross the cell membrane will be investigated. It is expected to develop personalized therapies, design a specific peptides for different tumors profiles, with the aim of overcoming the limitation of current therapies and then offering accessibly stable and low-cost treatment. Thanks.
Okay, Jorge, thank you very much. Any questions for Jorge? Oh, I'm sorry, I had the wrong person pinned here. Jorge, you mentioned, obviously there's a lot of people working on different types of cancer therapeutics. You mentioned the kind of advantages of your approach. Can you highlight them again? What are some of the key advantages of your approach over existing methods?
Well, the methods I use, yes, is an key adaptation of provide the specificity by design a peptide in the way that directly block the MDM2 interaction P53. We can selectively restore P53 tumoral suppressor. And then another method to use a computationally design, for example, use the tools PyMol, Google Colab, Autodoc, Binam, Protein MLNA for design and simulation all these treatment and the peptide. And we can generate and evaluate this peptide candidate, making the process faster and more cost effectivity to compare a traditional drug in use in this moment.
Okay, great. Thank you for the response and thanks for the great project. More questions for Jorge in the chat. Let's go next to Dr. Data.
Yes, can you hear me?
Okay, the floor is yours.
Okay. Good evening, everyone. My name is Data. Today I will present my work, affordable modular biotech equipment. Next slide, please. Yeah, I taught elementary and high school students to learn biotechnology and STEM education after my PhD. Most of the schools in Asia are focusing on the robotics, AI, coding. However, it's more interested in biotechnology, but the equipment is still very expensive and not all school and students can access the professional web lab. Is the problem. I got inspired by the Nintendo Labo using Omigami cardboard as their case. I made a device qualified to lab, which is affordable, portable, and modular biotech equipment. To make this work, it's based on we have a low-flow and cheap electronics recent years, and the modular and STEM design provide great flexibility and adaptability in different biotech and biorelated experiments. In this course project, we design, fabricate, and test our 3 key modules, PCR, centrifuge, and transmitter. We use 3D printer, laser cutter with our designed PCB printed circuit box and the off-the-shelf microcontroller. Next slide, please. Our first aim in this project is to design the hardware system by using Fusion 360 for video modeling and Lego for the electronic design. We first combine all the modules in an Omigami cardboard. The second aim is to fabricate the prototype and test it. We keep iterating our design so that we make the version with the module design and the base. You can see that we used we 3D printed some centrifuge top. Let's slide, please. The last aim is to plan what else possible module could we have. We brainstorm over 50 modules ranging from PCR, centrifuge, transmitter, Dragon power supply, shaker for the DNA reading. For ecosystem, we could possibly have the IoT power microscope, spectrometer, oscillometer, pitch meter. For microfluidics, synthetic pump could be designed, and even we could have a mini bioreactor for bioproduction. And finally, we could add sensor like RGB, infrared, UV sensor, and the AR power camera for monitor the experiment. In this project, we research and learn from previous similar projects like OpenPixel, BintoLab, PocketPixel. We send out one to all power. We decided our machine to have USB input and have an output from 5 to 100 volts. It's essential for the Gel 10, and we have a module designed for different biotech equipment, which provide flexibility and reduce the cost. Although we cannot access the wet lab to view test our equipment, we outline several biotech protocols to validate our experiments. For example, bacterial transformation, PCR modules heat swap the cell for DNA uptake, then stably incubate them for recovery. Finally, our LED transmitter visualize successfully forensic transformation or PCR verified DNA on gel, confirming our design works. Yeah, thank you.
Okay, Dr. Data, this is awesome. Are you familiar with the class How to Make Almost Anything?
Sorry?
How to Make Almost Anything? Have you heard of the class?
No.
Okay, so you should look into it. How to Grow Almost Anything is based on How to Make Almost Anything. And Neil
Yeah.
that class. Yeah, he's always wanted to teach a class about Fab Labs making biolabs, and that's what you're doing here, which is Fab Labs making biolabs, which is amazing. So it's almost how to make almost anything that grows almost anything. That's like the class
sure. Yeah, I know that.
That's our joint class. Yeah. Anyways, I love this. This is really great work. I love all the low cost lab work. It's very exciting. Mike, one quick question. You know, a lot of people have tried to make kind of low cost equipment like this in a lot of different forms. I'm curious what you think your main innovations are. Like, what is the new thing contribution here compared to all of the previous kind of open hardware types of designs that are similar to this?
Yes, we decided the module design. We somehow outline a standard so that we can use that standard to fit in different kind of equipment, so that it makes the people could design their own equipment, and also it's portable. I think it's one of the key things, because the student need to do that in their classroom on the tables. Yeah.
Okay, great. Yeah, modularity, that's definitely very true. I think a lot of those other systems are not very modular, so that's a great contribution. Thank you so much. That's great.
Thank
Yeah,
you so
please
much.
more questions for Dr. Data in the chat. This is a great project. We love to see it.
Yeah,
And
thank
for
you so
whoever's
much.
helping the timekeeper, if you don't mind keeping the timer going so we know how much past we are. Okay, so the next speaker we've got. I hope I pronounced your name okay. Is it
I'll say it is Heike.
Heike. Okay, great.
Yeah.
You are up next. Go for it.
Thank you. Hi, I'm Heike. I'm a designer in San Francisco. And if we could go on to the next slide, please. So this is a project, again, about the human microbiome. So what if everyone had access to their own biome and the global microbiome, and that we could optimize the biomes in an easy to use, reliable, and cost-efficient way? Because we know that our microbiomes are integral to our health, and we've done a lot of research about it, but we still don't know enough about it. So for example, does the microbiome cause changes to disease, or is it a consequence of a disease? So lots of questions and lots of research that needs to be done. But today I will not boil the microbial ocean, and I will focus on the oral microbiome. And specifically, we're going to look at our teeth, because life is better with teeth than without it. So what we will do in this project is we're going to devise a bioengineered probiotic, E. coli Nissl, to detect and destroy the main agent that causes periodontitis, which is when the bone loss occurs and you lose your teeth. And even better, during the destruction of P. gingivalis, a pleasant wintergreen taste is released. And the novel aspect of this system really is that we will have a sensory feedback therapeutic. So while you're taking it, you can actually sense that something is happening, that something is working. And the goal is obviously to reduce periodontitis. I did not know this, but apparently it is the 6th most common infectious disease worldwide. And like I said, everyone wants to keep their teeth. In the illustration, you can see there's someone who has gingivitis and eventually periodontitis. The delivery method of the E. coli, of the engineered E. coli Nissl, is a lozenge or lollipop. And then you can see how E. coli does its work, and the wintergreen taste is released. And on the next slide, please. The approach is that we create our plasmid. And as you can see, it's one promoter that starts, that kicks off the wintergreen producing enzyme, and then also the gene that goes into destroy gingivalis. In the illustration on the side, you can see it. E. coli Nissl detects gingipane, which is something that P. gingivalis creates. And then E. coli goes in to kill the gingivalis, and at the same time, like I said, releasing the wintergreen taste. So that is the synchronized response module. And if we go to the next slide, please. The mesh, what's this? Time's up. Sorry, I just heard. So measurement XSX is really that we can see that wintergreen production correlates with P. gingivalis killing. We also want to make sure that the taste is released in a timely manner. And we want to make sure that there is no significant impact on the beneficial oral bacteria, because again, a microbiome, there's so many that have to work together. The qual and the quant. So we also want to make sure, do people, is it too intense? Is it pleasant for them? And as you can see in the little illustrations there are many ways that we can validate our work. Ethics is important. So we also want to make sure that there's informed consent, that people could understand this is a GMO that they're ingesting. We want to make sure long-term safety, right? That we just make sure that E. coli, for example, that degrades. Usually in the mouth, when it gets ingested, it goes through us. The E. coli Nissl, this one. We know that we want to prevent serious health issues that are related to periodontal disease. So that's a good aspect in the ethics. And the last one, the justice one, I think that's been mentioned earlier as well, is we want to make sure that everyone can get access to this little lollipop or lozenge, because we don't want just people with money to have access to these tools, because again, it is a global disease. And that is it. To start with the oral microbiome, and eventually, if we could look at the whole global microbiome, human microbiome, and again, get interesting information for all of us. That is it.
Great. Thank you so much.
Thank you.
We are at your 5 minutes, unfortunately. I'm gonna ask you a quick question, but maybe you can answer it in the chat. You know, there's a couple of kind of technical, I think, challenges to think through. You know, you've got your teeth here, and you know, how are you making sure that you're delivering enough of these kind of therapeutic molecules, like, to the place that they're trying to go? Because if you're like sucking on a lozenge, you know, it's kind of going, how do you direct it to exactly the part that you want? And then you've got a bunch of different things going on, as far as I can tell. You've got kind of the creation of a scent. You've got the kind of therapeutic aspect. So just, you know, I just wonder what's your model system, how you can actually go and test some of that. So anyways, why don't you answer that in the chat? We have to keep moving on, but thanks so much for the great talk, and appreciate the great vision. So great work. David Vaca.
Yeah, can you hear me?
We can hear you.
Okay.
All right, you are up. Go for it.
Okay. Hello, everyone. My name is David, and I'm currently studying microbiology. And today I'm thankful for the opportunity to present my project in the optimization of kefir production and the development of a burn patch using it. And thanks. Next slide, please. Thank you. Kefir is a consortium of bacteria and yeast that ferments milk and sugar to create a probiotic beverage. However, in this case, I will specifically focus on the biofilm that forms within these grains that hold the consortia and is composed of an exopolysaccharide called kefiran. This compound has several properties, and the ones I highlight are its wound healing effect and its antimicrobial properties. And where would this be used? In burns, which are often related to infections by multi-drug resistant bacteria that are difficult to treat due to the biofilms. But what if through the application of this biofilm, we could prevent the formation of other biofilms? However, kefiran is only produced by Lactobacillus kefiran faciens, and this exopolysaccharide is not normally produced at high levels, since the bacteria seeks energy and this production is limited by signals from the consortium. Next, please. And that's why my first aim is to optimize the production of the lactobacillus through the activation of the enzymes in its metabolic pathway, where three main enzymes are involved: the phosphoglucomatase and the UDP glucose pyrophosphorylase, which are the one that direct the glucose into the EPS biosynthetic pathway, and the glucosyltransferase, which is the slowest one and is the responsible for actually synthesizing the EPS. I was able to find the sequences for two of these enzymes, and these are inhibited by the exopolysaccharide. And I identified nearby sites that act as suppressor of the promoter. And my idea was, through CRISPR-based editing, to silence these sites to increase the production by designing their guide RNAs. But now if I modify this, I might create an imbalance in the consortia. And that's why using mass spectrometry, I could characterize the kefir environment and its compounds, which I mainly expect to identify short-chain fatty acids, alcohols, and sugars in order to replicate this in a culture media. And once this is achieved, I would purify the kefiran, and due to the structure of the kefiran in its three hydroxyl groups, in an environment with citric acid, it can form a biodegradable matrix with PVA. And thanks to this property, I believe the best design would be the patch with a resistant adhesive layer, followed by a layer of PVA and the layer of PVA with kefiran, which will be solubilized and will provide the protection to the burn. And next, please. But going a little further, the next steps could be to carry out the necessary tests, both in animals and humans, to verify its safety and effectiveness. And as a third aim, in the future, if the creation of this product is possible, due to the metrics that is formed, I could explore the integration of a drug delivery function, and at the same time, improve the production of kefiran, maybe through cell-free synthesis, and once the entire metabolic pathway has been characterized. Thank you.
Okay, great job, David. Thank you so much. Some questions from anyone, students, TAs, who has a question for David on his project? Ben.
Great presentation, David. Thank you so much. I just have one question about the animal testing. Do you think maybe you can skip that part and not using animals and maybe using cellular models or something like that? I mean, I know that for some kind of treatments and testing is something that is neat, like normal steps. But maybe something less, I don't know. I don't like to use animals, so any other
Well, yeah, using cellular cellular tissues, maybe. We can like try to first see the Goon healing effect in cellular tissues instead of going to animals, and then we can just jump to the clinical trials, maybe. I don't know.
Yeah, David, there's different kinds of, organ on a chip, skin on a chip, these different types of in vitro systems that you can probably do some of that type of exploration with. But David, thank you so much for the great presentation and the great work. Really appreciate you. Yeah, great job. Andrea.
Good evening.
Okay.
Hello.
You're up.
Hi. Well, I start. Hello, everyone. My name is Andrea Tambo, and I'm present the project who is called Cracking the Silence. Mir21 detection. Next, please. Well, imagine your kidney, a pair of silent heroes, working 24/7 to filter your blood, remove toxin, and balance your body's internal chemistry. But what happens when they begin to fail? Quietly? Chronic kidney disease is a global health crisis that affects over 850 million people. What makes it dangerous is not just a prevalence, it's the silence. It often progresses undetected until it's too late, because you don't have the, or maybe have bad habits like non-stop smoking at all, you consume bad or fast food, and well, what if the solution was already following through your veins? Or maybe what is the science could break the silence? So this is the project that is begins. So next, please. I develop a biosensor that can detect early molecular signs of kidney damage, specifically microRNA found in patients in blood samples and the plasma specifically. These tiny molecules are emerging biomarkers. So the first step is design the biosensor, but it's not the typical biosensor into a bacteria. So I designed a structural backbone for the first strand. Then I designed the strand B, who is the detection probe, and the strand C, who is the patient microRNA. So I use RNA co-fold and RNA fold to simulate and predict the interaction. The first one is very stable. The minimum free energy is -30. And by the other hand, you can see the blocker it's in an inactive state, who is ideal to make specifically with the microRNA for the patient. So next, please. You can see the other interaction, ABC interaction. So these strands have this distinct Y shape with combined free energy. So this predicted model is really useful for develop the biosensor. So the future for this project is be selective, programmable, and modular biosensor, changing just one sequence and target different microRNAs. So this is a promise that early diagnostic can be simple, fast, and accessible. Thank you so much.
Andrea, great job. Really, really lovely presentation. I can tell you put a lot of time and effort into practicing it, and really shows. Great job. Any questions for Andrea? I have a question. Can you talk a little bit about the sensitivity of your biosensor? You know, what kind of detection limits are necessary, and what kind of technical advances do you think, to meet a good detection level?
Well, first of all, I think the in vitro proofs that are the key, because the most interesting thing is how the affinity, and we can measure the concentration. After that, we can prove in the plasma, because this is very sensitive, but they can be used or maybe have other interferences. Like in the plasma, we have a lot of other molecules. So this short sequence can be, will make an standard, right? So we can prove first in vitro in the laboratory, and then we can prove and change the sequence. But well, we can study a little bit more.
Yeah, that's great. But it's sort of, I appreciate what you're saying. It's sort of sensitivity, but also specificity. That's what you're bringing.
Yeah.
Which is wonderful. Yeah, great job. And again, I love the metaphors, and the presentation is fantastic. Great job.
Thank you so much.
Okay, we're in our last 3 presentations, everyone. Can you believe this? It is crazy. It's almost getting dark here in Boston now. I got up when the sun was coming up, and the sun is almost going down. Okay, our last 3 presentations. Piyush, I hope I'm pronouncing your name okay.
Yeah, that's right. I'm Piyush Acharya from the Great Pacific Northwest. I'm a junior in high school, and today I'm here to present my project on building a new transformer architecture for long COVID and myalgic encephalomyelitis chronic fatigue syndrome. Next slide, please. All right, so the problem here, so we have 2 conditions, right? We have long COVID and we have ME/CFS. These 2 conditions have stark overlaps, strong overlaps in their symptoms, as we can see here in this bar chart. There's a lot of overlap. For example, both long COVID and ME/CFS patients experience fatigue, post-exertional malaise, and cognitive dysfunction. This leads to around 90% of ME/CFS patients being misclassified as long COVID patients. And collectively, these 2 conditions affect up to 470 million people around the world. Imagine suffering from a chronic, debilitating condition, going from doctor to doctor to doctor for years, only to be told that the horrible symptoms you're experiencing might just be in your head. This isn't just a reality. This isn't just a hypothetical. This represents reality for the 470 million people who have these conditions. So what my approach here was is to collect 852 blood samples from the NCBI's Gene Expression Omnibus and employ a complex transformer on top of it. So we're using the data input here is those 852 individuals, and it comes from Illumina's 450,000 and 850,000 arrays. On top of the original, so I understand that our audience here isn't like super technical. I wouldn't like. I think that you guys are more on the bio side here. So let me just quickly cover what a transformer is, just like super quick. So all of you guys have probably used ChatGPT at this point. So the T in GPT stands for transformer. So the transformer architecture was originally used for large language models, for language. What I did here, along with Dr. Derek Jacobi from the Victoria Makerspace Lab, is we're applying that architecture with 3 key advancements to epigenetic data. So looking at our methylome here. Next slide, please. So our goal here was, can we use a new state-of-the-art epigenetics transformer and our epigenetics to make diagnostics more accurate, more precise, faster, and also figure out what truly causes long COVID and ME/CFS, and generally all post-viral conditions in general. So that's the second part of the mechanistic pathway. So this here represents generally how our workflow works. We have blood sample input, so a patient would take a blood test. We would process it using the Illumina 450K array. We would feed it into the transformer, and it would tell you what A, like what condition you have, also B, like what conditions and what CPGs, what genes are correlated with your condition there. So that's one of the benefits of using the transformer architecture over, say, something like a random forest architecture. So next slide, please. Closing on impact here, previous standard models. So baseline accuracy currently in clinical practice is like around something like 30%, 30 to 50%, because like I said, there's significant overlap in the symptoms of these two conditions. Using standard traditional machine learning, we were able to get it up to 80%, but then when we use the transformer, which has, again, 3 key advancements of mixture of experts architecture, which DeepSeek uses, and self-supervised mass pre-training, and also adaptive computation time mechanism. We were able to achieve 97% accuracy on those 850 samples. We had an external holdout test set, which demonstrates that, hey, this is like really true. And yeah, and you can see here that generally, like, our model performs significantly better than prior clinical tools and also traditional models. So that is our paper here. Thank you.
Okay. And did I hear you say so you're in high school?
I'm a junior in high school. Yeah.
You're a junior in high school. Okay, great. I'm not sure what I was doing when I was a junior in high school, but not this. Fantastic job again. Really, really, really great. Any quick questions for Piyush? I have one quick question. Again, I think I'm following you with these different models that you're describing. How would you envision this actually being used in a clinical setting? Like if I were actually somebody that was concerned that I had long COVID, what would happen?
Yeah, so if you think you have symptoms of long COVID or ME/CFS, you would take a blood test. The clinician would process it using the 850K array, and then we would give them the results back immediately by using the data from the blood sample. And this would typically be completed within like 24 to 48 hours, which is like insane timing for all of this. And you're getting really, really accurate results. And you're also getting to know like what causes it. So doctors, you're not doing just like a binary classification here. You're understanding what in your epigenome causes your condition.
Okay. we'll leave it there for now, but
Thanks for the
fantastic
question,
job.
David.
No, thank you for the great work and looking forward to seeing this in a clinic someplace, hopefully soon. Thank you so much. Fantastic job. All right, our last couple presentations. Teddy. Is Teddy in the building?
I'm here.
There he is.
Can I
A
start?
little fun story before I let Teddy go. Teddy and I sang in the same a cappella group in college.
We did.
Not at the same time, but the MIT Logarithms. That's why I know Teddy. Now Teddy's in How to Grow Almost Anything. I love it. Teddy, the floor is yours, sir.
All right. So I'm Teddy Toussaint, and I'm presenting a plaque assay on a chip. So our wonderful TAs simply don't have the bandwidth to test all of our MS2 phage designs from week 5. This inspired me to create plaque assay on a chip. First, I aim to design, fabricate, and test a fluidic chip that automates a plaque assay, characterizing the infectivity of bacteriophages. Second, I aim to provide a chip that to how to grow TAs in future classes, to provide that chip to how to grow TAs in future classes. Third, I aim to expand beyond the plaque assay and provide chips that help increase scientific productivity in a broader context. And so on this slide, you can see there are several biological samples of materials to prepare in a plaque assay, as you can see on the left. Then one has to perform a serodilution, so you can incubate cultures of bacteria and phage at serially smaller concentrations of phage. The serodilution is a time-intensive step that I've automated with plaque assay on a chip for the benefits you can read on the right. So now we can go to the next slide. And so this is the chip. And so David, if you can just like click every quarter of a second, I'm just gonna talk through voice over the sort of animation that comes up.
All right.
So yeah, that's the chip, and you can see the different chambers. And it's got the intakes and outputs. But what generally needs to happen, and it's got heaters, by the way, so it can do incubation. What generally needs to happen is we need to load the chip with the material and heat the soft agar in the heater zone there. We will move agar to the different chambers. That's the agar that's going to form the bottom layer on the Petri dishes. And then we sanitize. We then do the first mix of the 10 to the -1 phage. We store it, we cache some of that because we're going to need to inject that back into the second mix of phage. We sanitize the line, which discards the remainder of the 10 to the -1. We mix into the 10 to the -2. So we put 1 millimeter into 9 millimeters to make 10 to the -2. We cache that. We can keep on clicking more quickly. And then you can see that we keep doing that through 10 to the -4, -5. And then once we get to -6, we mix it, we cache some of it, and then we move the rest of it to the adsorption chamber, the 10 to the -6 adsorption chamber. The same thing happens, process repeats for -7. Then -8. -9, and -10. And so we heat up the adsorption chamber to incubate. We move that over to the Petri dish, and then we let that incubate for 16 to 24 hours. That's what a plaque assay looks like, that image that just flashed before you. So those little plaques represent virus-killing lysine, the bacteria, which this is not my result, but a result that I pulled because I have not actually run this yet. But yeah, that's how the chip works. So if we can go to the next slide. Thank you. So this graph was published by Gordon Moore in 1965, and it predicted that every 2 years we double the number of transistors on a microprocessor. In the next image, we see that Moore's law has held to this day. In the next image, we see Eroom's law, the reverse of Moore's law, showing the increasing cost of new FDA-approved drugs. It's like it gets, yeah, we can go more into that a little bit. In the next image, we have the game Snake, developed in 1976, when there were 10,000 transistors on a chip. In the next image, we have Call of Duty, developed today when there are tens of billions of transistors on a chip. So what would it be like if we tried to develop Call of Duty with 10,000 transistors on a microprocessor? What would it cost to develop any new game or computer application of this complexity? Would we reserve whole floors and buildings to develop new applications? And in this, as you can see in this image, would these buildings be left empty when we misjudge demand? My point here is that Moore's law isn't a law of physics, it's an economic mandate for the semiconductor industry. And in the next image, I propose an economic mandate for the life science industry, Moore's lab. Every 2 years, we should double the amount of simultaneous experiments of a given kind that we can do on a chip. I think this would end the Silicon Age and start the age of living machines.
Okay, Teddy, awesome job. We're over time, but I will appreciate all of the storytelling. This is awesome. For your actual device, you showed a version of this. Was that laser cut like acrylic, or and how are you?
Yeah, that
pushing
was
it?
laser cut. Yeah,
Is that
so
is this intended to be pressure based or?
yes, yeah. So it
And
is
then-
laser cut. It is
Okay.
acrylic, and it is pressure based right now. Basically, we just need to hook up pumps to the intakes and outtakes, which were labeled in one of those images.
Okay. I mean, I'll say one thing, just, as a microfluidics guy myself, like, I love the vision. One great thing about microfluidics is, for this type of system, it's a plaque assay, right? You're trying to get a dilution series and then kind of figuring out the response based on the dilution. So this is a classic data in, data out situation. So you're not collecting anything from the chip. So you actually don't need a huge thing. Like it could be literally like a pixel, just like what you're describing, and you could do, very fine resolution, concentration gradients on the surface. So, but anyways, I love the ideas that you're talking about here. And Adrian, I know you got a question, but you can ask Teddy in the chat. We'll continue the conversation at the chat if that's okay. But great job, Teddy.
Yeah, thank you.
We can sing some acapella songs for How to Grow at some later point.
Let's do it.
Yeah, we'll do that. Actually, Ren sings acapella too, right? Ren is a part of the Stanford Harmonics, I think, actually. So she's one of the TAs. So I don't know who else sings acapella in How to Grow. I'm sure there's at least one more. We can do a quartet. All right, great job,
we can get
Teddy.
a quartet for the final picture.
Yeah, that's right. All right, our final presentation. Vanessa, are you here?
Yes, I'm here.
Where is Vanessa?
Okay.
I don't see you, Vanessa. Do you have your video turned on?
Sorry, no, my camera is not working. I'm so sorry, I cannot turn it on.
Okay, no worries. No worries. Then go ahead. The floor is yours. Let's start the timer. Go ahead.
Okay, thank you very much. So, can you pass to the next slide, please? Yeah, sorry, I think the animation was not working, so I just pasted, so I'm so sorry about that. But I will talk about my project, which includes the disease that's called cystic fibrosis. Cystic fibrosis is a separate genetic disease that affects thousands of people. What happens in cystic fibrosis? Normally, we have the chloride channel that can equilibrates the amount of sodium that you have, sodium and chloride that you have in your lungs and also in other tissues of your body. When you don't have, when you have cystic fibrosis type 1, the channel does not come because the mRNA has a truncated protein which doesn't allow it to reach the membrane. And you have various symptoms, including the typical ones that are a lot of lung infections and also gastrointestinal problems. So most of the difficult cases to treat are caused by nonsense mutations, and these mutations introduce premature stop codons, basically preventing, as I mentioned, the full CFTR protein from reaching the membrane, and you don't have effective treatments or options for these patients. And my project explores an RNA-based therapeutic strategy to address this by using anticodon edited transfer RNAs that are especially engineered molecules that can recognize and read through premature stop codons, allowing the cell to produce a complete functional CFTR without altering the DNA. And what I did was check the ACETR RNA library And using that, I projected to do clone it by Golden Gate assembly and evaluate if the stop codon suppression is reported on an essay that it will be using a luciferase-based reported program, a system, sorry, if the tRNA successfully suppresses the stop codon, the cell lights up, literally thank you to the luminescence from the NanoLuc enzyme, and then we can suggest that it is actually reaching doing the assessment for the full length of the CFTR. And this is previous data that had been reported on how to do these kind of designs. And if everything is correct, then the second aim should be to apply it and cells that carry these nonsense mutations, especially W1098X, that we have found in my previous work that it is quite common in the country that I'm from, that is Ecuador. And if, check if the full-length CFTR protein is restored and functional. And the third part will be to groundwork for future RNA-based therapies by designing delivery strategies using either viral vectors or nanoparticles. And this project can be quite innovative because of the use in synthetic biology and also the use of Golden Gate cloning, and to rapidly generate and test the ACE tRNAs. And this can be, this approach offers precision, flexibility, and minimal genomic disruption. And thank you very much.
Okay, big round of applause, and thank you so much to Vanessa. Final questions for Vanessa on her project. Anybody like to chime in? Vanessa, one question for me. I'm curious, what is your experimental plan in terms of the first things that you're going to want to do in the lab? Have you been able to try any of this work yet in the lab, or are you, what are the first things that you're going to work on?
Yes, that will be the first things to do on the lab. What I have done until now is just the design of the ACT and the cloning, just designing how it will be on the Golden Gate assembly. And that will be the initial part.
Ben, go ahead.
Great. Thanks so much, Vanessa, for your work. It was really interesting. I want to ask you about the therapeutic itself, because it says right that you're going to use the tRNA. So how you're going to ensure that only the therapy will go to the cells that are sick, that have the mutation or that need this specific change, because they could go all of other parts, right? Yeah, that part. Thanks so much.
So from a previous study, it has been reported that actually it doesn't interfere with other sections that are not the ones that are related to truncated protein. So it actually goes to the ones that are having this part of truncated. So it reaches a specific and a specific area. The second part will be either, since as I mentioned, most of the classical symptoms are on the lungs. We can do it like just I forgot how to say that in English, but just like a breathable thing for the people with asthma. So that can be like a direct way of just localizing it towards that area.
Okay, Vanessa. Thank you so much
Later. Thank you very much. Sorry.
For the response. That was great. And everybody, let us give a big, huge round of applause for everyone that has presented over the past 5.5 hours, and also for everybody that's presented over this entire crazy day. Literally, the sun is going down here. I literally got up in the morning with the sun, and it's been, I don't even know how many hours of presentations, but it has been a lot. What an incredible day. I am so proud of every one of you. Really just phenomenal job. It's been such a journey. I know for each one of you, all of your projects, so much hard work, so much thoughtfulness and care that's gone into each of your projects. And maybe we can all give another big, huge round of applause, and thanks to all the TAs, all of the TAs from every single node, everyone that has done so much great work. So grateful to every one of you for all of your leadership, all of your great mentorship and support for all the students. I know it's been so meaningful for the students. It's been so meaningful for me. And, you know, again, I think your leadership as TAs, you know, I cannot emphasize enough how much How to Grow would not be possible without all of you. For those of you who don't know, you know, we have a TA meeting every Monday at 6 o'clock Eastern time, and we actually meet year round. It actually does not stop. We maybe take like a week off. I don't even know when we take a week off, but How to Grow is a full-on year-long experience just to get everything set up and ready. This year is the first time we set up this node process. And, you know, we had nodes before, but organizing the course so that the students were all getting the peer-based learning that happens in the context of nodes was a real big innovation for us this year that made, I think, a huge difference in terms of the fellowship and the connection. You know, I see all of the different chats in the WhatsApp groups, and it's just awesome, you know, seeing all of the different groups getting so excited and cheering everybody on. I mean, it's really, really phenomenal. So just want to really acknowledge everybody for all of your amazing work this whole semester. And as I said earlier, you know, this is not over by any means. For all of you global listeners, you still have 2 more weeks of work to do. So, so this was a big moment for all of you in your presentations. But I know there's still some lab work that's being done for some of you, and some orders that are still coming in. So we're really looking forward to your completed documentation in 2 weeks. And as we've shared earlier, we're going to do spend another 2 weeks and then do grading. And then we'll let you all know everybody who's able to qualify and complete a certificate. And then we'll do a special graduation ceremony for everybody that is able to get a certificate done. So, so that's basically what's going to unfold. As I mentioned earlier, I'm going to send out a survey that's going to go out a little bit later today. And it's also going to be a requirement for all of you to complete the survey. But for the listeners, you all have 2 weeks to do it. But if you get it done sooner, that's great as well. And part of that's going to involve expressing your interest to continue on with some of the group projects, the network-wide projects, like the plaque assay phage therapy project, which Teddy referenced, like the mammalian neuromorphic circuit project, which I think many of you did not have a chance to work on, but that we're going to set up a process to be able to work on over the summer. The Cloud Lab project with Ginkgo Bioworks is another thing we're going to work on over the summer. So there'll be a lot of opportunities to continue working on those projects. And then, of course, you know, we would love for you to join on as a TA. I think that's a big part of the journey for so many folks here is, you know, you come on, you have a great experience as a student, and then you join as an instructor. So we would love to continue working with you all and in those capacities in the future. And for now, just really, really, really deep gratitude. And you all should be so, so proud. Just huge congratulations again to everyone for all of the great work. So one more big round of applause and thank you to everybody for all of the fantastic hard work, all the creativity and the energy and the great spirit. It's been so phenomenal. And yeah, we can do one more last picture. If we for those that have missed some of the other ones, we've done 2 other ones. We can do one final group photo as well before we wrap up here. So just like the last photos, what we did was we all got an object. If there was an object handy that you thought was a good one, you could bring into the photo that, you know, connected to you. And oh, that's a good one. I like Escalite. You've got a nice flower there. Yes, anybody has some good artifacts you can bring into your photograph. I've got my Star Wars mugs. I wish I had my cat. I feel jealous of everybody that's got their cats with them. See, Adrian's got the cat. Oh, the cat's not going to cooperate though. We'll see if the cat cooperates. Cat looks very squirmy, Adrian. I don't think the cat's going to make it. Let's see what we can do. Everybody, here we go. 1, 2, 3. Oh, great. And hold on, let me move to the next screen. 1, 2, 3. Oh, that's great. And the last screen here. 1, 2, 3. Hey, everyone looks so good. Awesome, everyone. Thank you so much again. You know, well, I'm curious actually, do any of the TAs want to say anything while we have everybody here? We can have a few closing comments from any of the TAs who'd like to share anything before we wrap up. Again, during the graduation, we're going to do that's going to be a whole big longer thing with appreciations, and we'll have a proper celebration there as well. But it's good to take a moment and celebrate everybody here right now. Anything any of the TAs would like to share? Yeah, Dominica, go ahead.
Oh, okay. Yeah. Amazing final project. It was so interesting to see how different and varied and diverse all these projects can get. So many things I haven't thought about myself ever in my life. And it was such entertainment. Like I was glued to the screen. Like I felt like I was missing out whenever I had to fetch something. So yeah, great work. I feel it's getting better and better every year. Like I've been doing this for a while. So like it's fantastic to see like how many people we could squeeze in into one day. And then also to have everyone engaged over the whole period, over the whole day, and have such a vivid discussion. This is, I think, truly remarkable. And like, yeah, the growth of this whole course is truly amazing to see. And I think it's also like thanks to you, David, for like facilitating it and taking it to the next level every year. Like I've learned so much from like how you have started this project. It's like something like kind of leger, like chill, and like taking it up to add more and more complexity. And it's like such a good strategy and like so well thought out. So yeah, great stuff. Like it's just it just keeps getting better and more exciting. So, and I want to see those projects in real life. I will buy your products if you bring it to market. Just like, you know,
No, thank you.
just get it there.
Thank you, Dom. And Dominica does have a really unique perspective. What year were you a student, Dominica? It's been years ago now. It's like 2019, it was before the pandemic, right?
It was
2020 maybe?
at the pandemic, like 2020, I think.
Yeah,
It's
yeah.
it's 2020 or 21. I don't remember.
Yeah.
It was like in the pandemic where I had nothing to do, where I was like, I want to do science, but like everyone is closed down. I have no job. And like I was in the middle between finishing my master's and like going to my PhD. So like this was the lifesaver. Yeah. Yeah, it's
Dominica,
amazing to see the growth.
You're officially a How to Grow veteran, and you have seen, I mean, the last 5 years has been a huge transformation for the course. And what's crazy is we're at an exponential right now. So I mean, whatever this is, and as amazing as this is, it's going to get more amazing and even crazier. I mean, it feels already so crazy, but I think it's going to continue to get crazier. So, so we could not do it without you. And Dominica, thank you for all of your leadership. Anybody else want to share anything before we wrap up? Yeah, Danny, go ahead.
Yeah, so thanks, David, and thanks everyone. I think it's a very wonderful journey. Actually, I joined since 2016, and only like a 6 or 8 global student at that time. But now it's grown very fast. And I remember last year's only I worked with Digby, just one global members in London, but now I have so many members in LifeFab. So I think it's a very new beginning, and I think today is the end of the new beginning. So looking forward for a next generation and new courses in the future. Thank you so much, David, and thank everyone. Happy to work with everyone. Thank you.
Thank you, Danny, so much. Yeah, and Danny, you're right. Danny's been with us since the very, very beginning. So you've seen the whole kind of arc of everything that's happened over the years. And yeah, just shout outs again to all of the nodes. You know, it's been so cool watching LifeFabs evolve and, you know, just seeing the growth of your space and seeing all the amazing work of the students. And, you know, I think one of the really beautiful things about How to Grow as it relates to the nodes, you know, each of the nodes, you know, I know all of you have grown in what your capacities are and having all of the students bring life to your spaces. And so that's something that, you know, again, obviously we're going to continue to grow the network. I like to say we move at the speed of care and we move it at the speed of trust. So you want to grow, but you always want to grow with really strong leadership. And I think that's one of the key parts of the success of the nodes is that each of the nodes has really, really strong leadership, which is something that we really look for as we expand. So thank you, Danny. Any other comments from folks? Yeah, Ice.
Yeah, I just want to say that like you guys are doing so great. It's not easy at all. How to Grow has a lot of, you know, difficult classes that it takes like a lot of effort for all of you to go through it. So like please be proud of yourself. And like if you are taking the class from, you know, global setup right now, just keep in mind that like there's a chance that you can just like go to the in-person and like work together. Like when you're some of you go to the Life Lab with Denny, and like I was a global student just like you guys last year, and now I'm here like getting to help with the in-person stuff at MIT. So please like, you know, aim high and like keep working. Great job.
Keep dreaming. Love it. Ben, to you.
Yeah, thanks so much. I just want to say thanks to everyone, all of the TAs and all of the students. from our node. This was a really cool year. We have learned a lot from the node students, from the other nodes, from the TAs, and yeah, it has been really a fantastic year this year. And yeah, thanks so much for this opportunity and for all of the learning and for all of the cool projects and all the learning. Yeah, thanks so much.
Ben, thank you for your leadership. Again, just such an amazing job working with all of the Latin American students and everything you've been doing in Quito. So thank you so much for all of your hard work. You've done an amazing job, and thanks for all the great questions today as well. There's an art to asking a good question, and you ask extremely good questions. So thank you very much for that. Anyone else, and actually any students too, if anybody wants to share anything before we wrap up, we'll open it up for anybody that would like to share anything before we
could I say something?
Yeah, Richard, go for it.
So I am very happy to have gone through this. This is one of those experiences that can never leave you the same. They actually change you completely. And for a person who comes from an area where most of these projects, I've literally been here the whole time because I was amazed at the amount of projects that were there. And for a person that comes from an area where most of these projects are actually applicable, I really appreciate the work. So thank you so much, David, for the work that you do. And thank you for the whole team and TAs and everyone else. We look forward to a very great future in the next few years. Thank you so much.
Well, Richard, we need to work together and set up a node in Kenya. One
Yes,
of our glaring problems right now is that we don't have a good node in Africa, and we have colleagues there, but we haven't set up a good one yet. So we should work with you to set one up.
That's the next project. Thank you so much.
That's the next one. Yeah. Great, Richard. Thank you so much for your comments. Anybody else would like to share anything? And you'll have another chance to speak during graduation. So if you're feeling a little shy now, you can also speak later. But anybody else would like to share anything before we wrap up?
Well, I'd like to thank you, everybody, for, basically this course. It's a fantastic journey. Basically, it was the opportunity for me to, meet all the great, names that make part of the synthetic biology founding groups. It was an opportunity to listen to invited lectures about, teams as diverse as, genome chip and synthetic biology, all different areas. And it was a great opportunity to basically, get up to speed with what is actually being done in the field. And that's a field I would like to specialize further. So it's a great opportunity to get back to speed.
Wonderful. Thank you, Federico. Anyone else want to offer anything before we close? There's still a little bit of daylight out here in Boston.
Yeah, I kind of want to say something, David.
Yeah, go ahead, Jenny. Jenny, go for it.
Yeah, so I feel like this course totally opened up a new universe for me as I'm not from the biology field. And like I've learned so much, and I feel like in the end, when it comes to the end, like I evolved to a better person. So I really enjoy the process. Yeah. I hope like this course, continues on and on, so more people get benefited from the course.
Oh, that's so kind of you to say, Jenny. And we hope that you stay a part of it and help grow it with us. That's how we do this. We do it together. So thank you so much for your comments. All right, Trevor and Tanisha, go for it, Trevor.
Yes. Hey. Yeah, I wanted to appreciate and show thanks to everybody at the course. I know it's a lot of work and there's a lot of students this year. I think the curriculum was very well designed. So I joined the course. And I think what you're going to learn is the impacts of the butterfly effects of people who are taking this course escape the things that you can possibly imagine. So I picked up some new tools and ideas and things that will help me in the process of trying to cure my own disease. And I'm setting up a lab for that here in Boston. It's good to know all this new tools and tricks. So thank you for opening my eyes to some of this.
Thank you so much for your words, Trevor. And I will say to that butterfly effect part, one thing that I've seen now over the years, which has been so fun, like I'll be in just the randomest places and I'll meet people and be like, hey, I'm actually a student in how to grow. I'll be like, I had no idea. You know, you meet people that take this class. I mean, there's now like thousands of us that have been a part of this course, you know, over the past years. And so you meet all kinds of, you know, folks in all kinds of strange places. And there's something powerful about having a shared experience together. And, you know, again, I think one of the key parts about this course, it's not a MOOC, right? You're not like learning one-on-one with a computer or with like an AI agent or whatever. It's relational, right? We all here, you're actually meeting real people from all across the world that care about science, that care about biology, and want to imagine a better world and use these tools to do impactful things. So, so these relationships matter a lot right now in this world. So thank you for saying that, Trevor. Tanisha.
Yeah, I guess what I would like to say is like back in school, I was always like interested in both science and creativity, but then you usually have to choose one. And I always hated the idea that you need to use one and like sacrifice the rest. I did end up choosing to pursue design as a career, but then I always missed science. You know, I was like, I want to go back, I want to do that too. But usually there's a barrier to entry. Like if I want to say I want to study biotechnology, the last time I studied bio was back in grade 10. So they're going to be like, why would you do that? You have no background. But I really love the way that this course does not like assume or like put any barriers that, hey, if you did not study bio or you don't have grades back in high school or college, you can't come. The fact that I could come here and I could actually study and do like a final project was like insane, because that's exactly the kind of thing I was looking forward to. But I don't think it's there anywhere else other than here. So I really like appreciate this platform a lot, because now that I've done this, I can like go forward and do more, because I have something to fall back on and say that, look, I did this course, I know bio. And I think the other thing is a bit personal to me, but I remember when this course came out, I was so excited. I told my best friends about it, and they were like, oh, okay, let's do this together. And the last time we studied was back in school, like 3-4 years back. And I remember when the lectures would run, we would actually be on a meet, and the lecture would run, and we would be talking and discussing together. So it was like very back to school. We were classmates again, and we were like, that's crazy. We're classmates doing this thing completely unrelated as a side quest. And it was one of like the best times we ever had. So yeah, I have very good memories from this, and thank you so much.
Thank you so much, Tanisha. And thank you all again. You know, I love that. The side quest sometimes becomes the main quest, so you have to be careful about that. But everyone, it's been such a joy working with you all. Again, this is, Danny, I like what you said. This isn't the end. This is actually just the end of the beginning. So there's much more to come. We'll all be active on the Slack. And again, I know for the nodes, you're also working together and supporting during this documentation process and wrapping up experiments and so on. So, so we look forward to your continued great work over the next 2 weeks. Keep a lookout for that survey that's going to come out. We'd love your feedback. We love all of your thoughts and your kind of pluses and deltas, so that we can get all of your ideas for how to make this course even better for the next time. So thank you all so much again. Wherever you are in the world, either have a great breakfast, a great morning, a great afternoon, a great evening. But appreciate you all so much, and see you all again very soon. So have a good night, everyone. Take care. Appreciate you all. Bye-bye.
Bye, everyone.
Bye, everybody.
Bye,
Thanks, everyone.
everyone.
Bye.
Bye.
Bye-bye. Take care.
Bye-bye. Thank you so much. Bye-bye.
Thanks so much, everyone. Bye-bye. Congratulations. Take care.
Bye-bye.
Bye-bye.
Thanks, David. Bye-bye.
Bye-bye. Thank you.
Bye-bye.